Let me share the screen. I'm actually based in Hong Kong.
Hong Kong, yeah, you're right.
Can you see the presentation?
Yes.
Right. So my name is Alexey Reshchikov, and as I said, I'm based in Hong Kong University. And it's less about taxonomy, but about shout out about networking. And I work with Darwin wasps all my research life, and I studied these beautiful wasps. Several years ago, I was working in mainland China, and I faced difficulties accessing nature for the fieldwork. That was due to specific of the land. China is the country of fences, and also that's a specific of the academy there. To get funded for the field work, one needs to perform some significant results already for the topic. At that time, I immersed myself in the tea culture, and while drinking tea, I decided to go to farmers and ask them for help. That's how the initiative project Tea Fauna began. Tea species of the genus Camellium has their range of distribution from eastern Himalaya to southern China that perfectly match the scope of my entomological interests. This how the landscape looks like in northern Thailand. And this is how the wild picking looks like. Many ethnic groups, the so-called hill tribes, found their home in the region during 20th century and current century, and it goes by the civil wars in China and then in Myanmar. These people are picking tea from the forest, and the main tea product they produce is called miang. This is fermented tea leaves, basically, that's used for food, not for the drink. And this is how the mass monocrop tea plantations looks like anywhere in the world. Summing this, I decided to target biodiversity gradient of the family Ichneumonidae in different tea habitats. And fortunately, I've met this tea company based in Chiang Mai, northern Thailand, called Monsoon Tea. They developed concept of forest-friendly tea. When the product harvested from the forest and the farmers, they supported by consumers to keep their lifestyle. So simply saying, if farmers are paid for harvest from forest, they are not switching to cash crops, for example, corn, which is broadly everywhere, and it's base for the meat industry. We establish long-term sampling up north in the country with the MLS traps supported by farmers. And that's resulted in rather representative Ichneumonidae collection and discovery of several new species and many new records. And good number of datasets. This is INET released part of the project with significant number of observations. If anyone in the platform, please check the project and identify some of their groups there. So, since many challenges facing biodiversity in northern Thailand, as elsewhere in tropics, there is a need in such a tight collaboration of local communities and businesses and the academy. Even we have some good achievements with recent samplings. The Monsoon decided to go beyond and they established biodiversity traceability innovation project that's funded by GSMA, which is Global Organization for Mobile Technology. So the project aims to share and use data for all players. The farmers who empowered with this data for sustainable agriculture methods, and the company that uses this data for marketing and its development, and consumers that's navigating through their purchasing decisions, and the forest, which benefits from reforestation projects and expansion of the production. Later is the sustainable alternative to lease of the land for logging or cash crops, which is sadly modern reality for the region. So this scheme is based on this grading scale. Forest-friendly farming is exactly what it sounds like. Agricultural products grown and harvested with sustainable methods while using uh, while protecting the forest. And the next step of this would be using the accumulated expertise and transforming farming practices elsewhere in Thailand in tea production or other crops, or even out of the country in other places. And the scale is depicted here based on modern methods of biodiversity assessment and the base for the income distribution. So then the higher the biodiversity in the place, the higher the price for the products and the higher the farmer's income. That's main idea. Uh, there are following components for the scale realization. So the remote sensors and apps for both farmers and company, and database and information about origin of the products from different places and biodiversity there. The Monsoon use bioacoustic sensors to collect sounds from forest where the tea is growing. And GIS is another source of data helping determine plots and in the area and assign scores for them. So then they use AI for identifying the species and GIS for assigning scores for the land. And algorithms, they use ecological models for creating the plans, for example, for reforestation or for the current use of available land. And another thing is app, and app is developed for all the players, for farmers, for the company, and for the customers. And data obtained during the project coming, like helping to reward farmers with biodiversity services they provide and with their help. And the consumers, they navigate through their purchasing choice. Another thing that they're doing is community training. It's very useful to train and to introduce local community members how to help and how to use devices. So then that's basically everything, and you are welcome to participate if anybody is keen for collaboration. The company is open for sharing their data and samples, and providing logistics for help for other biodiversity research projects in Thailand or elsewhere in the world where tea is growing. So if anyone interested, please contact me by mail or so. That's pretty much everything. Thank you.
Thank you, Alexey. We have plenty of time for questions. So if you got questions also online for Alexey, please, now is the best time.
Question.
Yes.
Where are the voucher specimens being deposited?
Did you hear it, Alexey?
Right. With the type specimens, they distributed in the major museums. So we described new species, and the types are or in European museums like Swedish Museum of Natural History. In Hong Kong, we have a museum here. In Hong Kong University, in Kunming Institution of Zoology. So that's the main museums where the types are now. And the main collection are in Hong Kong.
Susan?
Yes, is tea mosquito bug a problem?
I don't think he heard you.
Okay. Tea mosquito bug, is that a major problem?
Is tea mosquito bug a problem over there?
Could you call the Latin? Sorry, I don't know the common name.
Helopeltis.
Not the area that we work with. So that's quite good balanced, and no problem with the pests.
Any other question? We have one in the chat. One in the chat. Matt is, do you know of other companies doing similar things? How did you first get in touch with the company administrations?
I believe there are plenty of businesses nowadays, because this is the very important things, the most important things in life, that to go in the direction of sustainability and work in the direction of agroforestry. But exactly this came when the everything quite transparent, and the farmers supported directly, and their income dynamic and really linked to the biodiversity measurements. It's quite unique from my knowledge. And sorry, what was the second part of the question?
How did you
how did I get to the administration of the company? That was a pretty long way. I started to communicate with farmers in China and starting to get help from them. And then I decided to expand and trying different countries with tea production. And I was just asking people, where is any tea production in around northern Thailand? And then I within the tea community, the tea lovers. So they pointed me the shop which is belongs to the company, and I met with I had a Zoom with the owner, which is Kenneth Rimdahl. He is a Swedish guy but living in Thailand for many years, and he understand what I wanted. Oh, I wanted long-term monitoring, sampling with molest traps. And then he invited me to Chiang Mai. That time I spent pretty much time in Thailand. And I jumped to the plane and we went to the forest straight away from the airport.
Fantastic, Alexey. We're a little behind, so I'm going to stop the questions right now. We're going to take a little bit of time after during lunch to get back on track. So thank you, Alexey. Please, another round of applause for Alexey. Thank you, Alexey. I see you in Hong Kong.
Thank you. Yeah.
So now we have our next speaker also on Zoom from Italy. We got Daniele Sommaggio with Syrphidae, taxonomy, old problems, new solution, question marks. Please, Daniele, take it away.
It's going to take a second to get him set.
It's going to take a second. So I'm going to entertain you with something else. Hopefully.
Daniele, you should be able to share your screen now.
Are you hearing me?
Yes, we can hear you.
Yeah, okay. Thank you very much. And good morning to everybody. I'm Daniele Sommaggio and from Italy. And I'm working on overflies and Diptera more in general. I would like to thank you to give me the opportunity to be here with you to talk about overflies and Diptera. I think it's from Davide graph, it's the only presentation on overflies in this day dominated by hymenopterologists. So anyway, my presentation is totally different from the previous one because I would like to present the state of the art of overflies taxonomy at a level higher than genera. So overflies is a group of flies, of course. It's a family quite large, more than 6,000 species known. They are very nice, and so for this reason, they are largely studied in the past and also today, also because their biology is very interesting. And more recently, we have an increase in the research about overflies because several species play a key role in some ecological services, such as, for example, pollination or biological control. And in parallel with this interest in the biology and ecology of overflies, we have been also an increase of interest in the systematics of overflies. And so we can say that we have done a lot of road since the end of 18th century, when the great dipterologist Williston almost said that the taxonomy of overflies at a level higher than genera is simply very, very difficult and dramatic, and so he suggested to focus on species and genus level. So we have do a lot of road, and here I put all of the publication about the systematic of overflies at a higher level, starting from 1950. So the decision is not a coincidence to start from this year, because in 1952, there was a very inspiring paper by Goethe, who was the first one to suggest to divide the overflies into big subfamilies, Syrphidae, and his Milesina, now Eristaline. And in this timeline, I divided the paper according to their approach. In red, you have a more morphological approach, and in green, paper with a research with a molecular genomic approach. So what is evident from this graph is that you have a lot of publication from in the last 10 years about overflies taxonomy, but with a molecular approach. And this greatly increased our knowledge about the phylogeny of this group. Here we have one of the most complete phylogenetic tree. And what we can say today, what are the more shared points in this moment? The first point is that overflies is a good family. It's a good taxonomic group, were supported by several morphological characters, such as, for example, the spurious vein. and also confirmed by all the molecular data. Moving towards the subfamilies level, we can find that at the beginning of a tree, there is a well-defined group of species, almost 500-600 species, which is the outgroup of all the other overflies, and now is considered as a good subfamily, the Microdontinae. Curiously, this is a Myrmecophilus group of species. Another very good, clear taxonomic group, monophyletic, is Syrphidae, a subfamily yet recognized by Goethe. And this is larger subfamily, almost 1,700-800 species are known. What is interesting is that the larval morphology is quite common with biology, which is mainly zoophagous and in particular Aphidophagous larvae. Another small group of overflies, which is clearly monophyletic, is Pipizinae. This is a group of less than 200 species, so very small. What is interesting is that the adult morphology is more similar to Eristaline, but the larval biology is more similar to Syrphidae, and in fact, they are all Aphidophagous. Now, the molecular data confirm that Pipizinae is the outgroup of all the other Syrphidae. If you want to consider this as a good subfamily, and so calling Pipizinae, or a tribe inside the Syrphidae, it's only a decision where you want to cut the tree. Anyway, the other clear point is that Eristaline is not a good monophyletic group. So it's not, from a taxonomic point of view, a good subfamily. And this is not a simple point. This is the most critical point in the taxonomy of overflies, because in many cases we have no support for the division in tribes, as suggested also by other publications. So we have almost little idea on how to divide Eristaline. And this is a problem, because it's important to remember that the bulk of biodiversity are inside the Eristaline. 2/3 of all overflies belong to Eristaline. So this is a point that we need to solve. And how to solve now this problem? One solution may be to continue with more and more publication research with a molecular approach and improving technique and using more genes, or improving the species data set. But we think that there is also another possibility, and this is to moving towards an integrative taxonomy, using this at a higher level. So what is integrative taxonomy? It's very simple. It's to use all information we have from organisms to obtain, to create a phylogenetic tree. Now, integrative taxonomy is something trending today, as you can see in this graph created in ngraph viewer. But I think we are only using new words for something yet well known. This is a statement from Henning in 1966, and he is clearly talking about integrative taxonomy. Anyway, in overflies, the integrative taxonomy has been largely used, but mainly to solve critical species in some very difficult genera, such as, for example, Merodon, like in this paper, or Eumerus, or Cheilosia, and so on. Not everything is easy with the integrative taxonomy, and one of the most important problems, for me at least, is the character hierarchy. Here you have an example we have to decide in this publication if Merodon constants and Merodon analysis are the same species or different species. We have no support from other morphology or from molecular analysis. We have a partial support from distribution or from morphometrics of a wing. We have a small support from male genitalia, and so the author decide to consider the two species as separate species, giving clearly a great importance to the male genitalia. And Grimaldi and Hagen states that entomologists are unnaturally preoccupied with male genitalia. Anyway, what we have to do when we move with integrative taxonomy toward higher systematics at higher level. So we think that it's not simple to use old character and integrate them with new data from molecular. But we have also to obtain new information from old character. And I will give you two examples, and then I will finish my presentation. The first one is a well-known synapomorphosis of Eristalini, which is a good tribe inside Eristalina. And this shared character, is the presence of a patch of setulae at the base of the femora in all Eristalina species. And this is well known from several years, of course, but the question is, what are these setulae? What is their internal morphology? What is their function? We have no idea about this. And it's a coincidence that this structure, this hair, have a very clear distribution in the femora. We have no answer to all these questions. We only know that there are the setulae and nothing else. And also we have no idea if something similar is present in other group of overflies. Here you can see, for example, the setulae in Eristalini, short and large, very well distributed. And in Merodontini and Caelosini, there is something similar, but not so short. And for example, in Syrphidae, it's absolutely not present. And what is their function? We have no idea. And probably a function is present because in Sarcophagidae, with some Sarcophagidae, we have something similar, but in trochanter, not in femora. And so probably it's a different structure morphologically, but with the same similar position. So probably they have some function and the same in totally different families such as overflies and Sarcophagidae. And finally, another very interesting point is the vena spurius. Spurious vein is one of the characters that you find in almost all textbooks. You find that spurious vein is the most important synapomorphosis of overflies. And this is real, but what do we know about the function, for example, of the spurious vein, if any function is present? What is the effect of this structure during the fly of this insect? We have no idea. And another point is, what is the development of this structure? We have no idea. It's a simple coincidence that this fold, because spurious vein is not a vein, is a fold in the wing, which occupy a position which is at the border between anterior and posterior compartment in the developing wing of the Drosophila. So it's a coincidence that spurious vein develop in this position or not. We have really no idea about the function or the development or anything else about this character. So concluding, we think that it's now the time to populate the many interesting phylogenetic tree with all this information about all the characters, because only from this information, probably we can have the opportunity to really understand the evolutionary trend in all these fantastic insects and also in many other insects, probably also Hymenoptera. Thank you very much for your attention.
Thank you, Daniele. We got a couple of minutes of questions. If someone has some question for Daniele, even online. I want to just mention that Daniele is the, I would say, the leading syrphidologist in Italy right now. He's only in, he started very early in the game in Italy when Syrphidae were not really that trendy, and right now is developing the Syrphidae of Italy, which is the biggest platform right now for data on Syrphidae, thanks also to TaxonWorks. So it's a good chance to ask him questions. What do you think about, I have a question. What do you think about the recycling morphological characters that we see when we're proposing this sort of integrated taxonomy? Usually we have good molecular new data, but we have the same old 50, 60 years old morphological data that's been sort of recycled. Is that happening today?
In a publication, for example, there is a nice situation because they said that they use an integrative taxonomy approach, but when you look at the character, morphological character, it's simple, the morphological character of 15 years before only recycled for this publication. So I think it's not really a good integrative approach. For a good integrative approach, we need to have more and more character, but unfortunately, the morphology is not trendy today. So this is a problem, I think. We have a good opportunity of instruments, a good opportunity to study the structure of insects also at low level, but we have, it's not trendy, of course. And so you have, it's more easy to obtain publication in other way than with morphology. And you know very well, Davide.
Yeah, I agree. I agree. Thank you, Daniele. Thank you for the lovely presentation. And right now we have Joe Martinez. He's going to talk about the monotypic taxa, cryptic speciation, and their crucial role in shaping insect biodiversity and conservation. Joe, please. You got 15 minutes.
Need a second to screen share.
Okay. You have a pointer or something?
Yeah, pointer right there.
Yeah. Okay, here we go. Okay, hello everyone. My name is Jose Martinez. I'm working, well, I'm serving as a research associate for the Forest State collection of Arthropods, and also working in the Hawaii Center for Lepidoptera Biodiversity as a curator in the Neotropical Lepidoptera. So for people that don't know me, I've been working for almost 10 years in the Florida Museum, working with Neotropical Lepidoptera in general, specifically in Noctuoidea. And recently, I started working with these three groups again. Well, this is a new one, and I started working again in Neuroptera. And I have the opportunity to work with my boss, Eric Rodriguez, there, working with in fruit flies, right? So probably you know me because a couple of years ago, I gave a talk about the reclassification of Noctuoidea, where I'm raising 13 subfamilies to family level after a lot of work in morphology plus genomics. But what I want to talk about today is about my passion. One of my passions is cryptic speciation. So I'm working in a really huge project in the Andes right now. So trying to identify cryptic speciation in general. So, something that people don't know is like, what we know about cryptic speciation? Literally nothing. So I start my work with cryptic speciation when I started working during my PhD with the genus Gaujonia. Gaujonia had 4 different species by then, and it was crazy because you can see actually here, you're good at morphology, you're gonna see that every single one of them is a different thing, right? So it turns out that I separate them in 6 different genera and 25 species. Yeah, so but one of the things is why they are so diverse, so why people never work on those things. And the main and most important thing is that they have probably 200 years since people start working with them again. And another problem is that, as Daniele was saying, so taxonomy is not a trending right now, unfortunately. But I start thinking, there's actually a monotypic species there or not. So one of the main things here that I explain, why I'm explaining this is because every single one of these taxa here, they consider it as a monotypic species, and now they are not. So, and when I was in South America, I started seeing these guys around, right? So I can see one of the most beautiful moths in the world, for me at least. And so what happens is that I saw it in Guatemala, and then I saw it in Colombia. Then when I was in Brazil, I saw it too. So why the distribution of these guys is so extensive, right? And well, probably it's just that they can adapt very fast compared with other groups. But what happens is this. I found out that they are not actually as single species, but multiple species. But also, if you're going to museums in general, you're gonna see that people start confusing some of these moths in general. So here in these guys here. These 2 guys are totally different genus, but they look quite similar, right? So unfortunately, right now, if you want to work in taxonomy, you have to add some genetics, and people don't like morphology only anymore. At least you want to keep this in a really good journal. Usually what happens is that they don't accept this because they need more evidence. But I try to transfer this in my knowledge in Lepidoptera to Neuroptera again. What I see is happening exactly the same. So for decades, the genus Polystocodes was considered monotypic, right? And there was a paper a couple of years ago that there was a new specimen in Arkansas because supposedly the population in the eastern side was totally blacked out, right? But we have a specimen right now. When I was in Canada, when I was collecting, I guess it was an Ottawa, well, so I found one of those. And I said, oh, what I need this, and I just, throw it away. I never expected I'm gonna be working on them today. So, but the situation was that it turns out that there are multiple species in the same genus. So the same thing happens with Olearsis as an endemic species in California. And now we know that there are 2 different species. Again, we need evidence for more molecular data. So sorry about these points here. They're actually gray, so they don't have really good support. But one of the things that I started getting right now, working with fruit flies, is that I started working with this species, Saquida colombiana species that is endemic to Colombia. What happened is that when I was reading more about them, happens that I saw this. So people that say morphology probably can figure out that there are 4 different groups here. And these 4 different groups, and I knew that these groups are totally different. Despite that they are put in Itsakida, I can see that they are not Itsakida based on morphology. So I started checking all these specimens, all the information that's online, and try to figure out what characters are important, separate these groups. Well, again, turns out that they are not monophyletic. So this is something that happens all the times in general. One of the main problems is that people stop using, again, morphology just because just bending everything to the molecular data. So for people that haven't worked in cryptic species have a problem. This introgression. So some of these species, they don't actually diversify genetically. So that's why you can actually make some mistakes using this genetics only. But the main problem that I'm seeing so far is that how these things help us to, as researchers, right? So one of the main problems that I'm seeing is that we don't know the true diversity around the world because we think that these monotypic species are just one species, right? Now, probably there can be more than one. So, and another thing is the insect decline problem, right? So there are so many species that are getting extinct right now, and they don't have a description. So we need to know what are those things. Related with conservation, we can actually identify things that are endemic to some places. So there are some species that are very restricted to small areas that probably predate more than the other ones, right? So in conclusion, so what I can say is that we need more work in monotypic species in general. So integrating morphological and genetic data is important, but we need to use more morphology now. The biodiversity species reveals are really good. The true biodiversity in general. Usually we have this problem around the world where people continue using the monotypic. I can see that it's probably we can stop using this kind of stuff, at least if you don't have a really good evidence that it's a monotypic species. And the impacts that it's having in conservation are really important right now. And I guess that's it. Sorry, I guess I run.
Thank you, Joe. Any question for Joe?
How do you recommend for a group you know has a huge cryptic species complex that is so undescribed? How do you recommend beginning to tackle that?
Okay, one of the things that I started with was usually one of the things that I never recommend people in general is just size, right? So usually you have specimens that have different sizes. So then regions start sorting these specimens by regions. Then after that, start checking morphology. Right? One of the main problems that mostly people have is that they check a couple of specimens and that's it. So this morphological data in general, usually when you're creating this, you have to do a lot of specimens. So people only work with 1 or 2 specimens normally, whatever is available, right? But in my case, when I was doing all these things, I saw these morphological characters are way different because I was doing more and more, right? But usually you don't have that. So in order to tackle this is just take good pictures because people are using drawings and I don't like drawings because people interpret things that are probably not there. But you can actually find more specimens and contact people from around the world. So you never know when the specimens gonna be. One of the things is that mostly of the Neuroptera they have here are from Italy or from Geneva, all these places. And it's weird because we have, well, those are the United States, right? So I wouldn't have them. But people don't like them, and that's it, right? But they're very uncommon, and they were collected. So that's actually one of the things that I usually propose. So another important thing is DNA. There are many methods right now that you can use. We've been using methods that you can get DNA from specimen 500 years old, something like that. You try to match those two, but usually I'm saying you have two species that are close related. You need to do morphology because there's integration between that. You can actually make mistakes there. Yes, this.
Any other question for Joe? All right, thank you, Joe. And now we got our next speaker, Matt Moore, Adventive Arthropods in Florida, as reported by the Florida Department of Agriculture and Consumer Service, Division of Plant Industry, from the 90s to 2023. Matt, it's yours.
Great. Thank you. All the co-authors here are from Division of Plant Industry, either administrators, scientists, and staff. And many people who are responsible for these records being identified in Florida for the first time are not on this paper, right? Because the data set goes back for so long. So we sort of owe a legacy to our long retired coworkers or coworkers that have moved on from the division. I have the Castillo de San Marcos in St. Augustine, Florida here. As a reminder, that in Florida, we probably have the longest history of non-native, introduced, and invasive species as anywhere in the continental United States. We can make that case. Really, there's a legacy of the Columbian Exchange that in part is present here. For example, feral hogs present in Florida are descended from 16th century introductions, really long 100-year-old processes. But that's really inconsequential to what we're seeing today. Forget the past. Let's go to the future and the now. On this map here, we have in red the major airports of Florida, blue the maritime ports, and 2 space ports. So you can be sure if there's ever an alien invasion, it's probably going to go down in Florida. Good. Place your bets. Okay, but because of all of this movement of people and goods, Florida has been called what could be called a sentinel state. Like you would plant a sentinel garden with, high risk commodities in an area that you would monitor constantly for new pests and diseases. Maybe Florida is serving this function for the country in some ways because of all of this movement of goods and people. But I think this term is a euphemism. It is kind of, it is good because you're like, oh, we're providing a service to our country, by essentially taking in all of the trash. I mean, really, it's not a good situation. Also, we have our proximity to the Caribbean, so many pest species that are present in Latin America kind of move up that way and find their way into Florida. We have our unique subtropical to tropical climate, which means we have a different sort of complex of pests and diseases that stay here. Our diverse agriculture, because of that climate and also year-round growing conditions, we never get a rest. Basically, pest pressure is constant in some parts of the state. And then massive amounts of trade, tourism, and immigration into the state. Florida was basically not populated very much until the mid-20th century. And now we have explosive population growth. There's more people, more business, more movements of goods, and the people has created a situation we are just accumulating new organisms in the state at crazy rates. This is a great paper. You should read it. Syrphidae et al. 2023. The lead author is from University of Miami. So he knows the area well. And he has come to the conclusion that South Florida is probably the most invaded continental land parcel in the world. And this is a map from his paper. It's a heat map with established non-native species. Wow. Red is worse, yellow is less worse, but still bad, right? And there's some important lessons here from this figure. One is that he thinks that the community ecology of Southern Florida is more structured like an island than it is a landmass. So if you think about it, you're surrounded by water on 3 sides, and then that dotted line is a frost line where it hardly ever freezes. So essentially, yes, you're surrounded by water and you have this huge kind of abiotic that separates the tropical fauna of South Florida from the subtropical fauna of the rest of the state. And because of that, it functions like an island. And we know islands are susceptible to invasion in many ways. And that's kind of one of their conclusions here, is that South Florida might be behaving like this. It's susceptible, tons of propagular pressure. We just went over that last slide. Empty niche hypothesis because of that island-like habitat. The subtropical and subtropical climate down there means there can be a different complement of organisms than there would be elsewhere. And then the massive habitat disturbance from human settlement and reorganization of the land. The invaders have some features that are in common. They have broad niche breadths and large native ranges. Their climate of their native ranges matches well with South Florida. They have almost all are selected life histories. And less important, but still a factor, is that their native communities they come from also seem to be maybe sometimes structured like South Florida. And so at DPI, the Diagnostics Bureau there, one of our functions is to identify things that come in from thousands of samples a year, and we report new records. So these are many different kinds: county, state, either the continent, hemisphere, or host records. And a lot of these get reported in gray literature. So we have a quarterly, I don't know, we call it a newsletter, journal, gray literature, called Triology, that records all these new records. And we also have pest alerts and circulars that we put out. And then a lot of those more high impact finds or things that need bolstered up diagnostic data get published on separately. So there are manuscripts that follow up triology reports, but not all of them. And one of the things that we found is that triology, which has been going on for a long time, it's just not as permanent as we thought it was. It's hosted on a state website. Some old issues can't be found very easily. The data is not that accessible. And so we thought that it was time to formalize a lot of these new records and publish them in a way that will protect their permanence a bit. And so that's what the rest of this talk will focus on, is that compilation of data and then a comparison of those data to other states and the country at large. So there are some data that are excluded, things that aren't in this list. Intentional introductions, that would be biocontrol agents, for example. So you won't see air potato beetle in our list. We don't count it. Interceptions and eradications are not included, so there's no Oriental fruit fly, things like that. Things that may have temporarily established, but then have subsequently never been seen again, we also don't include. And misattributed native species are another issue as well. So, Central Florida might be the extreme southern point of some native eastern seaboard insect. Rarely encountered here, but still there. Sometimes these might be misattributed as new records when really they probably were here, they were just unnoticed. Thing to keep in mind about this data set, taxonomic expertise is a big issue, right? We can only really identify competently the things we have scientists to work on. So not every group is accounted for, like Trichoptera, for example. There's no Trichopterans in our data set, though undoubtedly there are probably non-native Trichops in Florida, but they're not in this data set. And then sampling, we're the division of plant industry, right? If it's not in a nursery setting or it's not being shipped in and out of the state, we're probably not looking at it. So you won't see predators, scavengers, detritivores. A lot of those things are not in this data set, but they're surely out in the world. So this is going to be an underestimate of the actual numbers. Also, non-exhaustive digital and physical literature searches. We simply cannot go through everything. We didn't have the resources to do that at this time. And then we compared our data to other states that have data sets and then federal data sets as well to see whether there were discrepancies and agreements. So here's our results. 30 years, 450 Adventive arthropods established in the state, officially recognized by your government. 159 of these were state records for Florida, so those were insects that were, arthropods, excuse me, are already present in the United States, found their way to Florida. 249 continental records, and 42 Western Hemisphere records. 36% of them we consider probably have pest status. And they're representative of 4 classes and 16 orders. Over here, we've got this bar chart with a breakdown of the groups. And you'll notice the primacy on top of Hymenoptera, and then plus the Hymenoptera for Condylonatha superorder, by far the most represented in the data set. Next, you have arachnids, starting in the 2000s and 2010s, really went up. Interest in spiders, but also mites started being identified a lot more rigorously at the division. So we found more cases there. And then beetles, because of the historical legacy of research interest at the division, there's always been a lot of beetle records as well. Lepidoptera really took off in 2010s. We think that's probably because of CAPS programs focusing more on LEPs, and then things that were being caught as bycatch in the various surveys. We're like, oh, okay, we weren't looking for that, but it's cool that we found this thing we didn't know was there. And we're not alone with Hymenoptera. Other authors have noticed this as well, that Hymenoptera might be the most invasive insect order around the globe. And their global invasion success is driven by the suborder Sternorhyncha, which accounts for a huge proportion of introduced Hymenoptera. And in our data set, 20% were Sternorhynchans. So we're not alone there. And in context of the rest of the country, older data sets informed us here. So we had Frank and McCoy, who reported Florida Adventives from the 70s up to 1991. We added data for ants and also some moths. And there's missing data here because not every species has a proposed introduction date, but we can see a relatively flat proportion in Hawaii, California, and the lower 48 across time, whereas in Florida, we have this sort of explosive chart. There is no doubt missing data here that could be filled in, but we still think that this trend is probably real based on the demographics of Florida, which is changing so much in the last like 5 decades. That's probably a real pattern. Now, if we add everything up, we can see that Hawaii, for states with data, is the most invaded by arthropods. California comes in with a lot. Florida, nearly half of that. In the whole lower 48, 3,600 adventive arthropods. So in this case, you might say, oh, Florida's actually sitting pretty, we're doing good. Well, I don't know, not at the rates that we're accumulating species now. We'll catch up quickly. The geographic origins of these adventive arthropods, the neotropics seems to be where we get most of them. So the bar chart here, you can see the red bars on the top are the neotropics plus Nearctic, and the orange there accounts usually about 50% things that are reported new from Florida, followed by, if you lump together Indo-Malaya and Asian Palearctic, would probably be the next largest group. So really neotropical insects and then Asian tropical insects are probably the biggest contributions to our adventives. And there's some discrepancies here from our list. I'll touch on this towards the end, is that we reported 291 hemispheric continental records, but 190 of those were not listed by federal data sources. And we're not exactly sure why. This was particularly acute for mites. 92% of our mites were not recorded by the feds. And also, they seem to ignore our neotropical or unknown native origins. That's probably a data interpretation issue where they just are more conservative. So many of the mites may be only ID'd to genera or to genus without a species. And I guess they just don't think that counts. Or there's some uncertainty there that they won't make the leap. Similarly with the neotropical origins, maybe they just don't buy that. They're not native here. But I maintain our data are probably an underestimate. We average 13 arthropod species established annually in Florida, just from a governmental perspective, right? So that number is higher in reality. We're experiencing a continued increase of invented arthropods since the 1950s, and maybe the rate will go down, but we're still getting lots and lots every year. And the reporting from states may not be reflected federally. So keep that in mind as you're comparing data that you're talking about in your own work, is that, you know, a state agency may not agree with the feds and vice versa. There might be differences of opinion. So with that, I'll take any questions.
Thank you. Any question for Matt? One question?
Sorry if I just didn't see this, but is there a website or publication where this data?
Just got accepted in Florida entomologists. So hopefully within a couple months they'll go into production and you'll be able to read the gory details that you want. Yeah.
Got another one?
You have a question in the chat as well.
Recorded, done by citizen scientists, the iNaturalist platform. Yeah, yes, I think there probably is. That would be a question for our Hymenopterist. Maybe there was something, I'm remembering a case, but maybe not, it wasn't in Florida. It's happened in, yeah.
But we actually have to go get a specimen.
Yeah, those iNaturalist reports don't ever count as the records, but they definitely had tip our scientists off for sure.
We got another question from Matt, which is interesting. What type of data standards are behind the aggregation of all this data?
I guess you'll have to read the paper. No, I mean, I can tell you about what the standards are. There was things that I had criteria for including or excluding data, but I don't have
It's
time.
a long conversation. Yeah.
Okay.
Thank you, Matt, again. Right now we got Susan Halbert. Thank you, Susan. Ambrascia biguttula, sorry, a new pest in Florida. Susan. Yes, so you can change, go forward, and this is go back. You don't have the actual pointer. Doesn't work.
All right.
Thank you, Susan.
So I'm going to talk about a new leafhopper that we have. We're calling it two-spot cotton leafhopper. And it just showed up. And all these people are either DPI people, mostly CAPS, and then the ones at the end are the federal people that helped us, and Felipe is our lab leader. So it was found in November, middle November of 2024, just last year, by Amy Rhoda of the USDA in very badly damaged okra. And this might be important, that it's a very weedy field and it should have been plowed up a while back. So this is typical that pests really get going in a situation where the crop should have been destroyed after it finished producing any usable fruit. So background, it's native. You can see that part of the world where it's native in Asia, and the type specimens from Indonesia and Japan in blue. Adventive in Africa, adventive in the Caribbean, found in Puerto Rico in just a year or so ago, 2023. Also in the Virgin Islands, various Caribbean islands, some of which will not be named. I started getting specimens from unnamed islands, and then I realized that we needed to have a survey for this thing and have a pest alert. So I got permission for the pest alert. It took a while because there were several Caribbean authors to coordinate with. And 24 hours after it was accepted by my administration, Amy Rhoda found it in Miami. So, good timing, I guess. Anyway, they're Typhlocyvine leafhoppers, and they feed by sucking on cells. And so the feeding damage looks like thrips or mites or lace bugs or other things. And the damage is called hopper burn. Background, the hosts, a lot of hosts, a lot of different families. Eggplant is severely hurt. Okra is very severely hurt. Cotton is the major worldwide crop this affects. And several common weeds in Florida, the ones with stars on them, were found in Florida. And the rest were reported in the literature. And there are very many crops reported in the literature. So is it a vector of plant pathogens? That's always my first question because that's my background is I'm a vector entomologist. So that's my first question. Does it transmit anything? And the damage here looks a lot like the viral damage, but in fact, nobody has ever been able to find any transmitted pathogens, which is a really, really good thing. So how did it get here and how did it move so fast? Within about a month and a half, you can see the map. This is our public map that doesn't show the actual points where they are, but you can see how many counties in just about a month and a half where we recorded them. We went down to look in Miami, and by the time we got back, there were 2 more counties. So it just moved so fast. And I think we have maybe 3 more counties now. We continue to get them. And you can see the transect from southeast to northwest of Florida. And that was, I think, Jackson County, which is the one in the Panhandle right next to Georgia and Alabama where all the cotton gets produced. That came in, that county record came in while we were gone looking in Miami. So you can see it's really fast.
Seminole is positive.
Yeah, it is. And so are several others. Like I say, several more. This isn't the most up-to-date map. Okay, how did it move so fast? Well, here's a comparison with Diaphorina citri that you saw a portrait of in the last talk. And so it was found, and we found it really early, I think within 6 months to a year of the introduction in coastal Palm Beach County. And probably it took a year or a year and a half to get down to Miami-Dade County on its own, just flying on its own. And then it spread on orange jasmine plants to the rest of the state pretty quickly. I remember Julieta and I went down there to look, and the places that sold to the discount stores were loaded with these things. And so all the counties in crosshatch there, I think what? Is that like, I think Pasco County or something is a late inspection. But all the rest of them, the first finds in the county were in some sort of retail outlet. So it moved by human agency. And this took years to get all the way through the state. But on the other hand, for us, this bug, very short time. We found it, and then all of a sudden it was in suction trap samples. It was in sticky trap samples with lures for moths. It was everywhere all at once. And so I always poo-pooed hurricane distribution. I watched where we were shipping acres of plants to the Cayman Islands and from South Florida. And after the Hurricane Ivan destroyed their whole plant life, the sea washed over the islands. And so we were revegetating the Cayman Islands. And pretty soon, Greg Hodges had all the scale insects of Florida on his desk from the regulatory lady in the Cayman Islands. Well, where did they come from? Did the hurricane bring them? No, we shipped the plants there. I watched how citrus greening went to Louisiana after Hurricane Katrina. Did it blow there? No. They wanted to revegetate their citrus trees that blew away, and they got them from Florida. And all these infested and infected citrus trees were for sale on the northbound lanes of I-75. How did Georgia and South Carolina get greening? It's no mystery. But so I always poo-pooed this. But this map from the USGS right after Helene and Milton, it looks to me like the places where we're infested match pretty well where the distribution of some other pests has occurred based on Helene and Milton. So I wonder about it. You know, it's not, there's no way to prove it. One suggestion is that this thing's been here for a long time and we're just seeing late crops, and so we're seeing it blow up in various places. But we'll know in a year, all right? Because we'll know if next year we have an explosion of these things all over the place. So we'll know in a year. So what do you look for? You might see the damage before you see the bugs, because these little beasts are minute. You can see based on, I don't have a pencil by it or a dime, but you can see based on the leaf sizes and somebody's finger there, how big they are. They're just really, really small. But look for the spots on the wings. If you have a good hand lens, that will separate them from all the potato leafhoppers, which they're related to. And potato leafhoppers, there's maybe 1,000 species in that larger group. And I don't want to see a whole lot of different species of potato leafhoppers. And so we want to call this thing two-spot cotton leafhopper. The USDA wants to call it a jacid, and we have no idea what that is. It's an archaic name for a little leafhopper, and even Google doesn't recognize it. And in the hands of the public, it's going to become Jurassic. So I think we want two-spot cotton leafhopper for the name, my pitch. But okay, so Amy Rhoda, in that heavily infested okra, Amy Rhoda organized an experiment on the fly, and we figured out that the best way to sample for these things consistently is to take a tray, a beet tray, spray a little layer of isopropanol on it and beat over it. And those things are going to get drunk and fall in the alcohol. So that's the most consistent way to get a sample. Authoritative IDs, once again, male genitalia, you have to do a dissection of the males for any kind of record, for a county record, host record, or anything like that. All right, so the most important thing I'm going to say today is that this pest, I think, can be managed. Everybody remembers their IPM 101. Cultural management. Look at that okra. Somebody should have plowed that okra crop up a long time ago because it's sprouting from all the nodes. And guess where the leafhoppers are? They're on all that new growth that's coming out of the nodes. If they plowed that okra up after it was done, then you would not have that. In the eggplant, we have pretty good pesticides, I think, already. You can see the new growth of the eggplant. The old growth is toast because the bugs have been there a while. But then the farmer noticed he had a problem and he sprayed it. And the new growth is coming on pretty good without leafhoppers. So I think we already have some pesticides that work for a cleanup. In India, they're working on host plant resistance in okra, and there's some work on biocontrol in Japan. Maybe there's no silver bullet, but I think IPM is really feasible. It's primarily an annual crop, so you can start over each year. And it's not a vector of anything. It doesn't transmit anything. So that means that you can have a viable economic threshold. I think it's similar to Russian wheat aphid, which caused tremendous damage. You can see that the wheat is rolled up like a little straw and the bugs are hiding inside of there, and it also turns it white. And we are losing maybe a third of the wheat in southern Idaho. So it was tremendous direct damage, but it didn't transmit anything. So we got biocontrol from Central Asia. We got agronomic methods so that you didn't oversummer your wheat crop and the pests. And we got host plant resistance and a cleanup insecticide that was cheap enough to afford. So I think this one can be managed similarly. It took about 15 years before, now I think if you have Russian wheat aphid, you made a farming mistake. And I think we'll get there. So that's the end. Are there any questions about this thing?
Thank you, Susan. Any question for Susan, even online? Snapchat.
Have any other states reported this thing yet?
Not yet, but I bet you it's in Georgia and Alabama because it's right on the border.
So you got a big fan?
Go ahead.
Do you have a common society?
No, do you want to help me write one?
I mean, I just got it before, but
I never have either, but let's do it.
okay.
I don't want Jurassic leafhoppers.
Susan, Doris Lago-Kutz, it's a big fan of you as well.
Okay.
And is asking, do you think we'll get in suction traps in the Midwest?
Yep, I think it might. If it gets up there, it will get there because we already have them in the suction traps in Florida. Two locations. We have them in 2 traps in Immokalee and 1 in Winter Haven already.
Well, good news. Those are good news.
All right.
Thank you. All right, for the presenter years and also on Zoom, we are now on a coffee break for about 15 minutes. We're gonna be back 10:45. Yes. See you in a bit. The next presenter, a very good friend of mine, is a great honor to have him and hearing about his part of his PhD thesis as well, that right now is teaching, is an amazing Ichneumonidae researcher in Brazil. Got Alvaro Doria dos Santos with taxonomic review and phylogenetic analysis of microcarops, Roman 1910, which is a Campopleginae, very hard group in Ichneumonidae, extremely important for agriculture. He's going to be a recorded Zoom, so hope you like it.
Not separated by sclerotized bridge, as we have in Cremastinia wasps, the sister group of Campopleginae. The prepodium usually with a complete set of carina. The ovipositor with a dorsal subapical notch. And the tarsal claws usually pectinated, as we can see in the image here. From the Campopleginae genera, Microcarops is a New World genus with 23 valid species. This genus can be identified by the eyes strongly emarginated a little above the antennal sockets, the forewing without an areolate, the tergosternal suture almost ventral, the areola usually well formed but open apically, as we say, coffin-shaped. Also, the occipital carina is far from the base of the mandibles. This genus was described by Roman in 1910 based on the species collected in Tahiti by the Eugene Frigate expedition. Later, Henry Townes and Marjorie Townes transferred 6 species from the genus Carops to Microcarops. In 1985, Kasparian and Barr described Microcarops ussuriensis for the Palearctic region. In 1987, Gupta's taxonomic revision expanded the genus to 23 species, which he divided in 6 groups. In the same work, Gupta also raised the possibility that the labels of Microcarops tahitiacus had been swapped since the Eugene Frigate traveled from east to west across the Pacific Ocean. This hypothesis was corroborated by the fact that Gupta found many specimens of these species in several regions of Central and South America, but none in French Polynesia. In his revision, Gupta also questioned the placement of Microcarops ussuriensis. Dr. Gasparian discussed the generic placement of Microcarops ussuriensis with Dr. Gupta, which led to the shared belief that this species could potentially belong to a new genus rather than Microcarops. Thus, Microcarops seems to be restricted to the New World. The only phylogenetic work of Campopleginae at the genus level was carried out by Mia and Buya in 2001, and it placed Microcarops as sister group to Melalophacarops, and both genus as sister group to Genotrops, which seems to be fair because they are very morphological similar genera. So, despite the importance of Gupta's revision in 1987, it's still very difficult to identify Microcarops at species level with his work. So this motivated us to work on the taxonomical review of microcarps and to investigate the interspecific relationships through a phylogenetic analysis. So to achieve this objective, we studied material from the institutions listed in the slide. Here I would like to emphasize the importance of the Entomological Museum at the Utah State University because it holds a significant portion of the type material of microcarps and Ichneumonidae in general. Other institutions either sent material or were visited during this study. To begin the research, we dedicated significant time to studying the morphology of Ichneumonidae, primarily following Broad's work and the Hymenoptera ontology portal as well. Based on this initial morphological study of Campopleginae microcarps, we conducted a series of micro-CT scans and electron microscopy as well, photographs and illustrations to understand and develop a morphological guide, which includes also sculpture details, surface sculpture details. So here are some images of the morphological guide based on micro-CT scan. As you can see, we highlighted on Photoshop all the body parts of the specimen. And here's some illustrations as well of this work. Just to show here the surface sculpture, we did a little study on that based on Harris 1979. Additionally, we played a little with color descriptions. So next to each description, we included the corresponding hex code, and below the text, we created a color palette with all the codes, allowing the readers to easily visualize the described colors. New biological records were included from the material collected by Dr. Daniel Jansen and Dr. William Howatts in Costa Rica, which was also deposited at IMU's collection. This allowed the identification of several new host records for microcarps and to understand the evolution of the group. The phylogenetic analysis was conducted using 17 terminals, representing all possible microcarps species and newly proposed species as well. An outgroup comprising 6 species was also included. So character coding followed Sereno's proposal, which included a locator, a variable, and its variants. For example, the character 38, we have the clipius, which is the locator, the variable, which is the shape of apical margin, and the variant 0, which is truncated, and the variant 1, which is curved. For each character, we provide a detailed morphological description, as we can see here in the slide. So we have the description, the HAL code as well, and the explanation of the character as well. And here is an example of the continuous characters. As you can see, the character is the ratio between two measurements, which is explained in the description part of the character description. The analysis was conducted using TNT software, following the protocols in the slide. And we got a value of k with the script practical run of 13.8. And for each character, we calculated the number of steps, the consistency, and retention index as well. So here is our results. Our student concluded that microcarps comprises 31 species, including 12 new species, and some species that will be synonymized, which I will discuss shortly in the next slides. We also made a key for all microcarps species, and we redescribed the genus as well. So here's the redescription with a map of all the microcarps occurrence points. As you can see, it is restricted to the New World. In this slide, I'd like to show you the illustrations from Gupta's work. As we can see, they are highly detailed and a very well illustrated work for 1987. So why identification of the microcarps species remained so difficult? The reason for this was that the actual number of species was higher than previously thought. Furthermore, many of the species were morphologically very similar, and not all the species were illustrated. So to address this, our work includes illustration of all the type specimens described, as well all the new species that we are describing. Here you can see an example of synonymized species, Microcarpus lauricosa and Microcarpus nigricosa. These species were originally separated based on color, especially on the coloration of the hind coxa. However, we have specimens from the same locality with this color variation, while maintaining identical morphological characteristics. So, as you can see in the description of nigricosa, Gupta says that it's very similar to flavicosa, but the hind coxa are black. So it's not a very strong character to separate species, at least in microcarps. As we said in the material methods, new host records were obtained from the Daniel Jansen's material, and here's some new hosts for flavicosa. As we can see, we have a lot of new records for this species on the other species as well. Another interesting example is Microcarpus tibialis, which will be synonymized with 4 other species, as both specimen examination and phylogenetic analysis indicated they all belong to the same group. And here are some photos of the species that will be synonymized with Microcarpus tibialis. As you can see, they are very, very similar within each other. Among the newly described species, Microcarpus mic0 stands out due to its distinctive coloration pattern and the absence of the justacoxal carina, which places it as a nearly external group within Microcarpus, although it still retains diagnostic characters of the genus. Additionally, some species proposed by Gaud were described and included in the analysis. During my visit to Imur's collection, I found material that he had studied and proposed as a new species, which were never formally described due to his passing. The phylogenetic analysis included 180 morphological characters, of which 27 were continuous, 22 represented racial-based relationships, 81 were numerical, which 14 were associated with coloration patterns, 12 with surface sculpturing, and all these traits were illustrated in the study, enabling the reconstruction of the evolutionary study of microcarps. Here's some demonstration of the characters. And here is our tree, and only tree because our analysis recovered only a single most parsimonious tree, which bears a high homoplasy rate. But despite the high rate of homoplasy, we believe this is a natural phenomenon in microcarps, which follows the pattern observed in Ichneumonidae in general, as a clade with rapid evolutionary rates and intricate morphological convergence, most of them related to the transitions in host utilization. Another interesting finding was the continual characters exhibited less homoplasy than coloration or surface sculpturing traits, reinforcing the importance of using quantitative characters in morphological phylogenes. Unfortunately, the species groups proposed by Gupta have not been recovered, with the exception of the Fulvoritus group. We think this may be explained by the fact that many of these groups were defined based only on the ratio of the interocellular distance and the ocellular distance, a criterion that proved to be unreliable since the measurements for different specimens overlapped, prevent clear species separation. Another important aspect is the host data for microcarps, which has helped us to better understand the evolutionary history of the group concerning its hosts. So we believe that the microcarps is likely originated from an ancestor specialized in invalid butterflies. This hypothesis is supported by the sister group of microcarps, Melalophacarps, which also has hosts within Papilionoida. As we move towards more derived groups in our tree, the host range appears to become broader, to a more generalist parasitoid. This culminates to the Tibialis group, which attacks a long range of hosts. However, we still think that Microcarpus tibialis is one species, because we have a lot of examples in Campopleginae of species that parasitize a wide variety of Lepidoptera families, for example Campoletis flavincta and Venturia senensis. So, to summarize, our study provides the first phylogenetic framework for microcarps and a comprehensive taxonomical revision of the genus, nearly 4 decades after Gupta's foundational work. Our results challenge the previous defined species groups, revealing their paraphy and emphasize the need for a more integrative approach to classification. We also highlight that continuous morphological traits, rather than color pattern or surface sculpture alone, we offer a more reliable phylogenetic signal. Also, we observed that many species of microcarps exhibit high phenotypic plasticity, thus highlighting the need to consider interspecific variations for identification and description of new species. We also highlight the in-depth studies of the biology of microcarps may elucidate the evolutionary history of the genus. I would like to thank you all for listening to my presentation, and if you have any questions or would like to discuss this further, feel free to reach out to me at my email. Thank you again for your attention. Bye.
Not sure if Alvaro is online, but are you online, Alvaro?
He's not gonna be able to, I can make him a panelist, but
Yep. Okay, so if you have any question, we can write it on the chat list. Do you guys have any questions for Alvaro, also via Zoom? Right. How long did it take you, Alvaro, to finish up this project? I know it was part of your PhD, but what your timeline, would you say?
Hello, everybody. Can you listen to me?
Yeah.
Okay, nice. Thank you for your answer, Matt. So I did a very long PhD, almost 5 to 6 years, because we had the COVID in 2019. So I had to extend the timeline of my PhD. So yeah, a long time to understand everything. And I just went to the Emus collection in 1921. So it was a very important collection to visit since we have almost all the types there. So yes,
Fantastic.
Long time.
So there is a question on the Zoom for you by Matt. How did you find the HAO, the Hymenoptera anatomy ontology IDs for the parts, and what tools did you use? The portal, etc. What kind of tools for the different morphological sclerites parts?
Just the search bar. So I just wrote the name of the parts I was trying to understand, and I found the answer in the website. However, some of the images are not appearing in the website, so it's something that we need to fix.
Yeah. Fantastic. Okay, thank you, Alvaro. So now our next speaker is Rachel Behm about a very interesting cutting edge topic, I would say, transposable element repertoire of Braconidae and its potential phylogenetic underpinnings.
Right. Now for something completely different, as I'm sure, I don't think anyone here works on genomics, so I'm gonna talk about it more from the evolutionary scale, or lack thereof, as you'll see. So obviously, what are transposable elements? I assume not a lot of people work on them here, so I'm gonna give a little overview. They're mobile DNA elements that insert themselves into host genomes and replicate, very similar to viruses, as you can imagine. And this has a lot of consequences that I will get into. But much like the insects that we love, they are also classified systematically, with the broadest classification being the class level for retrotransposons that work a lot like retroviruses, and DNA transposons. Well, as you can imagine, they're separated that way because of the intermediate they use to insert themselves into their host. Retrotransposons use an RNA intermediate, sort of in a copy-paste mechanism where they copy themselves, then reverse transcribed into the host genome. And DNA transposons use, surprise, a DNA intermediate, but they do a little more variable mechanisms of potential craziness. And these are further explored at the order level, which is, like I said, these are the two broadest classifications of transposable elements. Most transposable element work goes further at the superfamily level, but just for everyone's sake here, everything I'm collapsing up into these functional groups of which these transposable elements work. Now, what do TEs do? A lot. They're highly correlated to genome size, especially in insects, with the smallest current recorded genome, the Antarctic midge, having the smallest amount of transposable elements. I believe 99 million base pairs is the Antarctic midge currently estimated at, and 1% of that is transposable element content. Whereas the migratory locust, which was previously the biggest genome that has been estimated for insects, not anymore. It's changed since I made this presentation at 6.5 billion base pairs with 60% TE content. Massively bigger, massively more TEs, highly correlated. TEs, as you can imagine, when you insert foreign things into your genome, depending on where it inserts, how much of it has huge consequences on the genome structure and evolution by either changing the GC content, by inserting itself into an important gene, by where it inserts all over. And if they accumulate, that can cause rearrangements of the genome, which have big impacts on the insect. In addition, transposable elements can cause novel functions, such as pesticide resistance, viral resistance, so many things. And not only do they transfer vertically from offspring to parent, they can also transfer horizontally. So between insects, between insect and plant, all over. And a recent paper has found that there's been at least 2,000 of these transmissions, and that's a very low estimate, very low. And so because of this horizontal transmission, obviously Hymenoptera, that's what I care about. However, the current studies, or what has been known previously, are from aculeates. No offense to aculeates. Mostly in bees and ants, is everything we've said about transposable elements in Hymenoptera has been based on these groups. And what we know about Hymenoptera genomes is that they're very small in comparison to the rest of insects. And that may be due to haplodiploidy, where the males will have half of the chromosome count. And perhaps it's been proposed that maybe deleterious or neutral accumulations of transposable elements are more easily removed because they can't hide in a male genome. So if they are deleterious, they can't hide phenotypically and they're removed, perhaps. What we know about transposable elements, like I said in Hymenoptera, is that the composition, not at the order level, but lower, varies quite a bit in comparison to the other orders, but the diversity of those lower levels, we know that they're low, mostly in bees and ants. We also know DNA transposons are more abundant than retrotransposons for these groups. Like I said, there's been a smaller sample size, so we don't really know the trends. But of course, there has been a massive underrepresentation of parasitoids. Now, you can imagine that that's very interesting when you're looking at it from a comparative genomic perspective, considering that parasitoids are most of Hymenoptera. In fact, Ichneumonidae, which are ichneumonids, and braconids, which I'm doing, are 33% of the described braconids, with way more, maybe more than beetles. I know that's kind of controversial, but I'm biased. So of course, I wanted to tackle, just to start off just with Braconidae. And new work has been done allowing for braconid genomes to become available. So this underrepresentation isn't malicious, it's just now more genomes are available. Because, you know, bees and aculeates are desirable, I guess. So Braconidae, a really interesting, important group for many reasons. Of course, parasitoids are great, they're obligate parasitoids. So you can imagine that very intimate relationship with their host, potentially cool TEs. They're hyperdiverse, and by that I mean there's currently over 21,000 species that are currently in 41 subfamilies. Like I said, they're parasitoids of, I believe, currently like 12 orders of insects recorded. Very interesting group. It's broken up into two major lineages that have been supported phylogenetically and morphologically, called the cyclostomes and non-cyclostomes that likely diverged in the early Cretaceous. So I want to see, how they compare those two groups. They also have viral endogenization, so symbiotic viruses that have incorporated into their genomes, of which they use to help them in parasitization of their host. And you can imagine that works pretty similarly to a virus or a transposable element, as they are viruses. So that made me think, we don't know anything about the viruses that much, really. We should investigate this further. And there's been at least 2 independent taking in of those viruses. And previous estimates have shown that Burkonidae have the smallest on average genomes of all of Hymenoptera. But of course, there wasn't that many to sample before, so we're going to look at that too. So of course, as you can imagine, my objectives are to look at the transposable elements in Burkonidae. I'm actually doing a bunch of comparative genomic stuff, but this is a fun first look into a certain part. And I want to test across the phylogeny of what's available, if that's tied to maybe those 2 major lineages, what is available to ask with what I can look at. So methods, I have to get genomes, which thankfully there's been new work. Darwin Tree of Life, located in England, has made a lot of genomes available of Ichneumonidae. Thank you, Gavin Broad. And so I have filtered and I have scrounged and I have begged and pleaded with whoever studies Burkonidae that I can to give me genomes of high quality so I can look at them. Then, of course, I want branch lengths, and I have to make a phylogenetic tree, as we've all mostly done, so that I can test these character traits over the phylogeny to see if they're distributed amongst the phylogeny. I annotated transposable element content, which is a lot of coding and a lot of R and a lot of coercing data. And then, of course, statistical analyses, comparing means, doing calculating phylogenetic signal, so on and so forth. So here's some results. So how much of the genome is made up of transposable elements for Burkonidae? It's a lot to look at, I know, but it's quite variable. So we can see that the blue is the cyclostome lineage and the red is the non-cyclostomes. And based on what we knew about Hymenoptera previously, they have a higher average and much wider distribution of how much of the genome is made of transposable elements. Because before we thought that Hymenoptera had a very low amount of transposable elements because their genomes are low. But Burkonidae, from the study, it's pretty variable, with the average being this dotted line in the middle at 26.6%. And the Hymenoptera, as before in 2019, when the last survey was done, was 16.5. Our range has pretty much doubled at the higher end because Burkonidae have a lot of genomes or have a lot of transposable elements. And of course, we've increased the sample size a lot, which just goes to show you how important sampling evenness is. And we can also see this high variability within. There's some clear patterns, right? But surprisingly, it is not tied to phylogeny. So even though there are clear patterns within, it's not at the lineage level. And there's no phylogenetic signal, which means that it is more random than something across the phylogeny or not distributed like that. Here's another view of that, where we can see there is totally a high correlation of genome size and transposable element content. And the lineages do better explain that fit of that data. Burkonidae have very small genomes, as previously thought, much lower than Hymenoptera, as per previous work. Of course, we've increased the upper limit with the sampling. Like I said, we can clearly see it's highly correlated, but for transposable element content, which isn't phylogenetically conserved, genome size is. And even though it's not at the lineage level, it's probably at the subfamily level, but I don't have even enough sampling at the subfamilies to look at that. So please get me some genomes. Okay, what TEs, those major groups? Very patchy distribution. So we see LTRs, lines, TIRs. These are things that are in all insects. We got them here. That's good. Helitrons and mavericks, which are a very interesting group of DNA transposons, we have in all but one, which is kind of strange. And everything else is very patchy. That's very weird because those major groupings, this is at the highest level of transposable element classification, should be very conserved. And when they're not, it's usually indicative of like a huge shift, either evolutionarily, phylogenetically, but there's no phylogenetic signal, so it's not phylogenetic. So how abundant? So here's a heat map I made of the copy number. So how many copies of each of these are there? We can see, yes, a ton of DNA, but with a 70-fold distribution and copy number. Very wide variety. And some even have a preference for DNA transposons or for retro. It's not consistent, which is weird in one family. So there are pattern-wise differences in some of these groups, like DLEs, Penelopes, Cryptods, LTRs, but once again, no phylogenetic signal. So not tied to phylogeny, tied to something else, which I have ideas about. So they're not conserved along the phylogeny. Okay, what about relatively of the transposable elements they have? What are those relative proportions? Because those should be pretty consistent, right? Wrong. It's buck wild all over the place. You don't need to see specifics, but you can tell it's all over the place. Like I said, there are lineage-specific differences in some groups, but once again, this relative proportion has no phylogenetic signal. And the elephant in the room, if this is the relative proportion of TEs, most of that's blank. That's because most of insect transposable elements have not been classified at the highest level. So that's a huge, what we know about transposable elements does not take insects into account. And what we know about insect transposable elements is usually from small model organisms like Drosophila. Okay, so this work, which I've actually just submitted, hopefully will be accepted, is the first comparative genomic transposable element paper at the family level in Hymenoptera. And it's the first one to include parasitoids, probably because most people care about achilles, I guess, but the genomes have become available. And the variation of all the transposable elements, what we know in Braconidae, have completely changed what we know about Hymenoptera. It's so wide and variable, just including this one family has totally changed what we know. It's just showing how important taxon evenness is when you do big comparative studies like this. And then for all of these things, only genome size itself was conserved. So if that's the case, then this crazy variation has to be from something that isn't phylogeny. For example, maybe host preference, maybe koino or idiobiont, maybe ecto or endo, which could maybe affect the rate of horizontal transmission. So I wonder then, if you were to compare the transposable elements of host and parasitoid, if maybe they would look more like each other than to more related species. So with that, I'd like to thank everyone and then open to questions, which I don't know if there's any.
Sorry, we don't have time for questions, but we got the lunch, so please reach out to Rachel afterwards. We're gonna quickly go to the next presenter, Robin Marquez, about turning over a new leaf, a survey of neotropical parasitoid Hymenoptera in leaf litter. Good luck.
Okay, hello everyone. Thank you to CSE for the opportunity to share my work today. As part of studying the biodiversity of tropical America, there are numerous initiatives to investigate the diversity of ants and beetles in the leaf litter of tropical rainforests. This project started with the study of ants. We used a standard collecting method for studying ants, which involves leaf litter sampling using this tall cloth unit called a Winkler collector. And I'll get more into this method later, but an important point for now is that Winklers often collect a lot of non-target arthropods. So when we collected these samples and were cleaning them, we noticed what I think was a happy accident. In our bycatch, there were a lot of these tiny insects called microparasitoid Hymenoptera. Microparasitoid Hymenoptera are basically tiny wasps that their life cycle involves laying eggs in a host insect. So we have two examples here on the left. We have this Cotesia congregata laying these white cottony-looking eggs in a tobacco hornworm moth caterpillar. And on the right, we have Trisulcus japonicus, which is emerging from the egg of a brown marmorated stink bug. And oftentimes microparasitoids like these two have hosts that are considered economically important. So in this case, the caterpillar is considered an agricultural pest, and the brown marmorated stink bug is an invasive insect. And so if we can understand the life histories of some of these microparasitoids well enough, they can potentially be reared and applied to controlling the populations of agricultural pests and invasive insects. And this is a simple diagram I pulled from a Forbes et al. Paper, which is an example of how some microparasitoid Hymenoptera, like P1, are considered generalist and might be associated with more than one host, or a parasitoid like P4, which is a specialist associated with one type of host. And so due to the huge amount of different host species that there are out there, and this combination of generalist and specialist parasitoid types, the conservative estimated number of parasitoid species is over 880,000. And this doesn't even consider the diversity of hyperparasitoids, which are parasitoids that lay their eggs in other parasitoids. So there is still a lot to learn about this huge diversity of tiny insects that play a variety of ecological and economically important roles. So going back to our project, typically when we are intentionally studying Microhymenoptera, we will collect them using methods like sweep netting or malaise traps. But here we have this bycatch that was collected using a untypical method, leaf litter sampling for ants. And we found this huge diversity of microparasitoid Hymenoptera that's likely unknown to science. And so this created an exciting opportunity for further investigation. And that's where I come in. Last summer, I had the fantastic opportunity to conduct research at the National Museum of Natural History, Smithsonian Institution, as part of the Natural History Research Experiences RU program. I was tasked with investigating diversity of the Microhymenoptera bycatch that was collected in those leaf litter samples. For one, because there's never been a dedicated study of Microhymenoptera diversity in neotropical leaf litter. And I also wanted to validate Winkler sampling as a potentially viable method for collecting microparasitoids. As you'll see shortly, Winkler sampling is very involved and physically labor intensive. And so we wanted to see if we are getting enough value out of this bycatch, then maybe it might be worth intentionally Winkler sampling for Microhymenoptera in the future. So for the methods, leaf litter samples were collected across 4 different countries: Peru, Colombia, Guyana, and Brazil. And though I wasn't involved in that sampling effort, I had the opportunity to learn the collecting methods for myself. So these images demonstrate the sampling methods that were used. Basically, we collected leaf litter samples, sifted them, and then placed that siftate in a mesh bag. That mesh bag is then hung at the top of the Winkler unit and allowed to sit for about 48 hours. And over time, the insects will emerge from that siftate and fall to the bottom where there's a collecting cup. So these samples were brought back to the lab. We pulled out all the Microhymenoptera from them and then roughly categorized them based on appearance, called morphospecies. And then species accumulation curves, Sorensen's dissimilarity, species and morphospecies abundance were generated using the R package beacon. On to the results. So this is a species accumulation curve, which is basically looking at how many unique morphospecies are collected as sampling effort increases. And the big blue line represents all of the sites. And here we have a zoomed in version, which is basically showing us that this very steep light blue curve, Colombia, we are the furthest away from discovering all of the morphospecies or the full amount of diversity that's there, as compared to this green line at the bottom, which represents Brazil, which is a little more flat. But really what this is telling us is that increased sampling at any of these sites could result in increased species discovery. This is looking at the number of specimens per superfamily by region. So this is looking at abundance. And we have some images on the right of the top 4 superfamilies that are really driving these patterns: Syrphidae, Phylogenetic, Chalcidoidea, and Diaprioidea. This is looking at the number of morphospecies per superfamily. So we're going from abundance to diversity. And this graph looks pretty similar to the last one, which makes sense because each morphospecies is represented by one specimen. But overall, we collected almost 700 different morphospecies. And we can see some of these bars tell us which superfamilies are really driving those steep species accumulation curves. For instance, this tall blue bar representing over 90 morphospecies of Syrphidae collected in Colombia. And then for Brazil, the highest bar is Phylogenetic, so that's likely driving the diversity there. This is a cluster dendrogram, which is showing us the similarity of the morphospecies between sites. Brazil, it's a large country, it's got many unique habitats, so it makes sense that this would be less similar than the other regions. Guyana might be less similar than Colombia and Peru because of the Guyana Shield. So historically, as glaciers formed and melted in this region, many organisms may have traveled in and out of Guyana to seek refuge from that flooding. And so some unique speciation may have occurred over time in Guyana to influence this diversity. So overall, results suggest that leaf litter does have interesting Microhymenoptera diversity. And future directions could include sorting our morphospecies more complete genus, intentional Winkler sampling for collecting Microhymenoptera in future research, or future research that more directly compares the Winkler sampling method to other methods like malaise traps. We're also wondering how these tiny insects endured this agitative method of leaf litter sifting. And so part of what we're thinking is that it could be part rearing chamber or emergence trap. So maybe when we collect that leaf litter, the parasitoids are still in their host. And then when it's hanging up in that Winkler unit, that's when maybe they're emerging from their host and then falling down, and that's how they remain intact. I also discussed another possibility with a mechanical engineer, and basically, as it turns out, physics can act very differently on very small objects. And so there's another possibility that even though we're sifting those leaves, and it looks pretty chaotic, we might not actually be generating enough momentum for that leaf litter debris to apply enough direct force to those tiny insect bodies. So it could be one, could be the other, could be neither, but that would require further investigation to find out. I'd like to thank everyone who supported me in conducting this project. And I'm happy to answer any questions.
Thank you, Robin. Any question for Robin? Yes.
What sort of habitat would samples come from?
It was primarily just tropical rainforest type.
Forest.
It was primarily natural areas, rainforest.
Do you still have a lot of bycatch
Oh, yeah. Oh, yeah.
that people have gone?
Yeah, I mean, someone had pulled out all the ants already, and I was pulling out the microhymes, and there were still just like beetles and springtails, you name it.
It kind of hinges on like most information availability for these, but like, did you see any trends towards groups that were like parasitoids of like ants or detritivores specifically being more overrepresented? Or
That's a good question, and I don't know if we fully know that yet, just because we barely even know what a lot of these are to begin with, but that's something that I'm definitely interested in, like those different host associations.
To that point, like anecdotally, do you have any idea how many of these are probably undescribed?
Probably the majority of them, I would say. Yeah, that's why I was like, maybe we could do genus at some point, but this was a summer project, so I had to be reasonable.
All right. Thank you, Robin. Now we have a tag team from DPI. We got Isabel Achia, fantastic, and Samuel Bolton presenting type imaging of Phylogenetic at the Florida State Collection of Arthropods.
Right, so yes, there's going to be two of us giving this talk. I'll start, and midway through, Isabel will take over. So yeah, Phylogenetic. So basically, I'm just going to start off with giving you a few key points about this, just for a little bit of context. So the FSCA houses one of the largest collections of Phytoceae in the world. And Phytoceae, for those of you who are not aware of this, they're one of the most important, in fact, the most important family of predatory mites used in biological control. We have over 1,400 type specimens, and we are trying to image them all to the resolution limit of light, which is 0.2 microns. So that's all of them, right? Now, I guess the question, one of the first questions that comes to mind when you're trying to do a project like this is, well, is this the right time? Right? Maybe in 20 years from now, someone will invent a much better microscope, will produce much better images, and then, well, we've just wasted, our time. We have to repeat it all. Okay. Now, I'm convinced that that is not something I need to worry about, and I'm going to explain this with a little thought experiment. So please indulge this little detour. But let's just imagine that I've been given infinite resources, right? And that's not what I'm talking about. In a set of thought experiment, infinite resources to try to describe as many species, to as much my taxonomy as possible, right? But with this one condition, which is I'm only allowed to use one microscope to do this. So the question is, well, which microscope? Do I use a phase, a low temperature scanning electron microscope with all this resolution that I can use and get to, really, really high resolution? Or do I use a confocal microscope where you have all these, the ability to generate 3D models, which is, of course, really, really cool? Well, there's a couple of disadvantages to both. These are SEM, it's cumbersome, right? You're looking at it from multiple different angles. You have to reorientate it. It just takes a long time. Now, confocal microscopes, they have the problem of standardization. You image it, now you take a 3D model of it, or you generate one, and then 20 minutes later, you do the exact same thing. And it's different because it's autofluorescing still, and it's autofluorescing at different rates. So that standardization is a massive problem for taxonomy. So I wouldn't use either of these. The microscope that I would choose to use would be a tiny fraction, okay, of the cost of these. And it will be the phase contrast. It is the best microscope. It really is. It's 100 years old, the best microscope we've had, and will carry on having for many decades to come for my taxonomy. It's already been around for almost a century. Here's the prototype from 36, was invented by an ingenious Dutchman, Frits van Eyck. Here he's wearing his Nobel medal, which he won for this invention. This is, as I said, one of the best inventions, I think, that came out of the 20th century. He deserved to win that Nobel medal. And I'm going to explain why, why he deserved it. So here's a simple sort of representation of phase contrast. I've lost the laser.
There's no laser.
Oh, there is no laser. Okay, let's go.
There's a laser.
Yeah, there was, but it's gone. Anyway, this is a simple representation of phase contrast. You've got the objective lens. You've got I've taken out the stage. I'm going to make it a little bit simpler. The slide and the cover slip are going to go away just to make it a little bit easier. Now you've got the condenser, the annulus here. Now pay attention to this, the phase plate, right? So you've got the phase plate up here, then you've got light coming up that forms a comb and it shoots through the mite, and you end up with the light going into the edge here. Now it's not going into the phase plate, it's going up to the edge. It's not passing through the phase plate, but some of that light will it'll get scattered as it hits the mite, and it's directed up in through the phase plate. So you've got transmitted light that's missing the phase plate, scattered light that's going through the phase plate. And a lot of that scattered light is in phase, in other words, aligned with the transmitted light, okay? But what happens is it goes to the phase plate and now it's shifted out of phase, and you end up with what's called destructive interference. And when you look at this, you end up with these, when you're looking down at the eyepiece, because they're kind of canceling each other out, you end up with this dark patch here, this dark area. Sorry, voice going. But then you also get some of that scattered light that's basically shifted out of phase by the specimen. And then when it hits the phase plate, well, it's shifted back into phase, and you end up with constructive interference, and therefore a bright area, okay, as well as a dark area. And this phase contrast utilizes this contrast, okay, this contrast between constructive interference and destructive interference, and you end up with these really, really good images of mites because of this. Just to give a bit of historical context here, this is Micrographia, arguably one of the best publications, biology publications to come out of the 20th. It's all good? Okay, to come out of the 1600s, you've got Robert Hooke, who basically depicted all these different microscopic organisms. One of the ones that he depicted was Pterophagus, the cheese mite, okay? But when you look at this, it's not a very good depiction. It's kind of inaccurate, and you think, well, yeah, it could have done a better job. But then you're thinking, well, wait a minute, full marks to the man, okay? Because this is what he was basing it on. Is that what you know?
No.
Yeah. Okay, because this is basically what he based his drawing on. And so you're thinking, you know, essentially for hundreds of years, mite taxonomists, for hundreds of years, this is how mites looked under a light microscope. Right? Actually, mite taxonomy was floundering in the dark ages, or you might say more aptly, the bright ages, right? There you go. It's essentially, it wasn't until the 20th century that Zernike came along, invented his phase contrast microscope, okay? And you end up with this glorious detail, there's all this information. And what's really important about this, not only can you see the mite properly, but you also have this sort of standardization that you don't have with something like confocal. I can take a slide, look at it, this is the one that we're using for digitization. Look at this slide under this modern one, then find the prototype, put it under there, and the images will look very, very similar. Maybe a little bit of improvement in resolution with our modern one, but very, very similar. That standardization is critical for good taxonomy. So my taxonomy is still in the era of the Zernike era, and I'm sure it's going to remain in this era, okay, for some time to come. But I guess that just leaves the question, well, why have we waited till now to digitize? Obviously, this is the right time, because essentially it's taken 80 years for digital technology to catch up with Zernike's genius. So the last 15, 20 years have been transformative. We now have sensor arrays that can capture everything to the resolution limit of light. We have terabytes of storage were a problem like 10, 15 years ago, and that's what we need, kind of a couple of terabytes. Well, that's now nothing. It's really, really cheap. And of course, algorithms, stitching algorithms, GPUs, everything has become much, much faster. So it's all eminently doable on a relatively cheap budget. So this is the perfect time. Anyway, now, moving on to you.
All right.
So I'm going to switch.
Okay, so I'm going to talk about how we created a standardized imaging protocol for our type collection. And to date, we've imaged all of our holotypes, which is 260 slides. And we imaged 75% of our paratypes, so 860 slides. And then by the end of this project, we'll have 1,400 slides digitized. Okay, so in order to create the protocol, we had to kind of consider what characters we needed to prioritize while we were imaging. And for Phytostaids, their classification is largely based off of the presence, absence, and morphology of their dorsal CD and the characteristics of their ventral plates. So moving forward, the consensus was that we needed to create image-stitched composites that represented these characters in like a meaningful way. So this is an example of what image stitching kind of looks like. I'm using a Zeiss axioscope with a Canon mounted to it, and I'm just scanning the mite, taking images, overlapping images. So one dorsal side of the mite will take about 30 to 60 images, and we're focusing on the dorsal characters. And then we'll repeat this process on the ventral side, focusing on the ventral characters. And then, so once you have your images, you can import them into a photo stitching software. The one that we chose to use is PTGUI because it has a high processing value. It doesn't require, it requires a very short amount of time to process high volumes of high resolution images. So you can see here, I imported 40 images into the software, and it came out with a composite in about 30 seconds. So as a result, you get something like this. You get a very high resolution view of the dorsal side of the mite and a high resolution view of the ventral side. And you can zoom in pretty far. This seta on the side here is a J dorsal seta, and it's about 6 microns long. And then on the ventral side over there, there is a pore circled, and that has a diameter of only 1 micron. So the resolution is very good, and the results were really clean. And you know, if we're imaging and it only takes 5 to 10 minutes to image a slide, and then it takes only 30 seconds to stitch it, we thought reasonably we could image all 1,400 types this way. The caveat was that there are certain species level structures that are not captured with our ventral and dorsal panoramic views. So we needed more depth of field for like additional characters like ketotaxy, the cheliceraal characteristics, and the genital structures like the spermathecae. So we decided to start image stacking these characters. And to do that for all 1,400 slides would take too long. So we decided just to do it for the holotypes. And then if our collaborator wanted to request additional imaging for maybe one of our paratype slides, they could do so. So this is an example of the focus stacking that we're doing. We created folders for each of these characters. On this side over here, that's 15 images that represent this tarsus and tibia of a leg 4 of a Eusaeus slide. And what I've done basically is focus from the dorsal side to the ventral side, taking images at every focal change. So we get the full representation of the depth of view. And then here is me just flipping through those images on your default image viewer, and it kind of creates like a simulation of what you would see in the microscope. And this is another example. This is another character that was important to consider. This is a chelicerae. You can see from the dorsal to the ventral side, you get both the movable and movable digits, and you can see all of the teeth. And then in post-processing, we used all free software. So we used GIMP, which is a free open source photo editing software, to crop our images and to create scale bars. And then to automate some of the process, we used a batch manipulation plugin called Behm, where we were able to embed scale bars into all of our focus stack folders automatically. This is using the watermark manipulation set. So we were treating scale bars as watermarks essentially. And this is a summary of our equipment. So we use Zeiss axioscope, Canon cameras, and then again, all of our software was free except for PTGUI. That stitching software is where you want to spend money if you want to do something like this, because of how fast it is. There are free alternatives like Hugin or Huggin, which is another plugin for GIMP, but there's a little bit more of a learning curve, and it doesn't have the same processing power. So that's it. Thank you.
We have time for a couple of questions for the two presenters.
Are these images available in a public database anywhere?
They will be at some point. We're still, in early days, so, yeah.
What photo stacking software did you use again?
It's manual.
So we're not actually, we haven't used photo stacking yet. We just used the PD GUI, the stitching. We may photo stack some of the legs when we do, but we also like the idea of keeping them free so someone can look through them, you know what I mean, like keeping them as an actual raw Z stack.
I used a Zerini with good success.
Yeah, Zerini's good, and there's others as well. It's difficult to know right now what we're going to use. So,
Okay.
Yeah. Yep.
I just want to take back that drawings are not important.
What's that?
I just want to take back that drawings are not important. But you guys said it's really important.
Well, no, but that makes a good point because there's a lot of interpretation with drawings, and I think it's important to have, when you have drawings, you also need, I think you should publish your face contrast images and things like that. So I think you're actually right that there's a lot of reason for skepticism about descriptions that entirely rely on drawings.
Last question.
What's the approximate size?
The composites, they're about 300 megabytes per image.
A file format to like a TIFF.
Yeah, TIFF with a compression.
Thank you so much. All right, our next presenter is on Zoom, and it's a great pleasure and an honor to have here Matt Yoder, which is the big mind behind TaxonWorks, HAO, at least the construction of the database, and I'm really happy that he's presenting with us today. It's perspective on TaxonWorks intersection with biological systematics. Thank you, Matt.
Thank you, and hello everyone. Can you hear me okay?
Yes. Yes.
Okay.
Yep.
Thanks a lot in particular to Davide for being a wonderful community member within the TaxonWorks community. This is a really nice opportunity for me to sort of step back and think about my career as a systematist. And it's been really amazing and sort of emotional to see everybody's connections to kind of the past work that's been done and to see sort of the next generation. So thanks a lot for this opportunity. So today I want to talk about TaxonWorks in general, and I want to do it by giving you a bit of how I got a perspective on the kind of things that were needed. So talking a little bit about the past, and then talking about some opportunities for collaboration in the future. And while this is sort of my perspective, I want to say that this is really a lot of work from a team of people. This is the species file group at University of Illinois under the PRI, Prairie Research Institute, INHS hierarchy, I guess. We're an endowed group, and part of the reason that I joined is that it's really a fortunate opportunity to sort of make the tools and create the tools that we sort of need collectively as a kinds of researchers that are presenting here today. So stepping way back, how did we kind of get here? One of my early influences was joining Texas A&M as a graduate entomology systematic student. And my original job was to code the TAMU insect collection databases. And to my knowledge, there are still some aspects of those databases being used today. That was over 20 years ago. My first revision during my master's was on Entomachus, and it quickly became clear to me that if I'm going to do this as a career, I'm going to need tools that are going to help me make the repetitive parts of that process much less mundane. And so here on the right, you can see a materials examined section. And in my experience with creating these collection databases, it became clear that I wanted to create my own specimen database and sort of generate these material examined sections automatically. And so this is an automatically script generated block of text. When the reviews came back, they said, hey, you should invert it. And so I just had to change the algorithm and boom, it was changed. And keep in mind that this was over 20 years ago, and I'll touch back on that a little in the future. Throughout my graduate studies there at A&M, the informatics side of things kind of expanded for me. I was in a PEAT project, the NSF PEAT project, that paired young researchers with people who were essentially retiring. And one of our goals or thoughts that emerged was that instead of having somebody retiring and pairing up with a junior sort of member and just kind of talking about it, what if they could also pass on their career database, their career knowledge as it was digitally provided, sort of their digital workbench? And that led to the idea of a project called MX that covered a lot of different data, not just for taxonomic work, but for a bunch of other projects as well. And at the same time, there was a lot of projects like MorphBank and MorphoBank that were emerging from the NSF process, and I had a lot of opportunities and was very fortunate to be a part of those workshops. From there at Texas A&M, I moved on to Norm Johnson's lab, and Norm was, you know, various folks here are alumni of Norm's lab. And Norm was working on the same things. It was an alpha taxonomy factory grant where the project was to describe 1,000 species. And so he had similar software, his VCS lab, to what we were building in parallel. And so that really influenced what we were doing. And you heard Davide mention the HAO. We got a grant from NSF to build this project. Nobody really understood what ontologies were, but it really let us dig down sort of as traditional morphologists to take advantage of tying that into new technologies and innovative approaches. We got a lot of funding to do kind of the basic research that we're all really interested in and that we want to do in. So I think there's a theme of really trying to adopt and embrace new technologies to let us do what we consider traditional things. And we've got good examples of how that makes a compelling funding argument to NSF. So during the HAO time, there was an opportunity that came up here with the species file group that had just sort of moved into its new digs at the INHS. And I took that opportunity in part because I saw it as a very rare chance to use a fixed set of resources to provide digital tools to what I thought was an underserved community of researchers, and that being taxonomists and collection managers. And there were similar projects being done in parallel. The 3i project from Dmitriev also appeared during the PEAT project, and the species file project that a lot of you have used the resources for, that were sort of all kind of aligned. And what we did is we decided to, when I joined up, to move all of what we knew in those 3 projects into a project called TaxonWorks. And so TaxonWorks is used for a lot of different things. We sort of classified those different projects into a couple of loose categories here. This isn't comprehensive. You can see that we have sort of world-class nomenclature handling according to the rules of the ICZN and other codes of nomenclature. We have digitization capabilities as your collections in Florida that now are engaging with and understand. There's descriptive elements. There's an excellent literature basis for citing anything in the whole database, and we've got some strong geospatial components as well. And what sort of emerged as we've made this platform available is that people are using it in a lot of different ways. And so just grossly, you can see the colors here. Some projects are using it completely for curation, digitization. Others are building matrices, and they just wanted to create a platform where they could manage their morphological characters, attach their images, add their annotations. And some are much more diverse. They're kind of equally split. Some of the richest biodiversity informatics data sets in the world, I would argue, are now in here. Things like the Orthoptera species file, the Universal Chalcedonidia database, the 3i Hoppers Kenarenka database are all in here. And they've integrated data in a more balanced way as to compared to these other projects. But it's really been both a plus and a minus to see this diversity. We want to be able to help people on many fronts. However, we also need to concentrate on specific features. So just to highlight a couple of the quick projects that come out of TaxonWorks data. TaxonWorks is a workbench where you come in and you log in and you work privately, but we expose that data in a lot of different ways. There's a companion project now called Taxon Pages that replaces what the species file looked like in the past. You can very quickly get these up and running with very little experience, with very little expertise off of GitHub, hosting your own pages, for example. But we also at the Species File Group host pages. So we're serving over 200,000, I think, taxon pages in this framework. And this is expanding with collaborators producing new sort of panels and elements going on their own to fund contributions to this open source code base. We are deeply sort of integrated with the catalog of life. Around 19% of the catalog of life now flows through TaxonWorks, either through directly curated data or through the sort of programmatic pipeline. So there are data sets where Jeff will scrape them off the web and feed them through the TaxonWorks sort of logic and models to validate them, and then produce the data set format, the ColdDB format that goes into the catalog of life. So those data aren't curated in TaxonWorks, but they make use of the engine. And I think this is a theme that we find really important. We want to be able to make the tools that we build accessible and useful, not just through user interfaces, but through a lot of different ways. iNaturalist uses TaxonWorks names and nomenclature as authoritative files for a number of insect groups, including the Orthoptera, Plecoptera, and there's more coming down the road. And what we've done is we've created a really neat way where they can come onto our issue trackers now and say, hey, this name is not right. And we're starting to see a big uptick in the number of feedbacks, sort of contributions that we're getting from iNaturalists to our taxonomists, particularly if you'd like to see it, to check out what the Orthoptera issue tracker is looking like. I can share some links later on. And TaxonWorks is being used for digitization right now. As a systematist, as somebody building out monographs, you are managing specimen databases. And it wasn't a big leap for us to sort of extend the functionality within TaxonWorks to serve collections. And this really gives us a nice opportunity to kind of merge kind of traditional digitization requirements with maybe this extended digital specimen concept where we can attach in keys or digital observations that you would consider for morphological matrices. As I mentioned, we also do nomenclature, I think, in a world-class way. We can export what is probably very familiar to most folks here, these kind of classic nomenclatural catalogs, and that will grow over time. So stepping back, thinking about that monographic process and what we can do and not do, and sort of where are we? And I think what we've got here on the left is that we can manage a lot of different data right now. One of my big goals was to be able to render that monograph exactly like we'd be able to see it in print. We do have models for DNA, and we do have excellent architecture for linking extracts to specimens. However, we don't have any DNA related user interface sort of tools that would let you sort of manage your day-to-day workspace for a wet lab, for example. Now, those kind of things did exist in things like MX, so there is a longer term planning to kind of make that possible. Now, managing data is not producing products necessarily. And so rendering out that full monograph is a real challenge. And remarkably, there's a couple of things like that specimen management, like building that material examined section, that aren't fully realized even 20 years later. And going back and looking at this, building this talk, I'm like, well, why are some things being built out and why were they prioritized and why were other things not? And I think it sort of reflects this idea that increasingly we're prioritizing things for this digital world, right? In this taxon pages format where we can share data about specimens. We can share it in a standard, the Darwin Core format standard that you can pull down, and it's much more actionable than sort of a traditional material examined section. So that tradition is critical and it's important to get it into a human readable standpoint. And it's a matter of sort of balancing that and making a lot of people happy with these kind of future digital needs as well. A while back, we wrote a paper in SSB that there was a special issue that talked a lot about different kinds of monography there. And we wrote a sort of model for how we think about monography, and I'd encourage you to check out that paper. One of the things that we have to consider now is that what you see on the right side is a single specimen and its metadata. And if you think about, if I go back and I think about my thesis that had 1,000 specimens, or the monograph I did with Norm that had thousands and thousands of specimens, and I imagine this graph multiplied by 5,000 times, condensing all of that data, which is very calculable for machines, but into a human readable format is a real challenge. And there's a lot more, of course, coming on. That's not just that we need to be able to handle DNA. You heard a lot of nice references, and it really warms my heart to hear people talking about the HAO. There's a lot of cool work that has gone on since then that will trickle in and influence what we do in TaxonWorks in the future. Gross morphology is not the only way to describe things, and increasingly it's important to look at, we can argue that we should do more morphology, but we should also do more cellular biology and proteomes, all of the different omes that are related to our cells, etc. 3D is far more common, and there's been a lot of requests for us to handle that. And then species concepts, like how do we actually define our species and what's useful, for example, thinking about eDNA, that's influencing our thinking there. And we, of course, are thinking about how to turn this graph of data into an aggregator so that it's computable by everybody, and how AI might have a role in there. For example, while the other talks were going on, I asked AI, ChatGPT, a large language model, about the HAO, and it seemed to do a really nice job of getting me the IDs for these terms. And then I asked to cross-reference the gene ontology and find me model organisms, and it's getting there. It's not going to be a one-stop solution, but I think we have to embrace it and hopefully get funding from integrating with it that trickles all the way down to the things that we love to do, going into the field and sampling leaf litter, doing that morphology, storing those images. It's really important that we integrate. Okay, I'm almost at 15 minutes. I want to plug two things. One, we have our annual conference, TaxonWorks Together. You can go to gettogether.taxonworks.org. Please check it out. You can register. We have our first set of roundtable speakers that are there. It's all virtual. There are lots of opportunities if you would like to present or ask or show your problem, show your biodiversity informatics challenge. There's opportunities there. Reach out. We also have weekly support meetings. I can share the link. You're welcome to come and ask anything you want about TaxonWorks and our community. And we look forward to hearing from all of you in the future. Thank you.
Thank you, Matt. Thank you for the presentation. We got a couple of questions. Yes, Jesse.
I'm wondering what you're doing to ensure the longevity of this resource online, because a lot of databases they're up for a while, they're really useful, and then funding falls through and they don't have money. So is there anything you learned from like HAO or VSIS Lab that you've been able to apply to make sure this is available for a long time?
Yeah, it's a great question. So, we're working with VSIS Lab to see about translating some of their material. We're in discussions with Norm into TaxonWorks. I think that there are a set of new repositories that are changing the game somewhat in terms of being able to make things accessible. I think we have some text, for example, on the webpage that talk about your strategies. And one of the big strategies you should ask, as you alluded to, is before you get into a platform, how am I going to get out, right? And what are the alternative platforms for me? And that has to be part of the conversation. And we think about it. I mean, we are really uniquely in a really unique opportunity in that we are endowed. So our longevity is longer than any project right now. If you look at the history of the previous two projects, then I think longer than most projects that have been out there. And things that we do are, there's a bunch of verbiage on our website on speciesfilegroup.org that talks about our strategy. Should we go away, which could happen, things are very turbulent. We make our software open source. We commit a lot of time to being able to set it up so that you can download it and run it offline. We're working hard right now with other people who would like to run their own copies so that they can be sustainable on their own. There's a bunch of strategies there, but we also want to make sure that the data follows standards at some level, that you can access the raw data and download it. And yeah, I think it's just really a big part of it is having that conversation and knowing what you're getting into and knowing how to get out and really thinking about that hard.
You got a question on the chat that is from Doris. Great talk. I've been identified tons of aphid species from suction traps. do I need to get a QR or an archival number to be entered into TaxonWorks? So how can we collaborate essentially?
Yeah, so Doris is a member. She collaborates with Dimitri, I believe, on the 3i. We have a number of different classes of objects that archive, including, as of the last year, we can basically handle and we'll have import capability iNaturalist field, what we call field occurrence observations, where you don't have an object in place. So it depends. I would strongly recommend that you have catalog numbers, be they QR codes or catalog numbers. Modern taxonomy, you need to have those codes on your specimens. If you want, we all know what it feels like to go back and look at an old collection and be reading the old literature and go, oh, this is what Nixon looked at in 57, right? It has to be. It's got to be the only one. And this is a wonderful feeling. And you can get that feeling, and you can make it possible for the future taxonomists to get that feeling far easier if you put a catalog number on your specimen. So do catalog your specimens. We handle a lot of different identifiers in TaxonWorks, and we allow the system to create what we call namespaces. So Doris, if you have your own namespace, you could create those, and at minimum, that's a start. Then you would work with Tommy or other folks who may have archival identifiers to think about adding those as well. So there's a bunch of different solutions there. It's extra time, but it is actually, it's critical, I think. And it's not that hard to have a huge bucket of them to slap on those identifiers. Yeah, especially if you're going to want to share those, you will make them accessible. You'll make your data citable in many new ways coming off of GBIF's tools. I think it really is critical. Vouchering is huge, and it's going to be huge to expand the role of vouchering for our collections if we're going to want to get funding for exactly your kind of work, I would argue.
Fantastic. Thank you, Matt. It's been amazing.
Thanks so much, Davide. Thanks for the opportunity.
So for all of you here, we got lunch provided by the CSE. Of course, it's not possible for those of you on Zoom, but we are also having the poster session that you can read more at the back of your program. And you're gonna have the physical poster outside of the room. And for those of you online and for those of you in this room, we're gonna also slideshow it during this period. It's about 2 minutes per poster, so you should have time to read both of them in turn. So thank you all, and I'll see you back at 1, so we keep on the pace. Thank you. Then you want to sit there? You want to sit there?
Afternoon section, with the first talk, we have a live Zoom presentation by Donald Chandler, and it will talk about insects of the presidential range in New Hampshire. So Don, if you are online, the stage is yours.
Don, you are still muted, but we can see your slides. Don, you're still muted. Now you're not muted, but we have not heard you say anything yet. We can see you now. We can see your.
Maybe he's on a different audio.
You haven't changed the audio settings.
No, him maybe.
Yeah, Don, I recommend that you change your, whatever you did yesterday when we tested this, change your settings to that. Don, we still can't hear you, and if you're trying to convey what's going on, maybe you can type it in the chat if you're having trouble. But I don't know what else to tell you. Like all the sound here is working on. Yeah, so Don, in the audio button on the bottom left of your screen, there's a little caret symbol next to the microphone. Click on that. There should be options to select a microphone. There should be a lot of different audio options. You should look through those and use the same ones you used yesterday.
Can invert the presentation.
Well, right, Don, you just turned your video on. Can you try talking? We can't tell if we can hear you because you're not saying anything.
Can you hear me now?
Yeah.
Yes.
Didn't know if you wanted my video on or not.
That doesn't, it's up to you. As long as your audio is working, you can see your slides, you're good to go.
Okay, well, I'll turn it on.
Well, no, it's not talking. Don, you can start whenever you're ready.
Ah. Okay. I can't see my little picture though.
I don't think anything about that on your end. Okay. Screen share is working. We can see your slides. So as long as you navigate between them.
Okay, everything's ready to go.
Yes.
All right. It's insects of the presidential range in New Hampshire. The presidential range basically runs north-south in the White Mountains, the central part of the state. And it's well known because it has the highest peaks in the Northeast in New England. And in particular, these peaks are of interest because they are the best representation of an old type of biome that was common in New Hampshire at one time, the tundra biome. And here if you can see a beautiful view of the Alpine Gardens. And in the far distance is the Tuckerman's Ravine. Those of you who are skiers will be familiar with that. And it's, I'm not a skier. I would never go down that in the winter, but people do. Let's see. Now, why isn't that working? My screen seems, there we go. New Hampshire really has a young vegetation and fauna. Roughly 11,000 years ago, 10,000 years ago, the Wisconsin or the Laurentide ice sheet began to retreat. And along with its retreat came a particular type of flora, the tundra flora. And this is a type of grasses and herbaceous plants well adapted to living near ice fields. And as the glacier retreated north, this particular fauna and flora moved north with it, except where there were mountains which could maintain this really cold, dry climate. This flora and fauna has sort of been hanging on. And as it continued to warm, of course, the flora and fauna would move up the mountains. And in New Hampshire, the mountains are high enough that it has basically maintained itself over this period of time. To the right is, upper right is the Arctic tundra. And basically it's the treeless plain based on a Finnish word. And they're with basically grasses, low herbaceous plants, and the soil tends to be saturated, it's frozen, whatever. There's plenty of moisture. The other kind of tundra, which we see in New Hampshire, is alpine tundra. Same types of plants, same conditions, cold and dry, but there tends to be really well-drained soils. So they are different, but yet the flora and to a certain extent the faunas are very similar, if not the same. Today in New Hampshire, we have basically three major biomes, plant, animal-based ecological relationships. And this was recognized to a certain extent by Samuel Scudder, who was famous entomologist in the late 1800s. And he noted that there's basically three different areas, types of vegetation, biogeographic areas, whatever biomes, that are found in New Hampshire. And so this makes New Hampshire unusual, and it has such a diversity of habitats in a relatively small state. And the first is the tundra biome, which is in the White Mountains area where the presidential range is. Excuse me. The second is the boreal biome, basically dominated by coniferous trees, so colder and moister, and basically the upper half of the state. And down here at the bottom is the eastern deciduous Alleghanian forest biome, the lower half of the state, dominated by deciduous trees. But yet in the middle of the state, basically all of these intermingle, with the tundra being sitting on top of the mountains, but the boreal and the deciduous forest basically intermixing in a way that the straight lines completely are misleading in terms of implying that there are definite boundaries there. They really interdigitate in a way that makes the place, New Hampshire, a wonderful place to go collecting so many different kinds of biomes, habitats, dry, wet areas. It's a marvelous place. Okay. Now, generalizations are in, if you can go to, this is just east of Mount Washington at Glen House, and you can actually see the three biomes without any trouble. The bottom, the mixed northern forest, where the boreal and deciduous forests are mixed together, approaching the fall. Colors, trees are beginning to turn. Here, you can see coniferous forest, a very nice zone dominated by conifers. And up here at the very top, you can see it looks just sort of grayish. That's where the alpine tundra is. There's lots of rocks there, but lots of grasses, herbaceous plants. And the alpine tundra basically is 4,000 and higher feet. The lower elevations within that 4,000 feet tends to have plants that are called krummholz. And these are actual trees, but they never grow very tall. The winds are so cold and icy and strong that basically they prune these trees as they grow and kill the growing tips. So they only survive in protected areas. And this makes it a much more varied habitat, open with many more species, and that there's a nice leaf litter, very protected. In general, this is where I target when I'm up there because I'm after beetles that are in the leaf litter. And this would be the so-called subalpine area, where you can find krummholz above 4,400 feet. It's very hard to find it. These are generalizations. The east and west side of the presidentials are a bit different in terms of the west side gets a much stronger effect of storms, the east side less effect. It affects these particular boundaries. Now, C.P. Alexander, well-known tupulid expert, wrote a paper on the insects of Mount Washington. He said Mount Washington is an Arctic island in the temperate zone. Well, so true, so true. Just a little over 2 years ago, we had a nice day up on Mount Washington. Temperature minus 46, wind gusts up to 127 miles an hour. The wind chill was basically about -109 Fahrenheit. It was a little more delightful down here in Durham and Dover. It was only, the wind chill was only about -30. This happens occasionally. Doesn't happen all the time, but it was memorable. Now, what makes this condition possible, and in fact leads into what's Arctic and what's alpine in terms of tundra, is that over here there's a pressure temperature curve. Don't bother to read it, but basically as you go up in elevation, air is less dense, it retains heat less well, heat radiates away, it gets colder. And so basically for Mount Washington, which is 6,288 feet, for every 1,000 feet of altitude, and you go up 6,000 feet, it's about 30 degrees cold on top of the mountain than it is down near the ocean. So if you're having a pleasant day at 80 degrees in southern New Hampshire, it could be 50 degrees up on top of Mount Washington. Always something to keep in mind when you're planning a vacation. The other thing is with latitude. Again, looking at temperatures, global temperatures, basically for each increase of 320 feet, it's equivalent to about 1 degree increase in latitude. That means that if you do the calculations, sea level to Mount Washington is approximately 20 degrees latitude increase. Mount Washington is at 44 degrees. That means that at the top you get a climate equivalent to 60 locations at 64 degrees latitude, which puts you in northern Labrador. Now, I haven't really been there, but I have to imagine that it may not be exactly the same as the top of Mount Washington, but a lot closer than I would particularly enjoy. So, focus really is the alpine, subalpine tundra, which is unique, not unique. In New England, it's very rare. And there are a number of mountains that are high enough to have alpine tundra, basically above 4,000 feet at these particular latitudes. And so over here we have Mount Katahdin, 1,000 feet lower than Mount Washington. Number 4 here, Sugarloaf and Saddleback. This big blob right here is basically the Arctic subalpine areas of the presidentials, the White Mountains. And Scudder formed a very nice little graph right here. The main thing is that it is the highest, the largest, and the most continuous such area of tundra in actually eastern Canada. There's no mountains in eastern Canada that are as high as Mount Washington. So as the glaciers retreated and the tundra habitats moved north, these habitats became isolated on these higher peaks, and by far the best example of the flora and fauna of the alpine tundra is actually in the White Mountains in the presidentials. So the insect part. Why is that nice? I mean, there's a very high mountain on the Labrador-Quebec border. Takes 7 days to get there. The good news is for Mount Washington and presidentials, you can drive up on the Mount Washington Auto Road to Mount Washington. You can take the Cog Railway, which is a fun way to go if you ever have your family. That's everybody who does it says it's a memorable experience. And you get up to the alpine areas in here. And then, of course, the alpine, subalpine areas are widespread. And there are hiking trails everywhere. It is an extremely accessible, large example of alpine tundra. And it is easy to go collecting there. Surrounded by National Forest, White Mountain National Forest. The very top is a natural research area, but it's a good place to go collecting. It's very possible. Now, Annie Trumbull Slauson, winter summered up at the Grand Hotels in northern New Hampshire, very popular with reasonably well-to-do people in the cities further to the south. And she collected about 2,000 species. Alexander noted that about 3,000 species had been taken in Mount Washington. Suspected maybe that the numbers aren't quite that high, but those numbers are out there. So at the university collection, the UNH collection, we have about 13,000, probably closer to 14 now, insect species identified. 1,000 of these are basically taken from the Mount Washington area. And we have enough species, and these are in the collection, not known necessarily all known from, or not all the species known from Mount Washington and the presidentials, but we have enough that we can really look at some characteristics of species distribution patterns. And number one is, are there any species that are found only in the presidentials, in the alpine, subalpine area? Number two, are there alpine, subalpine species that have populations isolated on the northeastern mountains, but are also found commonly in Canada or the western United States? The Rocky Mountains have lots of alpine, Arctic, subalpine areas. Or which ones are generalists? There are widespread boreal and eastern deciduous forests. And despite those really sharp lines, the eastern deciduous forest goes into southern Ontario, goes into New Brunswick, in isolated long patches. Certainly boreal forest is widespread there. So basically widespread. Which is which? Well, we have some ideas. Endemic to the USA. Well, I'll throw in the one plant from the presidentials, Robinson's cinquefoil, Potentilla robinsiana, one species, a plant. That's it from the alpine, subalpine areas. That's unique. 3 species of insects. One is the stonefly, which is found at Lakes of the Clouds. That's the only place it's found, although we did collect one adult that had apparently been blown off the mountain down into the valley below. One leafhopper, Samotetics alexandrii, feeding on something in the lawns, the so-called lawns, the tundra around the peak. And then one species from Staphylinid that's found basically in leaf litter. 3 species endemic to the USA? We think so. Now, subspecies. Okay, there's 2 famous subspecies, White Mountain Arctic, White Mountain Fritillary, both found in the tundra habitats of the presidentials. They are isolated populations of very widespread boreal tundra species. And so they receive some recognition for that. But the point being is they're members of species that are very widespread. Secondly, widespread alpine, subalpine species. There's a number of species that we know the beetles pretty well. They're easier to collect, that are found in the alpine area, subalpine area. But they're also found widespread in Canada, even to the Rocky Mountains. They are not separated into subspecies. I never know about the Nebrias. And then there are also, yes?
Sorry for interrupting you. You have 30 seconds to wrap up, if you can.
Thank you. I'll try. And then there's widespread eastern boreal species that have. So the Aleocrine road beetles of New Hampshire were studied by us, and they were particularly interesting in that we picked up 257 species in New Hampshire. Almost all were Canadian also, with species names like Atheta prurohensis, up here, Boreophilia nomensis, Mount Washington. So basically the fauna of New Hampshire is a Canadian fauna, and very little is shared with the states further south. And interesting also is that 31 New Hampshire species for the United States were only shared with Alaska, of course, widespread in Canada. So, questions, maybe.
Thank you, Don. We don't have time for questions, but check the chat online. You have a few questions there, I think.
Thank you.
Yes, thank you. Now our next speaker is Wills Flowers, and he's going to talk about the cosmopolitan genus Phyloides in the New World.
Good afternoon. This is a project that I got involved with 50 years ago and I was an undergraduate at Cornell University, and we were at a summer practice requirement. So I did mine at the New York State Museum at Albany with John Wilcox, who was a world expert in the Gallaroo science. And as a naive undergraduate, he picked out a well-defined simple problem as a small genus of flea beetles that there were only 6 described species from the United States. And he had a few specimens that looked like something different from the Southwest. And so I worked on it, and the time ran out on the internship. And I got distracted into other things like getting a master's, getting a PhD, as one does. So what is the Phyloides? This is the genus I worked on, and this is the most easily recognizable genus of all the North American flea beetle genera, because it can be distinguished by it has only 10 antennal segments. All the others have 11. And on the hind tibia, tarsal insertion is pushed way forward, leaving this big process behind the tarsus. The distribution, known distribution today of Phyloides, there's around 200 species worldwide, of which most of them are in the Palearctic. There are now 19 species recorded from the Nearctic, but 7 of these are adventives that are of old world origin. And taxonomic workers on the genus, there are a number of them in the old world, some of them still very active, particularly Konstantin Nadine. He's describing a lot of new things from Eastern Russia and Northeast Asia. In the New World, we have Henry Fall, who wrote the original key to Phyloides in 1933, and then there's some other guy. The characters to distinguish Phyloides below the genus level get to be a little complicated in the sense that they're sometimes not that distinct. Color is still a main one. Structure behind tibia. The sculpture of the head in Europe, they're particularly put emphasis on the ocular sulcus and how it connects, if it connects to the antennal insertion, whether or not they're functional hind wings. Spermatheca morphology is good and quite diverse in Europe. And then there's some emphasis on the general body shape. And here are the original 6 from the United States with two additions. Appalachiana is a recently described Phyloides from the Great Smoky Mountains area This is a flightless species that lives in leaf litter. And this is just about, this is, I think, the only known leaf litter flea beetle in the United States. There have been a lot of new leaf litter and moss feeders from the Caribbean. And I mentioned that John had specimens from the southwest of something new, and that's, here's an individual that's labeled TX1. Now, I picked this project up again when I retired. And I got some few more specimens from the National Museum, but Sean Clark and Ed Riley sent me two big collections of unsorted Coleodes that they'd been collecting out west. The two most common North American, or the most widespread North American species with many duplicates in those two collections, Punctulata and Convexior. And both of them had a question of how do I distinguish these two? And they kind of look a little bit different from the external characteristics, but there's a lot of variability, whatever you pick as an external character, punctation, body shape. And similarly, in the male genitalia, the male genitalia aren't that informative from species to species. But in the Spermatheca, in Punctulata, it's the only North American species with this contorted spermathecal duct. Whereas in Convexior, the spermathecal duct is just a smooth little curve into the reservoir. But now these are the adventives, the stuff that's been found, the old world species that have been found over here. Most of these apparently came over in agricultural products in the 50s and 60s. The two new additions is Attenuata and Dulcamari. They've just been found in the last, published in the last year. Algerica is something I found in Ed Riley's collection. There was one specimen in Texas, but it's clearly this Mediterranean species. And I published that 2 years ago. Calcamera was brought in deliberately as a hope for biocontrol of the musk thistle. And it passed all its tests, its feeding tests with flying colors, and it was released, but apparently it didn't, it hasn't established itself. The rest, in general, these adventive species have more or less stayed in their original point of origin in the United States and Eastern Canada. Two exceptions which I could track from these collections is the Coleodes affinis, or the European potato flea beetle, and Coleodes napi. And actually, I had a hand in finding both of these things in this country, as during our activities in Albany. One fine morning, John Wilcox and I decided to bag the laboratory and go out and do some collecting down at the Port of Albany. And we came up with Coleodes affinis and sent specimens to Dick White at the Smithsonian, and it was published in one of the flyers. You can see they've moved into other parts of the United States, but not in any kind of a serious way, really, and which kind of indicates they're still relying on human agricultural commerce to do their moving around. Now, south of the border, there are 5 species of Coleodes published in that Jacobi described in the Biologia Centrale Americana. And thanks to Michael Geiser, I've got photos of all the holotypes. There's the two in Guatemala, then and 3 in Mexico. Now, in the Mexican species, Melanocephala and Capitata are also now known from the United States. They're down in the places like the Huachuca Mountains. We've picked up a number of individuals of another species of Coleodes in Costa Rica. This is all from in the Central Valley or on the sides of the Vulcan Barba and Poas. Now, in the literature, Coleodes is, they call it a Brassicaceae specialist. And in the North American fauna, a couple of introduced species in here. But you can see the Brassicaceae pops up repeatedly as one of the associated plant families. Now there are some other families, particularly elegans, which may be a Florida endemic. There's a few other things that look like it in other states, and I haven't quite decided if they're really elegans or not. But anyway, but you see Asteraceae, well, that's a collection record off of goldenrod. So they're not that likely to be an actual food plant. Punctuate is kind of interesting because lots of those collections from Utah were collected on sugar beet. And this is apparently, it was a pest on sugar beet. Sean says that there aren't any recent records from sugar beet because the cultivation of sugar beet out west has apparently collapsed in recent years. The last year's symposium, Jim Hayden told us about the mustard bomb, how it affects various Lepidoptera. And as Brassicaceae feeders, at least some of the Coleodes are dealing with the same thing. And there's been a couple of recent studies in Europe about the relation of chrysomelids, in this case, 2 genera of flea beetles with mustard plants. It's an economic issue, particularly in Europe, and the Phylogenetic is very damaging to oilseed rape cultivation. So this is a study in 2019 with Coleodes chrysocephala, which is one of our Adventives, as if you remember, and Phylogenetic striolata, which is a major species of the Phylogenetic group. And this is a diagram of the chemical changes that both of these species, both of these genera use to process the mustard plant chemicals. It's somewhat different. Chrysocephala apparently has 3 alternative pathways for detoxifying the mustard oils, whereas Phylogenetic tends more like to simply just sequester the whole thing in its body and use it as a repellent for its own predators. And last year there was a bigger study just of Coleodes. They did a molecular phylogeny of all of the old world species, and they found that there was, and then they mapped the host plants onto the species, and they found that there was a definite phylogenetic signal in the choice of host plant by the various species. So, and again, the biggest clade of Coleodes were brassica feeders. So getting back to our own side of the pond, the sure records of feeding. Again, they don't show that there's really many serious pest Ciliotes in the United States. Finis was a former pest of potato in Europe, and there's some old literature in German about how to control it and damage. But all the recent European literature I've seen, when we talk about potatoes, the big problem is Epitrix species. And Ciliotes affinis feeds in this country on Solanum dulcamara, which itself is a European introduced weed. Now, attenuata apparently has become a problem in Quebec on hops. And that's a recent quick creation of a potential new pest. It's also recorded as feeding on cannabis, which makes it kind of interesting for Florida, possibly. Chrysocephala, the cabbage stem flea beetle, is a problem in Europe, but apparently not a problem over here, maybe because it hasn't moved out of Canada, whereas the oilseed rape cultivation is out west. Nape is a brassica feeder, but it's only on weeds, both here and in the old world. And I mentioned punctulata tied to Amaranthaceae. A lot of the records I have more recently, it's feeding on, it's on or associated with sagebrush, which is, all the sagebrushes are amaranths. However, it's also recorded as a secondary pest of oilseed rape in Canada. And from what I know of the sagebrush ecosystem, of course, it's dominated by amaranths, but there are a lot of small genera of Brassicaceae in there as well. So teasing out exactly what it's doing in the sagebrush ecosystem might be an interesting problem for another undergraduate. Thanks.
I don't have time for questions, but there is one online and we can answer maybe later to that one. Our next speaker is James Aiden with a talk Messidae or Messidae, an unexpectedly diverse taxon with a pivotal phylogenetic position.
Thank you. All right, thank you very much for coming today and listening to this talk. This is going to be like 2.5 talks compressed into 15 minutes, so there will be no time for questions. I cannot be questioned. Do not question. So the Messidae, it's a junior synonym. This is a recently recognized family, elevated family from subfamily level 2 that's historically been in the Ichneumonidae. And the first half of this talk is going to be about species level diversity, new species that have turned up in the collection in the past year. And the second half is going to be about the phylogenetic significance of this. And then I'm going to spend a couple slides at the end talking about like some speculation, some kind of interesting speculation about the evolution of the female genitalia in Lepidoptera. So Messidae is historically, it used to be a subfamily, Messidae, and it historically was a dump group for little bitty tinniad moths that had produced wing venation. And about 11 years ago, most of those other genera were moved out. They're found to be true tinniads, but the Eudarcia, together with its many synonyms, including Mesia, was found to have a special position to it. So it's moved out of the Tinnidae. So currently Messidae consists of Eudarcia and one other monotypic genus called Pathrixina. Eudarcia includes well over 100 species described globally, mostly in the old world. There are a lot of new species in the new, about half a dozen described and many undescribed in the new world. So when I talk about when I say that this is a undiscovered, unexpected diversity, I'm mainly talking about what has turned up in Florida and potentially in the tropics, neotropics. So I'm not going to talk about these characters in this slide, but I'll be hitting a bunch of these things later on, off and on. There's a lot of diagnostic characters that you can see if they have a fuzzy head that looks like a typical tinea. It's a tinea, one of the two closed moths. They look like basically like that. But they also have a few, like the details of the antennae, the scaling of the antennae, and a lot of wing venation characters and genitalia characters can really verify that a given specimen is one of these. Like in Florida, the only thing that could be really confused with this on external characters is a single species called Diacorycia. So if you're going through traps, you might find a bunch of this, or you might find a bunch of these things. So one of the take homes from this is very preliminary. I'm going to show some very preliminary results, but I'm going to encourage you to please collect more specimens. We can't really advance to DNA or anything else until we have more specimens. What originally got me into this was that these things started to turn. We caught a bunch of these things in traps in the Malaise trap on Big Pine Key some years ago. Whatever this is, this had the longest valve of any Lepidoptera genitalia I'd ever seen, proportional to the body. These are like tongs. They occupy like half of the abdominal cavity. So I made slides. I thought, okay, whatever, they're tiniids. Okay, whatever. Maybe they're gracilariids. I don't know. I don't have time for this. Put them aside. So then a couple of years later, actually really last year, past 23, 24, this thing started to turn up at lights around Gainesville. And we also had some bug guide from other parts of the state. This, I believe, we only have females of this so far, but I think these are too closely related to distinct species. And this has been probably in South Florida since the 50s, but has been moving northward in the past decade. Wing slide, hit the books. Only Eudarcia has this particular stalking right here. So when everything else after that started to fall in place. This has the most fantastically beautiful wing pattern of anything in the genus. That's another thing that throws people off. Most of these are boring brown gray things. It's probably related to this thing, Protodarcia argyrophea. Protodarcia described from Puerto Rico. Protodarcia is one of the many junior synonyms of Eudarcia, but because these are unpublished combinations, I'm not going to. So with these two things in hand, I started to look around in the collection and actually look in trap samples. And many of you some of you in this room have collected specimens. I'll be mentioning a few names that are here. We have like 3 specimens of this thing from Big Pine Key in the lower keys. This is more typical of Eudarcia's genitalia. It's really boring. This thing is probably a native thing, endemic to Florida, Putnam County, Sand Hills. There's a few specimens in the U.S. National Museum. Tineidae species 30 of Don Davis. Don Davis is the late expert on tinioids, who I'll mention repeatedly. We've got a couple specimens from Central Florida. Also, it's in Arkansas and Texas. This thing is probably close to the type species of Protodarcia. This thing, singleton male, again, Malaise trap, Big Pine Key. I don't know. Species 5, I know from 2 specimens. One that I caught in a dry Malaise trap in my own backyard, and another one in a Malaise trap from Texas. This is neat because it has really elongate valvae, and these other bits up here, which look a lot like Eudarcia 1, except the pattern is a typical gray thing. So there's probably good. This would be good for a phylogenetic analysis, but these are good characters, but I haven't caught them. Some of these things are known only from females. So Eudarcia species 4, thanks to David Serrano for getting me out to Highlands Hammock. 1 specimen, it's perfectly good condition, but it's a female. Like to see more. Eudarcia number 9, I think, is collected at the same time as some of the other things in the keys, but I think the tails are a bit different, wing slides also. So yes, it would be nice to tie these together with DNA barcodes, but we need more specimens. These are awfully small. I don't really want to pull a leg until. Bathroxena heteropalpella is widespread eastern in North America. It's a monotypic genus. New state record for Florida. And we've got a singleton new species from Big Pine Key that Debbie Matthews collected in the 90s. So I can group these in species groups by genitalia. As I said, there's only about 6 or so species described in Andromeda so far. So why are these things so rare? One of the things is that they're not really strongly attracted to light. We're getting traps once in a while, but not often. They more often turn up in malaise traps. Dry malaise traps would be good. They're univoltine. They fly, at least in Europe, they fly only in the one generation in the springtime. Larvae make cases on lichens. So what the Europeans do is they often just collect it there, or they sweep over lichens in the morning. This is distributions of all the specimens that we found in Florida. As I said, many of these are singletons or doubleton specimens. So the second point about this that's interesting is that this little family, effectively one genus so far, seems to be sister to the rest of Ditrysia, which is the major radiation of Lepidoptera. I mean, this would be like, it's kind of like Orussa being sister to the rest of the Hymenoptera, or the flowering plants, for example. The Ditrysia is characterized by this unique modification of the female genitalia. In the primitive condition as moths is that copulation in an oviposition happens through a single orifice, whereas in Ditrysia, it's two holes. There is male Stixus phallis in here, spermatozoa follow up, go up to this duct, fertilization happens. This other canal, oviposition happens here. Okay, so copulation in different orifices for copulation and for oviposition. So about 10 years ago, Ruggiero and company published this big phylogeny of the Tinioideae and found that it includes, the Tiniidae and the bagworms, the Psychidae, and a couple of little families like that. They found that it was paraphyletic with respect to the rest of the Ditrysia. Wow. And then the Mysiidae is this Eudarcia, is sister to everything else. So when I first read this, I thought, whatever, this is molecules gone wild. They just wanted to cook up this result to, make it interesting, and I don't care. But when I started to actually get specimens in hand, I started, to take interest. Subsequent molecular analyses keep showing the same signal. This is a Keto Kawahara's paper from a few years ago, for example. So they talked about the morphology of this in the Ruggiero paper. This is written by Don Davis. The problem is that they described most of the morphology really nicely, but they didn't really put it in a phylogenetic context. They didn't polarize their characters. They didn't map them on the tree. So there's good stuff in here, and there's good stuff in Don Davis's other papers, but it was kind of lost in a wash. So I'm going to try to talk about this and make it better, what they should have put in that paper. First of all, Mysiids have microtrichia all over the forewings, on all surfaces. These are fine little hairs interspersed with the scales. And whereas they're restricted in distribution of the wings in higher Lepidoptera, including the Tiniidae. These can be observed only under microscopes. For that reason, actually, this is an old character. The old people knew about this, Spuler and company, back over a century ago. And then for that reason, they placed Eudarcia and Mysia in the Incroviridae, which is a Monotypic group. Okay, this is before they knew about the genitalia differences. Another thing is that they have this fragmented sternum 2. If you want to look at sternum 2 is really important for Lepidoptera. I suspect that this Monotypians at this grade, just outside of the Ditrysia, have a couple of sclerites right here, which are homologous to the detached anterior margin of the second sclera, or the second abdominal sclerite. These have muscle attachments. The true first sternum not have muscle attachments. So it seems that Eudarcia has these same kinds of, and it's at least one thing in same position, possibly. What I'd like to see is do these have muscle attachments? This is beyond my ability. I'd like to see some properly fixed material under SEMs. Third one, a minor one, these apodemes here do not extend into the body cavity, whereas they do so in higher Ditrysia. I'm going to mention just a couple apomorphies of Ditrysia that should be investigated. This is beyond my ability to see, but there's a couple of whether or not the tergopleural apodeme is fused to the subtegular or not. If it's not fused, that's argues that there would be plesiomorphy. Somebody should section the tongues of these things still to see if this muscle, lateral muscles are present or not. These are things that should be investigated because they're apomorphies of Ditrysia. So the final point I'm going to make is that a bit of speculation about how did this Ditrysian conformation even evolve? What's its adaptive function? Now, I haven't really done any thorough literature search for this. I'm sure that somebody's asked this question before. What's the adaptive advantage of having two orifices? I haven't read enough about this. But I am making an interesting observation. First of all, let's back up and talk about the apomorphies of Ditrysia, of Tinioidea, traditionally, if it were monophyletic. It's basically three things: feeding on lichens or other fungus, short tongue, reduced proboscis, and a long and elongate ovoscapt, telescoping scap. The ovipositor comes out telescope. So if you think about it logically, if the Tinioidea is paraphyletic with respect to Ditrysia, then those apomorphies of Tinioidea now become apomorphies of all Ditrysia. They have to be added to the suite of apomorphies of Ditrysia. See? So what do we have then? We have two orifices, reduction of microtrichids, things like this, and these things right here. Now, I can't explain the reduced tongue, and feeding on lichens, that's interesting, sure. But if you think about it, it should be really obvious. Having an elongate ovoscapt like this, it'll be just really difficult to mate. I'll try to copulate here. So you would have, so it's to the advantage, I guess, to be able to copulate through here, and then lay it, if you want to lay it out here. And an elongate ovipositor evolves only, is not present among any monotrysians, anything below this grade. We only see elongate ovipositors in Ditrysia, but we also see short ones too. It's free to evolve in this. So my point about this is that we have to conceptually think of having an elongate ovoscapt as part and parcel with the Ditrysian two orifice complex. It evolved all at the same time, for the same, for an obvious reason. Doug Dale 1974 pointed out that it would be the ovipore which moved posterior rather than the copulatory orifice moved forward. So these things just evolved to stretchy ovipositor, probably to do what modern tinioids do, is to lay eggs in little tiny crevices. Okay, they hide them. So this is all speculation. Okay, why were they doing this? There was probably a grade of 15 million years, a period of time when they were doing this. You know, did some predator evolve at that time that was suddenly preying on, you know, depredating Lepidoptera eggs? This would be great ground for speculation. So that's all I have to say. I just want to thank a lot of people. Don Davis was a late expert on tinioids and discovered many of these characters, and he passed away a few months ago. Including some of these things in manuscript, but yeah, I'd like to owe a lot to him. Many people in this room and other people helped collect specimens, you know, light traps. Alexey Reshchikov for collecting advice in Eastern Europe. Jadranka Rhoda and Robert Hoare are actually working on a phylogeny of Tinioidea that includes some of this stuff. So there's going to be more to come from this. People are interested. Megan Neal from the talk. Thank you.
We can have time for one quick question.
How do I recognize the eggs on lichen if I'm looking for you? For real.
These are, thanks. Well, these are so small. I would suggest looking for the cases of larvae. The eggs will be super tiny, but the cases are, there's a lot of photographs from Europe. To be sure, I haven't found any cases of any of these things in the New World. Don Davis had found collected cases of other species up north. But we can give you photos of what the cases should look like.
Awesome. Thank you, James.
Thanks.
Okay. Our next speaker is Monique Raymond, phylogenetic implication of the Metazoma in Platygastroidea.
Just the. Okay, right, left. Okay. Great. Well, thank you. And thank you everyone for coming here to hear more about genitalia, right? Our favorite topic, mine personally. So I am a new PhD student of the past month here at the University of Florida. But this project, which is still in the early stages, got started with Ishban Miko at the University of New Hampshire, where I completed my master's. Yeah, so we're focusing on the Metasoma specifically. So Platygastroidea, these are microscopic endoparasitoid Hymenoptera. You know them, you love them. They are diverse in form. There are 8 families, and they are parasitoids of about 8 different orders of hosts. So they're important for biological control applications. And believe it or not, these images are not stretched in any way. They are elongated and compressed in this way. One of the kind of ways that you know you have a platygastroid when you're looking from far away is that they have this small to tiny size. They can have intricate microsculpture or this shiny smoothness, and they tend to be kind of compact. You'll see very reduced wing venation, sometimes aptery, and reduced flagellum. Okay, so advances in visualization techniques, genetic and genomic technology, and just the pure persistence of taxonomists and systematists have brought us a very long way with the superfamily Platygastroidea. And more recently, molecular work and a soon-to-be published UCE phylogeny have produced this critical backbone of insight into the evolutionary history of this group. And we're very fortunate that we have a superfamily that's undisputedly monophyletic. The topology of the lineages and some of their dating has been established. So we've got something really wonderful to work with today. And something that I'm kind of picking up on in integrative taxonomy, DNA has had its moment, right? It's very impressive, fancy. And now there's kind of this movement back to looking at morphology. And with Platygastroidea, there's still a lot to be wanted in terms of understanding the synapomorphies of the groups. Okay, so our mission, we want to identify, using that backbone of information about the superfamily, we want to identify the phenotypic evolutionary successes. So we want to assess patterns in diversity, in quality, in synergy of characters and character systems. And yeah, in order to do this, we want to look at more of a system outlook instead of individual character states, because a lot of the characters that were identified in the past and used in cladistics, they were just kind of chosen for convenience, and they aren't necessarily the most informative about how systems have changed over time. Okay, so the cool new culture of morphology today. So we have obviously classical character mapping methods, we have newer stochastic methods, we have ancestral state reconstruction, and now we have the matrix. So the Paramo pipeline and the Ontophilo applications are a way that you can take individual characters and kind of put them in a bigger context in terms of like body section, body system, and look at changes in the phenome overall. And this is definitely ontologies, right? This is definitely one of those things that some of us want to avoid. But this might be the way of the future of looking at morphological evolution. So how are we going to do this, right? How are we going to approach an entire superfamily and all of its different morphological forms? Well, fortunately, we have a good foundation to work with. There are some systems that have been described in good detail. Unfortunately, the metasoma has been somewhat neglected. And the metasoma in Platygastroidea is kind of significance because they are parasitoid wasps, but it's also really interesting in the metasoma of platygastroids because they have this interesting simplified version of what you would see in many other groups of parasitoid wasps in the male genitalia, in the ovipositor, even something as simple as the abdominal spiracles. They're often reduced or absent. It's really bizarre and elegant. So we are going to start by looking in the internal part of the metasoma, and we're going to build on what's known about the skeleton and kind of try to make some inferences about the muscles there. So for background, there are 2 ovipositor system states that are convergently presented across the superfamily. So the Syrphidae type, that's the plesiomorphic type, and it involves antagonistic muscles. And sometimes it's actually very long and coiled inside of the body. Both of these system states are fully internal. The second, the derived type, the celio type, doesn't have muscles. Instead, it relies exclusively on hydrostatic pressure to extend the ovipositor in this really long telescoping tube with kind of membranous sections that are everted. And the celio type is not the only one that relies on hydrostatic pressure. The Syrphidae type is believed to partially use this as well. And the building of the pressure, if you're trying to kind of imagine it, platygastroids, they tend to have a posture to the body where the head is kind of lowered down. And they're believed to do a kind of like head movement that allows them to kind of pump up their body. And then the ovipositor extends from that pressure all the way back in the metasoma. So the first thing we wanted to do that no one has done before is we wanted to identify the muscles that are involved in that Syrphidae type ovipositor and homologize them, right? Because we're trying to put this into context. That's what we have to do. So for activation 1, there are two primary things going on here. Activation 1, the rotation, protraction, and retraction of the ovipositor. We know that in parasitoids, this is done by muscles. We'll abbreviate them M1 through M4 because morphologists are always using these big tongue twister terms. I'm not going to bother with that. We'll just call them M1 through M4 there. And up until recently, only one retractor muscle had ever been identified in these systems. And it was just known as a retractor muscle. That was it. That's what we had to work with. So we were able to homologize M1 through M4 in platygastroids. We had a look at 5 of the major basal lineages out of the 8 families in the superfamily. We found M1, 2, 3, 4. So this is a great first step. What is kind of interesting, though, is that for activation 2, we have not yet approached that. So we haven't identified which muscles would be involved in the opening and closing of the metasoma and how they may have been accepted. Something to look forward to. Another system complex that had been identified in previous literature, just kind of skimming on the surface, was this second gonapophysis assembly highlighted over there on the right. And so by using homologized muscles, we were able to determine that this assembly is part of the second valvula muscle on the terebra, the process muscularis and articularis. And this is involved in kind of a little bit of the extension and protrusion of the ovipositor. So one thing that's really interesting about this, though, right? It's just this gonapophysis assembly. What's the big deal? Well, in other parasitoids, like this one down here on the bottom, this is what facilitates that jackknife-like movement of extending the ovipositor and moving out of the body. And the location of this assembly in platygastroids is much lower down. And so biomechanically, it just doesn't really make sense to have that same function. And we don't know what it would be used for in this case. It could be a locking mechanism. It could be involved in like alternating lateral movements. But this is just another one of those big mysteries that we have shined a little light on so far. Okay, so into the male genitalia. One thing that is kind of interesting going from looking at females to looking at males is that the males and females both have those reduced abdominal spiracles and sometimes absent abdominal spiracles. And we don't know why. It might be an inconsequential failure to decouple the adaptive trait, but we simply don't know. What we do know about the males, it's essentially exclusively skeletal. There's been characterization in the works by Snodgrass, Ozer, Johnson, for the Celionidae family. Their male genitalia just skeletal in nature. And that is actually one of the most derived groups, and it's one of the more simplified forms. And so we haven't been able to find anything in the literature that indicates intermediate states. And so we don't know how the simplification has happened and what the parts are homologous to. So over here on the right, those are the Celionids. You can see it's this really continuous linear edigo-voseller shaft composed of something. don't know entirely what. Okay, so another beautiful array of male genitalia here. So what we were able to determine so far, there is an independent edicus, there is protractor muscles present. We also found parameres, and some of these characteristics are things that they have in common with other Proctotubomorpha, so Ctenopoidea and Chalcidoidea. And by having this kind of landscape mapped out in a little bit more detail, we're able to look at the more derived cellionity and identify the edicus and the volzella kind of fused in this edigo-volzellar shaft on the dorsal and the ventral part of the male genitalia. So we weren't able to figure out, at least not yet, what's going on with the pressure and the spiracles in the males. What is kind of interesting is that there are some groups that have very elongate male genitalia, as seen here, with this sort of hinge, this resilin-rich biaxial joint. And those tend to be in ones that have closer to functioning spiracles or less reduced spiracles. It's kind of interesting. Along the way, some of the extra fun stuff that we found, an unofficial synapomorphy for the superfamily is the presence of these anterior T2 pits, long believed to be associated with glands or a point of muscle attachment. We were able to kind of cancel out those possibilities using a lot of confocal laser scanning microscopes of various taxa. And instead, our theory is that these are sites of biomechanical articulation. And there's obviously exceptions to every rule that you try to make. And Cenopeus, so this is a platygastrid. And one of the things people know Cenopeus for is it has this fusion of the two anterior-most metasomal segments. So it's very fixed in that part of the body. And you would imagine that there's a reduced level of maneuverability with oviposition because of that. Cenopeus is also known for having very elongate genitalia and very elongate ovipositors, sometimes that long coiled ovipositor inside of the body. So the lack of articulation here at the anterior part of the metasoma, that reduced maneuverability, is met with this other adaptation. Hypothetically, this is one of my conspiracy theories. We'll figure it out. So along the way, lots of fun. We were able to identify the presence of some canal cells possibly associated with class 3 glands. These are paired in the metasoma. Class 1 canal pores as well over here with this really lovely GIF. I look at that. And something new that we're just digging into, paired glands that are at the attachment point of these like fan-shaped muscles, intersegmental muscles. Not in all groups, but I mean, look at these SCMs. Those are really, really lovely. And an exception to this trend is in Nixonia, which is kind of an odd one. They do look like they might have paired glands, but it's in the opposite end of the sclerae. And they have kind of a linear pattern of musculature instead of these fan-shaped paired. So in conclusion, to be continued, this is just the beginning, but I think that some of the best fun in doing taxonomy is just like looking at things and exploring and asking questions. So I encourage you to do the same. Just get the microscope, dissect something, take a look, throw a laser on it. You know, if you've got the lasers, use them, right? So we've got a few findings so far, but there is more to be determined. So thank you. I don't know if I have time. Yes.
Yes, we have time for questions.
Oh.
Great talk. I was wondering, so you mentioned, you know, a hypothetical association between this fixed T1, T2 junction and the elongated ovipositor. Isn't it in a stemma? Because that also has a really long ovipositor.
Oh, that's cool.
Yes, that is a good question. So inostema is the one with the sheath, like the horn-like sheath, dorsal sheath, and that houses the ovipositor. And, you know, I will say I have dissected that. I have looked at it and it was unremarkable, but I think that will require a little bit more additional care. Yeah.
Yes. The medicine by gastroids is to have rows of toby along the interior margin of each tergite. These have Lebanese amber and things like Syrphidae. But then secondarily, I think some of them have become hits or become associated with glandular speciation. Some of them you can actually see spores that are associated with CD hits.
Yes, yeah, I think you're referring to like in Nixonia. Yeah, so in Nixonia, the like the alternate presentation, they have pits. They have these pits in the at the margin of the sclerite, but there's a bunch of interesting oppressed hairs inside, and they're kind of bent at a 90-degree angle. And so naturally, it would lead someone to believe that that is associated with glands, but to be determined, to be continued. Yeah.
Okay, thank you.
Yeah.
Okay, our next speaker is Davide Dal Pos with the talk Host-Driven Parasitoid Syndrome in Ichneumonidae.
All right, well, you know me by now. I'm going to explain a few things before starting and showing the results. Essentially, we're going to talk about, it's very wordy title, but essentially I'm going to explain what a syndrome is and then what we intend for convergent evolution. I want to also mention that I presented this talk slightly different at the ESA, and it went really bad. Out of 10, I got 5, and one of the judges told me that my volume of the voice was very high. So either you're looking at a dumb person or a very loud one. So prepare yourself, please. So what is the background? What is convergent evolution? We heard talking extensively in papers. What is convergent evolution? We went with the Lozos one, which is essentially the independent evolution of similar features in different independent evolutionary lineages. And why it is important? Because according to Staden, it is substantially ubiquitous in the life history, especially of insects. And so if we think about what can be a convergent evolution trait, you have a phylogeny, the black lineages are where that character appears. If it is independent and substantially appearing multiple times, that is sort of, we can consider it a sort of convergent evolutionary pattern, right? And so if one example could be a subterranean fauna when you have all this beetles and different arthropods that are sort of pale in coloration. But what is a trait syndrome? So if you take a look at different papers, they talk about trait syndrome, they talk about syndromes. They take it at multiple angles, but what exactly are they? What is the definition of a syndrome? So you take an example, the first one that I found was mentioning syndromes, is more like on the genetic side, so on a sort of a bad conception of syndromes. Can be a sort of an ecological syndrome in invertebrates, could be also a more like a behavioral syndrome, like in the army ants, or also sometimes they don't even mention the definition of a syndrome, but they simply use it as a sort of ecological pattern and evolutionary. So I was left like this. So what am I dealing with? What is a syndrome? And luckily, this paper coming out recently in 2021 actually sort of explain it, but I think we need to get a step further. And I think that the work that I'm showing you will push it a little bit further. So you need to have 4 main features to be a syndrome, to have a trait syndrome. One, you need to have convergent evolution, a dependent evolution of multiple characters. Then you have to have multiple traits that evolve convergency. This trait needs to be concurrent, so they need to appear all together, almost all together in the same lineages multiple times. And of course, they sort of underpinning, the idea is that they're underpinning the same environmental pressure. So the same environment, let's put it that way. So if we take a look at what it means is you got this 2 phylogeny, this 2 pattern of traits. This is the same phylogeny, all right? But the characters, you are different. You got a X and a Y character. So they appear in the same phylogeny, roughly at the same stages. Here, it's roughly different, but we have character X appearing here, character Y appearing almost in the same lineages. So we can assume that in this case, we possibly have a trait syndrome. But you cannot stop right there. You need to actually assess if those characters are actually belonging to a trait syndrome. Okay, so why Ichneumonidae? Why do we study in Ichneumonidae? Well, because that's what I'm doing mostly, the majority of my time, but 42 subfamilies, 25,000 species, a huge diversity. They are hyper diverse, also in life history strategies. They are parasitoid, so high trophic level, mostly important. And they have a very notable rate of convergency, which makes it possibly good for the study of convergent evolution. And of course, attacking different substrate, we have an opportunity to actually assess according to a same environmental pressure. And one of these substrate is the wood-boring environment, which is very easy to study since we know which parasitoids are actually attacking the wood-boring environment, and it's extremely neat because it's isolated in a way. And this is the wood-boring pattern cycle. So you take your wasp that needs to drill into the woods to reach the host and lay the eggs. So if we take the pattern, so we got the wasp looking for the host within the wood, it needs to oviposit, so it needs to drill down to find the host. So got a huge amount of substrate to go through. And then you get into the wood substantially, you lay one egg, the egg develops, the larvae of the wasp eats the larvae of the host, the larva develops, pupate, and then of course comes out of the wood again. So you get this very neat pattern, and I want to thank Devon Henderson for the drawings, and I was just simply able to put together the cycle. Okay, so you got this very neat pattern, this very easy to study, relatively easy to study environment. So what do I want to do? So first of all, I want to understand if there are characters that according to the substrate are correlated, okay? Are those characters correlated? Perfect. Then are these characters correlated with themselves? So they are in a sort of pattern, they are in a trait syndrome, and then are this connection actually holding up when we think about a syndrome, or we got other underlying patterns behind it? How we did it? I'm gonna go relatively quickly here. So we needed to build a phylogeny. There was a phylogeny, but it was not usable for us. So we took a phylogeny, we combined it with some other data. We created a chimeric tree. We used morphological characters from Bennett et al. That we could use and we can test. The matrix was 141 characters. After this, we had our phylogeny. We chose the characters from those 141. How we chose it, we went back to the wonderful literature by Wilhelmsen, mostly Thurisi, that actually developed this, and they had some sort of hypothesis about the wood boring. And then we test for correlation, and we simply use Bayes traits, and we did substrate against character versus character, and we came across some sort of evaluation of those. How did we did the evaluation of the syndromes with the ancestral state reconstruction that enabled us to understand where those characters actually appear in the phylogeny? Okay, so here are our results, and they're pretty big results in a way. So what you see here, this is our phylogeny. The blue stars, the empty stars are where the wood boring is not present in Ichneumonidae. The full blue star is where we have the appearance in Ichneumonidae of the wood boring pattern. And you can see it's only at the base of the tree. This is the same tree. And so we have roughly 7 independent origin of wood boring within Ichneumonidae. At least that's what we thought. With this 7 independent origin, how many characters are actually correlated with wood boring? We found 8 correlated characters. And there are these one here. Do not worry about the moderate bias, log bias factor. It's not important for the moment. So we found 8 of those. We took all of those and we plot them into a phylogeny, and you can see the coloration. Do not worry about reading it. For the moment, just follow me, right? So you can see that wherever it appears, the wood boring, we have some characters, sometimes all of the characters, sometimes just some of the characters. In some other types, we have characters that are not associated with wood boring Ichneumonidae. So you need a discussion about these characters. You cannot take them for granted like this. So we assess that only 6 of these 8 characters actually belong to a syndrome. And we excluded the modified cleaver margin because the matrix was not clear in the resolution of that character. There are possible multiple characters into that. And the ovipositor guides because only appear in one group, in one lineage, which are the resign. So those cannot be assessed comparatively. So today I'm going to talk to you about these two with the asterisk and the ovipositor guides, because why there is a bigger story I'm going to tell you in a second. So let's talk about the included character. The first valvula with teeth. The first valvula is located right here, and it is the lower valve, and those are the teeth in the lower valves. For many years, we thought this is correlated, of course, with wood boring. So we tested that. What you're going to see is that the violet pattern is where the teeth are present in the valvula, and so you can see where it independently appeared, okay? What pattern did we see here? So let's zoom in one of the group. Here is where appear the teeth on the first valvula, and here where the wood boring appears. So it is almost consistently that the teeth appears before the host shift, before getting into the woods. So could this be a precursor to get into the wood? Could this actually change the landscape of the morphology of Ichneumonidae to enable the entering into the wood? It's also, however, appeared also outside the wood boring taxa, and this is possibly because it has also a broader functionality, and also here we need to resolve what it means to have teeth on the second valvula. Like in this case, we have a polysphinctine group, which is a pimpline. They don't go into wood, they attack actually silk and spiders, but they use the teeth to actually clinch onto the host and avoid it escaping while ovipositing. So what it means to be having teeth, it's a long discussion. Another included character is the elongated terebra, which simply means the length of the terebra, that in case of the Ichneumonidae is extremely long, 10 times the body size, right? Where does it appear? Same reasoning. It appears in all this pattern within the wood boring taxa, but also outside the wood boring taxa, and appear consistently with wood boring. So if you take this, whenever there is a wood boring, you got a long terebra. Pretty clear here that you need to have a long terebra to reach your host underneath the wood. That's not something that you cannot have, but it's also present outside the wood boring, and this is surely present into a syndrome. But why non-wood boring taxa also have a long terebra? Great discussion also here, nobody knows, but it's possibly used for better maneuverability whenever you use it for ovipositing into non-lignified wood, for lignified plant tissue. Okay, so that could be a possibility. We still don't know. There is a need for refinement of the character as well. Okay, now let me talk lastly about the ovipositor guides, which is the character that one of the two characters that we excluded. I want to see if I'm really dumb or not, so follow me. What is the ovipositor guide? They're located underneath the metasoma, underneath the abdomen there. They're those things that you see, those clenching thing, and they're used to make sure that the ovipositor during oviposition goes through that for bracing, for enhancing maneuverability. So to allow this sort of pattern here, okay? Because otherwise you have a long ovipositor, they're not able to keep it together, it's gonna break, okay? So you take it, same reasoning, and it only appears here. So this is why we excluded it. We don't have a comparison. However, if you take the labenine, which is another wood boring, part of it is wood boring, actually have a similar structure, but in another location. So the location is the hindcoccyx, and they do exactly the same thing. So they actually do the same thing, the clinching, the buckling of the ovipositor. So sort of similar structure, but in two different body parts with the same function. Can we therefore increase our knowledge or our interpretation of what a syndrome is? Can we talk about functional syndrome and not just morphological syndrome? Can we use this to say, well, these actually are two same characters, but they simply take a different path to do the same thing? Could this be a syndrome? I would say so, otherwise I wouldn't have put it here. Conclusions. There is a wood-boring syndrome. We're pretty sure about it. There could be other substrate and therefore other syndromes. Difficult to study right now. Sometimes we don't have host association. The evolution of multiple traits open up the idea that it's not just a single trait evolving, but possibly you got one character that evolves and also change other multiple characters. Possibly a genomic background there is behind it, possibly also a linkage, we don't know. And of course, you need to have an evaluation of the traits beforehand, doing your metrics, and after that. And of course, we try to expand the concept of syndrome, but nobody's listening. We published this. If you're more interested in our reasoning, you got everything. We got also a review of the entire characters in the history of Hymenoptera that are associated with wood boring, with the associated function, location, and references. So if you're interested, please do that. Also, I want to mention one last thing. We published this in PLOS ONE, and they decided to publish it like that. So nobody can read the phylogeny, which was the heart of the paper. So if you need, please let me know. But I want to mention this is fantastic because you pay a lot of money and this is the result. I'm not really sure. All right. Yes, I want to thank two people that enabled me to become a better morphologist. This is Ishvan Mikko and Lars Wilhelmsen. Both of the two believed in me when nobody actually thought I could become a morphologist, and I'm able to call them friends right now. And there is a genitalia of an Ichneumonidae. Has no meaning on the presentation whatsoever, but they have a genitalia, so they should be there.
We have time for one quick question.
Yes.
Says independent traits that you're evaluating for a syndrome.
Yeah, it's an indirect, right? So you need to have the phylogeny, and you via coding via the matrices, you're able then to create an ancestral state reconstruction that enables you to understand what node actually appears, and those are done like that with that sort of assessment. It's not super clear from being this fast, but we actually did that, so we know what lineages are actually appearing and when in the node, what node.
Are there metrics that you can apply to say that these characters, some index of independence between your characters?
I think there are, yes. We should have tried to do that, yes, as well. That's also a next step, yes.
Thank you. Okay, this is the end of the first session of the afternoon. We will try to be back in 5-10 minutes. We are a little bit behind. So I'll see you, yes, in 10 minutes, let's say. Okay. Presentation is Get the Most Out of Your Museum, Mapping the Distribution of Jewel Beetles in Idaho. Please, Luc.
Okay, you can see the talk?
Yes.
Okay, well, I'm at the University of Idaho Bar Museum, and it's a bit cold here. It was -24 Celsius Wednesday morning. So I'm looking forward to come to Gainesville next week. I'm arriving next Saturday to teach a course on fruit fly identification. So you'll see me very soon. So the Barr Museum was founded by William Barr when he joined the University of Idaho in 1947 out of UC Berkeley. And actually, the entomological collection was initiated 130 years ago by John Aldrich, a dipterist. It didn't grow, it grew into about 30,000 specimens, and Bill Barr joined the museum, and he's the one, the university, he's the one who really made the museum grow. And after his retirement, Frank Marickel, who Bill mentored and trained, was the museum manager, and I took over from Frank when he retired 10 years ago. So this museum grew from a mere 20,000 specimens to over 1 million pin specimens in 143 California Academy of Science cabinets. So it's a respectable collection for a small university. I wish the budget I get was proportional to the size of the collection, but it's not. I work on minimal shoestring budget, and I teach entomology courses to help preserve my position. And also, we do a lot of outreach I'll be talking about. So one of the highlights from the museum is a large collection of buprestid beetles, because Bill Barr in his time was one of the leading experts on buprestid taxonomies and clerid beetles as well. So we have over 1,300 species collected by Bill Barr, Frank Beer, who was based in Nevada, and Rick Westcott, who's based in Oregon, and he's still around, still very active, and by Malcolm Furness, who lives here in Moscow, Idaho. I'll tell you more about Mal Furness in a few minutes. So I don't have graduate students, I don't have technicians, so I'm pretty much the person in charge of the museum, but I do have one at a time, young, talented, young undergrad students working a few hours a week. And I've had one museum volunteer in the past, Bill Turner, who was a dipterist from 2015 until he passed away 3 years ago. He came every week. He was the museum volunteer. So these undergrad students, they've done a lot of assistance in curation, but I took their work to the next level too by producing some nice material for the museum, nice outreach material, because what makes this museum survive is not the research, it's the outreach. So we host school groups regularly, and they handle our tarantulas. They get their souvenir buttons if they hold the tarantula. And with the Entomology Student Club, we have some fundraising activities such as laminating into earrings butterflies from these thousands of papered common local species we have in the museum that are not worth mounting. So we produce earrings. So we have an annual Haunted Museum event as well. And here is Dr. Mad Bug. And that's my alter ego here. I put this one on during outreach events to entertain visitors. So it's a tropical fruit fly. It was made by my former student, Ellie Hitchings, who's a very talented artist. So anyways, yeah, we do specimen digitizations, and we have like over 57,000 records online and thousands more that I will upload once publications are come out that make use of that material. And from digitizations, byproduct was the production of the first outreach poster of the museum on the butterflies of Idaho. And the publication here with Bill Turner, right before he passed away, with Anthony Thomas, the lead author, we did the tabanids of the Pacific Northwest, and all the data is digitized. And last year, the tabanids of Florida came out, authored by Tony as well, and I helped him in the process. So the first poster, so when Emma Ekens joined in 2021 as the museum assistant, I got her into special projects. The butterfly poster was so popular, and you can download your copies of the posters on the museum website. I got her to take photographs of every one of the species of buprestids found in Idaho. We did not use expensive fencing equipment, just the Olympus TG6 camera that has an integrated focus stacking function and 2 cheap LED panels. You can buy them for $25, $20. And then she took photos, and with Photoshop, she was able to remove the pins and enhance the photos. And we came up with the Jewel of Beetles of Idaho poster, 104 species. Number 3 poster that just came out last month, the stink bugs of Idaho. This one is part of a publication on the stink bugs of the Pacific Northwest. I'll give you a talk next year on that if you want. So that's a project in progress. But anyways, the jewel beetles, over 7,500 museum specimens that were identified by Bill Barr, Rick Westcott, Frank Beer, and various other buprestologists. And therefore, the identifications verified and by the experts. And I included some human observations from iNaturalists, the ones that are research grades and that are credible. And in total, almost 3,000 occurrence records. And occurrence records is one or a series of specimens collected in one locality on one day. And 989 localities. So using quantum GIS, I'm very low tech. I use freeware because buying some expensive software would be more than the annual budget of the museum. I was able to produce distribution maps. Here are all the localities. And the first buprestid published record was in 1876. Over the years, it came up to 104 species. So the 7 most commonly collected species are here with more than half of the occurrences. And some of them, like Taxa inornata and Phenops drummondii, they're common like dirt. Others are more commonly collected because they're jewel beetles, so they'll attract more attention. So we have large series of these buprestis because they're cute. So, and Emma's work with the photos really greatly enhanced it. So we have a manuscript that's under review, and it will be the annotated checklist of the buprestids of Idaho. So here's one example where the first Idaho record and number of localities, occurrences, and specimens, the counties where they're present, and the larval hosts, only the host plants that are found in Idaho, so that to indicate what they're known to feed on in the state of Idaho. Then there's a photo and a map with distribution. So these are localities, and each county with the locality present is shaded, and some seasonal graphs based on the occurrences. So there's another example. So you'll notice that Oregon and Washington and Nevada are in yellow. That's the states where that species is known to be present, not known in BC or Montana or Wyoming. And here's a common species, and that's common in all the surrounding states. So you have a lot of distribution information summarized visually in these maps. Here, the type locality, this species was from Spalding in Nez Perce County in Idaho. So there's a type locality, and there are some human observations records to help add some records. So it's in review, and my wish is to see it published in Insecta Mundi. So I'm following all the requirements, and the two reviewers are Rick Westcott and Josh Basham. They're both very, very knowledgeable Buprested taxonomist, and Rick Westcott was a graduate student of Bill Barr back in the 1960s, and Malcolm Furness was also a graduate student of Bill Barr in the 1960s. So Rick just sent to me yesterday his manuscript review. Oh, the most thorough, meticulous review I've ever seen. Thank you, Rick. That will make sure that all these records are really good, and Josh will send his review pretty soon, so it will come your way pretty soon once I'm done with the edits. So Malcolm Furness, who was also a graduate student of Bill Barr, I'm going to see him tomorrow. He lives here in Moscow, Idaho. Rick and Mal, they're like these, and Bill Turner was too. They're these amazing people who I look up to and I spend time with as much as I can. And they're really amazing. So Mal is a retired forestry entomologist, and he's one of the ones who has published more after retirement than during his career. And his expertise is ecology of bark beetles, of scolytines. So yesterday, he sent me, and he called me and he said, I have a new manuscript and I'd like you to have a look at it. And I'm going to, and tomorrow I'm going to go spend time at his house with him. And it's all SEM photos that he took of the prescutum and scutellum of bark beetles. And it's a very elaborate manuscript. I never thought he would come up. His last publication was last September. I never thought he would come up with another one manuscript. Guess how old he is? Any guess?
100.
Yeah, he's 98 years old.
Oh.
Still very active. I wouldn't be surprised if he comes up with another publication after this one. So that's it for my talk. I'm exactly 15 minutes, so I did well. So you have questions or comments or criticism or?
There's time for one quick question, very quick question. Okay, no one for now. Luc.
Okay, thank you. I'll see you next week.
Thank you. Our next speaker is Jessica Watt, and her talk is Platygaster and the Olive Grave.
Well, hi, everybody. So today I have a talk. I want to walk you through our epic quest to discover the holy grail of Platygastroidea. So this is Platygaster. It is the type genus for its superfamily, Platygastroidea, which Monique gave us a really nice introduction to. And it is a big genus. It has about 700 described species, which is approximately 10% of the superfamily. It has 15 plus junior synonyms, and it has basically no synapomorphies. So it's always found at the end of keys. Does it not have any cool characters at all? It must be a Platygaster. Platygaster has a lot of problems, and one of those is that it seemed like it's not monophyletic. So this is some preliminary ultra-conserved element data from Miles Zhang, Bonnie Blamer et al. And you can see there's sort of this main Platygaster clade there in the middle, but it's very messy. And in order to figure out what is the real Platygaster, we need to go back to the type. And this brings me to the holy grail in our story. This is Celio ruficornis, which was described by the great French zoologist Pierre-André Latreille in 1805. And this is the type species of the genus Platygaster. And he described it with, fairly limited, it's a short description. It's not very descriptive. And the only collection data is near Paris on plants. And, Latreille lived and died in Paris. He worked at the museum there, and it would make sense that this specimen should be in the museum in Paris. However, it is not. Many of us have visited on different occasions and checked. It is definitely not in Paris. In fact, we couldn't find any of Latreille's Hymenoptera in Paris. So where could it be? To answer that question, we have to go back in time to 1830, when an entomologist named John Obadiah Westwood, who probably some of you are familiar with, he ended up being the first curator of entomology at the Oxford Museum. He actually went to Paris in 1830. He went to Paris a lot to study and take notes. This was the only time that we know of that he looked at any Platygastroidea there. And he took notes, he studied it, he illustrated it. Up at the top, there is a picture of the specimen itself. And he even drew a picture of Latreille, who was still alive at the time. These are not published anywhere. I took these pictures with my phone while visiting the Oxford archives. They have a whole collection of Westwood stuff. They even have his shoes for some reason, which are really fun to look at. They have buckles on them. Anyway, so this was the first and last recorded examination of this specimen. 3 years later, he published a paper in which he described a new genus, Epimeces. And in this paper, he quite politely criticized some of his contemporaries, Halliday and Curtis. And he said, so he basically said, they have not looked at the type. What they said is wrong, and I have seen the type. And this is not the same genus as Platygaster. A couple years later, Francis Walker, who many of you may have been personally victimized by, synonymized Epimeces with Platygaster. And he also drew Platygaster ruficornis, which that's his interpretation. We have no evidence that he saw the type. He may have, but he never wrote it down anywhere. He also synonymized it, or he considered it a senior synonym of Platygaster grandis. Remember the name grandis because it is going to be important in a few minutes. And he criticized Westwood a little bit less politely this time and basically said he's wrong about Epimeces and he's wrong about Platygaster. Okay, there's grandis. Then about 20 years go by with no significant mention of ruficornis until 1856, when Arnold Förster, one of my personal heroes, mentions the species as part of a new genus, Isocybus. Now, this is really bad, right? We would never take the type species of another genus and put it in a new genus. But in Foerster's defense, the ICZN didn't even assemble until 1895. So we can't really blame him for not following rules that did not exist yet. Now, Isocybus had no type species, right? There were no rules. And so Foerster was not in the habit of assigning type species for his genera. The type ended up being designated later by Ashmead, who mentioned Platygaster grandis. Remember Platygaster grandis? So where's the type of that? Is also yeah, Mace's collection was destroyed in World War II, possibly even earlier. The legend is that after he died, his collection was taken over, his office was taken over by a geologist who threw out all of his insects and put rocks in the doors. I don't know if that's true, but the museum was definitely bombed in the 20th century. So they're gone. But anyway, so the type was missing and it is probably destroyed. But there is hope. In the Natural History Museum in Vienna, there is the collection of Arnold Foerster. And in the collection of Arnold Foerster, there are a bunch of these specimens marked original exemplar. And an original exemplar is a specimen that is obtained from the author of that species. It doesn't have type status, but it can be really helpful if the type has been destroyed to know, okay, well, the author thought this was the same thing. And so we chose one of these original exemplars to be the neotype of Isocybus grandis. And you'll see it has these very characteristic characters. It's got this blocky head with coarse sculpture, and it has a setose depression on the mesoscutellum. So back to Platygaster ruficornis. Throughout the late 19th century, we'd often see, Isocybus ruficornis in various collections and literature, but really no one had given it any serious thought. And no one knew where the specimen was or really even paid any attention to it. So where is the type? The big break in the case actually came in the form of a footnote in Westwood's general entomology textbook that he wrote much, much later. I think he had maybe gotten tired of fighting with Walker in the literature, but he did kind of slip in his final argument in this textbook. And he writes, again, I have seen the type and I am right. I saw the collection. I saw it in the collection of Baron de Jean. Now we are getting somewhere. So those of you who are coleopterists in the audience are probably familiar with de Jean. He was a coleopterist and also a soldier. He was Napoleon's aide-de-camp, which I think is kind of a personal assistant. And he was known for collecting on the battlefield, even at Waterloo, which did not work out super well for his boss. Most importantly for us, the collection is traceable. This is a book that some of you may have heard of, Collectiones Entomologicae, that attempts to assemble all of the great insect collections of history and trace where they went. The section on Latreille is a dead end, it just says Paris, but there's a very good detail about what happened to de Jean's collection. And we learned that the Hymenoptera went to an Italian Hymenopterist, Spinola. All of the Hymenoptera, including Latreille's specimens, according to this book, went to the museum in Turin, which has been closed for many years due to budget. Luckily, we know a guy. Our colleague, Francesco Tortorici, actually works at the University of Turin and knows who has the key to the museum and was able to get it and get into the collection. And hey, we found it. We found the specimen. It's not in the best condition, it's missing its antennae and its metasoma, but the labels look legit. And there's enough there that you can identify what genus it is. It has a blocky head with coarse sculpture and a setose depression on the mesoscutellum. Oh no. Yeah, so this opens a big can of worms. According to the International Code of Zoological Nomenclature, this would make Isocybus a junior synonym of Platygaster. So we would transfer 32 species from Isocybus to Platygaster. What we've been calling Platygaster is actually Epimeces. However, Epimeces was preoccupied, so they would go into the replacement name Parapemeces. So 700 species, including biocontrol agents, agricultural stuff, all go to Parapemeces. And obviously, nobody wants this. This is a terrible result. The code does have a provision for reversal of precedence, and there are two criteria that must be met. Yeah, so first, the senior name has not been used since 1899, and the junior synonym has been used for a prevailing concept for the last 50 years or so. Only one of these is true. It's only true for Isocybus. So we have to petition the commission in order to ignore this, to prevent this massively unstable result. So this is our petition. It was just published in December. And what we want to do is essentially remove the type status of the specimen in Turin. It doesn't belong to the prevailing concept of platygaster, and it's not really available for research. What we are proposing is actually to make the type specimen of Epimyces ensifer into the designated type of Celioruficornis. This would ensure that the definition of platygaster matches the prevailing concept, and it will also destroy Parapimyces, which is good because it's an ugly name. So our petition is now open for public comment. If any of you have any strong opinions, I hope they are positive, but if you have negative opinions on why this shouldn't be done, you are also able to submit those to the commission. They will vote on it in no less than 8 months, so probably much longer than that. And in the meantime, we've been finishing up this UCE phylogeny, which will revise the whole family Platygastridae at the subfamily, tribe, and genus level. And yes, we do have a monophyletic platygaster at last. So in conclusion, the moral of the story is be careful what you wish for, and sometimes it is better to let sleeping dogs lie. I want to thank everyone who helped with this. Thanks very much to the organizers of the CSE for having me and putting on this lovely conference. And thanks very much to my funding agencies, especially the German Ministry of Research and Education, which very generously funded my PhD in travel. And thank you all very much for listening. I will take questions if I have any.
Yes, we have time for questions.
Monique.
Okay, how did you find the reference in the footnote of a textbook?
I spent a lot of time reading because when I moved to Germany, it was 2020 and I couldn't go anywhere. So I just spent like a lot of time learning German and reading old literature. That's how I found it. Yeah. I also got like a little bit obsessed with some of these guys and was just kind of curious about their lives and what they thought about things. Paul?
I'll talk to you later about Turin, but in my group, what ended up is because of the French Revolution, a lot of French lectors, the aristocrats, left the country and went to Italy.
Oh,
So that's like the
interesting.
first place to look if something's missing out of Paris.
Oh.
Good choice.
I want to talk to you about the name of that museum.
Yeah, I'll talk. Yeah.
Any other questions online?
No, nothing in the chat.
Okay, perfect. Thank you, Dave. Okay, our next speaker is David Serrano, and he's gonna talk about the search of Orazema minutissima in Florida.
Getting crashed. There we go. I guess Ashley didn't want to join.
All right, so I just thought John will take questions if I leave time for him.
Ashley is on a plane.
Okay.
Okay.
All right, we'll forgive her. All right, so we're going to continue the trend of little tiny wasps, which tends to be trend this year. Beetle people stepping up, right? So yeah, bit of a story we're gonna tell here, but first, to kind of take it back to where most of you know me from. So my collection is the Broward College Insect Collection. I do a lot of surveys. And so I come across a lot of materials. So some of you I played with and collaborated with. And then so this is where this story comes through, because we're endlessly collecting, finding new things, as we all should. All right, so first, with any epic story, there's got to be a villain and a monster to overcome, right? Sorry for all you ant people in the crowd. So Wasmannia, the little fire ant, it's been here for a while. So it's been in Florida since 1924. I could personally attest, hence my little bit of hatred for him as a Miami native. I've lost many battles with them, especially coming from a construction family, doing construction myself for a little while. They love to hang out and make nests like between bricks, like lumber, stuff when, you know, dad tells you to move stuff and you put it on your shoulder and then just burning feeling all over your shoulder and such. So you can imagine my hatred, which also lends itself to why it's been able to move around so much and get transported. It loves to put those nests in those kinds of situations. So here's the somewhat current map. I believe Andrea put this map out on one of the featured creatures of where you could find Wasmannia. So keep in mind of that map as I ask for your help later on. All right, and so why Wasmannia is problematic. It's a terrible, severe pest. It makes Hymenoptera pests worse, moving them around and such. The sting and irritation we've covered, but also hits poor little baby birds in their nests and attacking their eyes, and but also domesticated and native birds as well. And then it's just bad news for a lot of native invertebrates, especially where it's a big problem out in places like Hawaii and Guam and all those kinds of areas. All right, so worldwide, it's gotten around. So you see the green is where they're coming from. So that's their native range. And then you can see all the color coding there of the brownish red is where they're popping up. So you can see Florida and Texas and US and all throughout the Caribbean. We see these guys around. And so, yeah, they get around. All right, so two major clades they've figured out. So there's a sexual reproducing one and a clonal reproducing one. And hold on, because as you know me, I'm not necessarily the expert on this group. This is why John's waiting in the wings. Check my notes there. And so, yeah, it's in the Caribbean for the most part. But for these, once again, we have the tropical and the big invasive clades. So that's what you're seeing there. And so it utilizes, like I said, sexual reproduction and clonal. And the clonal we see in all the invasive populations. So there's a hint there of why they tend to get around. Which also, when we start looking at genetics, makes it a little tough because the CO1s are pretty much identical. So it's hard to suss that out, but gives some clues and looking around. So when we kind of look at their movement patterns, what we're going to focus on here is that orange up there. And so while looking at this, the data, the Harity lab over at UCR has sussed out is that we're pretty sure it moved from the Caribbean and Cuba and came into Florida in the 1920s, and then sometime between then and 1999, where they first discovered it in Hawaii, it probably moved. We're hypothesizing that they moved from Florida over to Hawaii, right? So yeah. All right, so our hero of the story, this is where the Harity Lab's interest is with Orosema, so one of his eucaridids. And so it's a parasitoid of both Wasmannia and Phylogenetic. So it hits both of those guys. And it's one of the only known native parasites of Wasmannia. So it's a good one. So we find this in the Caribbean, Central America, Northern South America. So he's been around. He did a big study in Dominica. And then in Hawaii, they're looking at stuff too. Right? And so now, so we have the Hawaii and Florida. Where did it originally come from? Once again, we see this parthenogenic clonal reproduction. A lot of mostly females with the occasional rare male. There's been discussions of using Orosema as a biocontrol agent. Hasn't really been implemented, and possibly because eucaridids have a really complicated life history, if you're familiar with those guys. All right, the other interesting thing we see Since we have two hosts, we also see three different size classes of Minutissima. So we see the ones hitting Wasmannia tend to be the small group, the small size, and then the medium and larger we see are mostly coming off Phydoles. All right, and so there you can see the vast difference. So if you're familiar with those ants, you can tell Wasmannia are really tiny, and then Phydoles are bigger. So hence we see this range there. And so the reason why we think we see the size difference on the two bigger ones is they're hitting different casts of the Phydoles. All right, and then so there's some statistical stuff actually to kind of pull those apart so you can see they're definitely distinct. All right, so for their life history, so if you don't know, so the eukaryotes come in, they're going to oviposit, they're going to put it into the cavity of the leaf surface area. For our focus, we're going to talk about ferns. So they deposit the egg in the plant tissue. There we go. There she is doing a hell of the work. And then which leaves these characteristic scars, which, there it is. And these characteristic scars, which hopefully you get an eye for. So once again, when I do the call for help later. And so then once they hatch, they come out, the ants will pick them up into the little buccal cavities. They pass that on through tropophylaxis and feeding young, where they pop out, get ingested. They're internal parasites of the ant larvae in the beginning. They end up popping out later and more of external feeder on the pupae. And then they pupate themselves. And like most England, the ants are like, oh, hey, more of us. And they just kind of, tend to them, care for them, ignore them, kind of thing. All right, so that's the background of our characters. And so now our story begins where I got pulled into this UCR group. And so most of our collaboration is starting with food and drink, right? So places like here, ESA, we make friends and like, oh yeah, you work on that? Oh, look on that, I have that material, no kidding. So then we stay going. One year I had some NSF money to use up at the end of my my 5-year term with that. And so I reached out to a bunch of friends and such. And so Davide was one of the ones I brought in, taught my students about Ichneumonidae. And then we figured out, because wasps are too big, we need someone who specializes in smaller wasps. And so we brought in Luc, which somehow deserves an award for the most random presentations in this conference, but not being here. Third time his name popped up. And so once again, he also gave a workshop and a seminar like Davide did. And so then in the between times, he's looking through my massive collection. So I do a lot of malaise sampling throughout the Florida state parks for a lot of years now. And so hence when you're doing malaise trapping, as most of you know, you get a lot of material. And so he was looking through that and he came across the Orcina minutissima. And so that made John very happy when he told him. And then but the problem was that site I caught the Orcina. It was early in the collection. I still hadn't defeated the FDA and all the proper paperwork to get my ethanol. So of course I had to use isopropyl. And then of course the DNA was no good for Ashley's. And so John was less happy about that. So we need to go out and collect them again. So this culminated into this new adventure that we are now going on, or we just concluded our adventure. So Ashley, John, and Christiane came to Florida, and as when visitors from the West come, first we need to show them the sign of the scary alligator. They need to see the scary alligators, and then we must eat the scary alligators. And so then once they get used to that, then they're comfortable in the field, and we can set our traps. So the first stop was at Fern Forest Nature Center in Broward County. And so this is where the initial find that Luc pulled out of the samples was from. And so a mix of dry prairie, malt palm, maple forest, but more importantly, lots of ferns, right? And so we went and set his yellow pan traps, did some sweeping, no luck, put down the peanut butter boards. And yeah, there was some Wasmannia there, but no luck, it was kind of rainy. This was in December, the original catch was from like February. So that was a little bummer, but in the second stop, I took him up. I'm like, all right, I know a bunch of other spots where I got lots of ferns, which the habitat looks great, right? And so remember for Wasmannia, they like human disturbed, human changed, altered habitat. So I was like, I got a few of those with a bunch of ferns, let's go. And so we went up to Jonathan Dickinson State Park. So we did days of YPTs, white yellow pan traps. Nothing day one, it's all right, we go, had beers at the researcher cabin, and kind of, commiserated, oh no, it's like the third day, we don't have anything. But we found a lot of Wasmannia, so hooray, so we're hoping. So we put down another line of traps in a particular place over by a bridge, so a little bit of human disturbance over by. And we found them, so, which is another interesting thing, over and with other position scars on old world climbing fern, so on Lagodia, which is the big invasive issue. We're found only in Florida. We also found some on some Brackenturner fern. Don't hold us to that ID. It's still in the plant press. I still have to do the ID on it and such. But we found those oviposition scars. Of course, John was really, really happy because we finally got those going. And so, yeah. So then third stop, now this is all bonus gravy. So my current year-long sampling for the past 2 years has been in Highland Hammock State Park, where you can see there. We stayed for 2 nights because I have a researcher cabin I have access to, so it's free. And so, it's awesome. The park biologist is fantastic, supportive. So we swept, we set up yellow pan traps, didn't find anything the first day. And then once again, by the camp store, a little bit of human altered habitat with a bunch of ferns. Let's put a line down here. And then of course, there we go. So that's what they look like. Once again, little tiny wasps. And so, yeah, so we found them, so hooray. So now it's really exciting, right? So now on top of just finding them again, now we've delineated a northern extreme of the range that we haven't seen them before. Nice. So we're really getting far up there. So next step that Ashley is going to take, so she's going to do some genetics. So we found enough that she could run some sequences on, because remember, they're little wasps. We need a good amount of them to run these things. So we caught 12 at one site, so it was enough to send out for sequencing and stuff. And so now they're going to start looking at difference, what's going on in Hawaii, what's here, and some of those further questions. That's for her PhD. And so hopefully you'll catch her at an ESA or some other kind of conference so she could run you through that. All right, let's see. What else there? All right, now here's the most important things. I'm charging you guys. Oh, first I did a side quest. And so on fireworky kind of holidays, I like to take my dog and take him places far away from fireworks as a gift to him. And for me, so I don't have to fix things. And so I went camping at Seminole Collier State Park, and you see it's way west. So that's on another new edge possibly of the range. And so with some marital negotiations, I was able to bring some yellow pan traps, which is easy. My wife is also an entomologist, if you know her. And so, but running yellow pan traps is really easy. All you need is yellow pans, soapy water, and a fish net, right? And so laid those down, found, once again, a little bit of a disturbed habitat with ferns, which was by the bathhouse of the campground. Laid those down, went and had our family time, and on the way back, picked it up and found some of the leaf scarring. So I dropped some pans right there, and lo and behold, found another one.
Oh, nice.
So now we've expanded it. So now we've got the west, so I can see my great Photoshop skills there. So the red is kind of where now we've confirmed where my new Ptissima is. But we want to see how far they've gone out. Have they followed where we see Wasmannia? Are they following? We've only seen the little one, so we haven't. So we're focusing on Wasmannia here. If they hit Phydolia, hooray! But we've only seen the little version, to my knowledge. But see that big blue arrow? That's here. So you guys that are here, maybe throw down some yellow pan traps if you got some ferns in your backyard, stuff like that. Take a look and maybe extend it out. It's pretty easy. You mix some unscented soap, a couple drops a pan at a time, or about 10 mils per whole gallon. Pour it in, come in 24 hours, put it through a fine mesh aquatic net, and then dump the contents in a net with isopropyl. Oh no, sorry, John. Into ethanol. In case you get a lot, then they could do some genetics. But still, if all you have is isopropanol, still do it so that at least we could delineate this new edge of the range for it. And then send it to us. If you don't want to mail it all the way there, you can give it to me. I'll get it to them. So on and so forth. And of course, thanks to our permitting agencies. And so Broward County Parks has been really supportive with permitting, especially at Fern Forest. And once again, great support from staff. And there's our contact. So if you want to do an easy little side quest and help us out, then please do. That, leave them all up, right? Yeah.
Thank you, David. That was a great talk, and I'll let you give all my talks in the future.
One quick question.
Yeah.
Burn connection.
So that's where they're ovipositing into.
Always burn to any.
I believe, John, correct me if I'm wrong, but I believe the larger size they get on the Phydolia hits some kind of broadleaves, right?
Well, in Dominica, it's kind of weird. They partition themselves in a weird way. So the Bosmania ones are always on ferns, and the ones on Phaedolia are in a separate little area on broadleaf plants, but genetically they're identical. And in Florida, we collect the large ones and the small ones together, and Ashley's been able to find the large ones in a nest where they're probably attacking the queen brood. So they're having a greater impact by killing off queens in Hawaii than we know of in Dominica.
We got that one
Yeah.
there? One more?
If it's very quick.
Is it quick?
Yeah.
Just real quick, little fire.
You're talking about Wasmania queens, John?
Sorry, yes, the Wasmania queens. In Hawaii, she's got them side by side with the large ones we get in Hawaii don't occur in an area with Phaedolia at all. But we have found them in the nest and side by side with a queen, they're almost about exactly the same size.
Okay, thank you.
Thank you, David. Thank you, John.
And sorry, I should clarify.
Okay, our next speaker is Jack Grady. And the title of his presentation is Urban versus Natural, a case study of arthropod assemblages in the Orlando metro area.
Sorry, this is more like Adobe. The remote should work with Adobe. If it does not, ready? It's not working. I've done it before. It's at this button.
Yeah, works.
All right, so most of you are here from Florida, so I'm sure you're all very familiar with how quickly we're becoming a very urbanized state. We talked about it earlier today. Orlando, our study area, is one of those particularly fast-growing areas. And now over 90% of Florida's population lives in an urban area. So I've been listening to a lot of your talks today, and a lot of you are studying in natural areas, and that's super awesome. But because urbanization is becoming so popular, it's also important to study how arthropod communities change in urban areas. So urbanization impacts arthropods in different ways. That's part of why impacts of urbanization are so hard to quantify. Obviously, across the board, it's kind of negative. You know, we destroy their habitat, they don't have anywhere to live. But across groups, the effects of urbanization are different drivers and different degrees of impact across different groups. So this is kind of a broader study to kind of see larger trends of that. So our main objectives for this study were to compare community assemblages between urban and natural parks, which isn't done a lot in the literature. Normally, people will study only urban or only natural environments. And then beyond that, we wanted to study differences between different habitat types within those parks. So this is our study area. For any of you who aren't familiar, this is Orlando. Disney's down there on the bottom left. So we studied 9 urban parks and 3 wild areas across this Orlando metro area. And these are some of our habitat types for our natural parks. We have hardwood hammock, which is characterized by, you know, our big old oak trees, some lower canopy, but a lot of heavy leaf litter. Our pine flatwoods, which are our good pine prairie. We got our longleaf pines and just low-growing shrubs. Our sandhills, which still tend to have those pines, maybe some turkey oaks thrown in, but predominantly grass in the understory. Oh, sorry. And then our scrub, which I just clicked past, which is a lot of saw palmetto and shrubby oaks. And then our urban parks, we have our canopy, which is pretty similar to our hardwood hammock. our spontaneous, which has a lot of overstory and understory, a lot of shrubs in there. And then our good old turf grass. Don't need to explain that. So, like I said, this is ground arthropods. So we use pitfall trapping. We triangulated little pitfall traps in each of our habitats for each of our parks. And these are preliminary results. So I am only going to be able to tell you about our summer and our fall. So spring is coming, look out for it. And so far we've sampled over 60,000 arthropods and in over 450 samples. So that's kind of the scale we're working at here. So like I said, I kept this very broad. I know you guys are taxonomists and you like to go all the way down to like subspecies, but I kept this at order because I was looking at pretty broad trends. So I used some NMDS and indicator species to see correlations between our natural and our urban areas and then our habitats. So I don't know how familiar all of you are with NMDS. Yeah, my advisor scared me about it, so I have refreshers in here. It's called non-metric multidimensional scaling. It's pretty much just a way to put like your big giant Excel sheet for all of your count data into a 2D space. So these are like some examples of what an NMDS can look like. In these ones, each of those polygons are going to be a sample group. So in this example, there's no overlap. So those communities are completely distinct from each other. And this is what it would look like if they had some overlap. You know, they still had distinctions from each other, but there was overlap in our counts and what we saw. And this is a nested subset in which that large polygon right there is a lot more heterogeneous than this habitat right here, which would be much more constrained. So I'm going to go over indicator species in a second, but I want you all to think about what you would expect from comparisons between natural and urban. So this is our indicator species. It's just correlating it with a variable. And this is like the stat that indicates that correlation strength. What you're going to notice is that I do not have beautiful values like these 0.9s because of how broad my taxonomy was. But that isn't exactly why we used it. We used it to kind of show patterns in that strength, not necessarily to be like, oh, we find, you know, this group here, we find this group here. So this is my first NMDS for summer 2023, comparing natural and urban. So what we ended up finding was that our urban habitat was actually a lot more heterogeneous and a lot more broad than our more constrained natural environment, which I wasn't expecting personally. So yeah, we also have our indicator species over here. There are certain groups, like I said, Coleoptera and Columbella orthoptera, those are like relatively strongly associated with our natural environments. Those numbers are kind of what you're going to see in like the upper limit, just to kind of give you a bit of perspective. And we have other groups like our isopods and our amphipods that are more associated with our natural environments, or our urban environments, sorry. But to break that down further, this is a breakdown of our habitats in natural parks. So we've got our hammock there in blue, or pine, yellow, sand, pink, and our scrub is orange. So right away, you can see that most of these habitats have distinctions from each other where you only see that one color. So most of these habitats have at least some distinction in which their profiles are unique. And I'm going to click through these here because I don't have enough time to go over individually every group here, and I'm also going to show them later for our seasons. But some groups like our ants, they're strongly associated with our sandhills. Our coleopterans, which I'm going to get there, but they're associated with every group except sand. That happens sometimes too, like with our Columbella, they're found everywhere except our hammock. And this is our breakdown of our urban parks. Same thing here. We have distinctions in our different habitats and different groups associated with those habitats, like our hemipterans. We found a lot of Systematic in our turf grass. Our isopods, we think, liked that overstory canopy. And just to go through some of these groups, but this one right here is interesting that I wanted to talk about. When you break it down by habitat, some groups come up as correlated with both urban and natural environments or habitats within them. So we don't really know what was up with that. And this is what our fall breakdown looks like. So don't worry, I'm going to put them next to each other so you can see the change in a second. But our natural habitat remains constrained, but it lost its little poke out. It had like this little tiny part of that polygon that poked out, this teeny tiny bit of heterogeneity, and that's gone now. And the correlation of Columbella with our natural environments is also gone too. And those stats all got much lower. And our species, or our groups, sorry, correlated with our urban environments, that list got pretty long. So that's kind of what those look like next to each other. So you can get a sense of how those different groups and their associations changed over the fall. So this is a breakdown of our fall natural parks. You can see that while we still have those distinctions, certain habitats got more constrained, like our sand. And this was why I wanted to bring it back to you. Remember how in summer, Coleoptera was only was associated with every habitat except sand? That is now all it is associated with. And we would have to obviously break that down to a species level to see what caused that. But on a broader level, it's kind of interesting. And we've got, our orthopterans, they're still with our natural areas, but they're found only in scrub as opposed to with hammock and scrub last time. And then our good old spiders. So this is what those look like next to each other. So you can see how much more constrained, for example, the sand habitat got in fall as opposed to summer. And you can see like some shifts in some of our groups, like our Formicidae, which was originally associated with our sandhill habitat, isn't even registering as significant in fall. And this is our urban graph. Back before I narrowed this down to arthropods, I had snails in here, and it made my graph a nightmare. So I'm so happy to have been able to limit that for you today. So it is actually readable. But some of our groups stay the same, like our Hymenopterans, they stay with our turf grass. Our spontaneous group got kind of big. And then we've got, our earwigs and stuff. But this is to kind of give you that side by side and see how much broader and much more heterogeneous that grass habitat got from fall to summer. So we actually don't know why that is. And that's kind of what I wanted to emphasize. I know you guys are all taxonomists, and I would love for you guys to come back and look at these kind of broader patterns on a smaller level. I can't, I'm an undergraduate, I will be leaving. But like so far what we've been seeing is that our urban parks tend to be more heterogeneous than our natural parks. Which is kind of isn't what you would expect, right? You would expect, our kind of generalists to hang out, but that's the thing. We can't tell you why that is. In the future, we want to come back and do like some linear mix models to see if there are any correlations with our leaf litter composition, our below ground temperature, and our soil moisture to see if that is maybe what's driving some of our trends, like for our broader groups. But in the future. And again, some of our taxa are very strongly associated with our natural versus our urban environments. Our coleopterans, they stay with our natural parks. Our isopods, they like our urban areas. But yeah, and there are also seasonal changes. So you saw some of our groups lose, like their associations change, they gain significance, they lose significance. So like being able to come back and study the drivers for those larger patterns would be very, very interesting. Yeah, and just kind of a quick thank you to everybody who let us sample, and thank you to Alessandra, whose project I actually got this from. So yeah.
Any questions? We have time.
So the heterogeneity in urban places may be due to adventive species. So yes, because I know we're doing a similar study here in NATO.
Oh yeah.
And there are a whole bunch of adventive species that are here, in addition to the native ones that are not in the parks. And yes, there is a temporal seasonal variation. There are some species that are abundant in the northern states that are present only during the spring, well, right now, winter. Yeah, So that's just sprinters.
Yes.
So I can imagine that for the other groups, it's probably going to be the same thing.
Yeah, locally, that was kind of what I thought, because like when you're looking at your count data, you would have like, for example, like in an urban versus a natural one. In a natural, you would find like 4 ant specimens, but they would all be very distinct species. And in an urban one, you'd find 400 fire ants. And like, okay, so it's gonna skew your results, but you know, it could go to species. But yeah, those broader patterns are so interesting. I mean, I would love to go back and do that. Yes.
What sort of differences are we getting in absolute numbers in natural and urban areas? Are there big differences?
Don't remember if we do that now.
Number of what do you mean?
Of
Like
specimens.
the raw count between natural and urban.
That's a good question. Right now,
I'd have to go back and look at it.
Really, because when you have low numbers, then you have more potential for deviation. So that possibly have to explain heterogeneity, the fact that you just have low numbers, you're going to end up with these anomalies just as an artifact of that.
It can skew NMDSs. In general, we had pretty good counts for like we had. I think we cut out we cut out all of our missing traps, all of our low counts. We took out groups that because we had some like lepidopterans that were kind of just bycatch, you know, they don't live on the ground. So we took all of those out to kind of keep that skew out. In terms of the catch broken up by natural and urban, I couldn't tell you. But overall, our counts between groups were high enough that we didn't think that it would skew it too significantly.
okay, thank you, Jack. Okay, our next talk would be live Zoom. We have Alexander Orfinger. He is a study of the catfly's Linnean shortfall.
Hey, can you hear me and see my screen?
Yes, sir.
Yes.
Awesome. Okay, very cool. So sorry to all the beetle and wasp folks today that I'm going to subject you to caddisfly talk for the next 10, 12 minutes, but we'll get back to more beetle and wasp things soon, so hang in there with me. I hope you also appreciate the pun of case studies here for those who are familiar with caddisflies, but for those who are not familiar with caddisflies, you will be a little bit more here in a few minutes. But what everybody is familiar with is how little we know about insects relative to their biomass and their diversity. Considering how diverse they are, I mean, you can see on that figure on the left, significant shortfalls in terms of just the names we can put on species currently. And I think I'm preaching to the choir on that front, so I won't spend too much time on that. What a lot of people don't appreciate, though, is the fact that even if we can put a name on a species, a lot of these descriptions that we see could be characterized as like fragmentary, that and people have mentioned this throughout this meeting today, that entomologists have a preoccupation with male genitalia. I mean, a lot of times we just describe the males, which is a good start, but definitely shouldn't be the end all. And a lot of people tend to describe males, put a name on a species, and then move on from there to the next species, which shouldn't be the case. So for example, this is particularly a problem in the holometabula, where the adult and the immature stages are completely different, not only morphologically, but ecologically as well. This is seen in sexual dimorphisms, like I said, and like others have mentioned today, earlier today, is there's a strong male bias in a lot of taxa, in most insect taxa, I would say, in alpha taxonomy, and caddisflies are no exception. Of course, leps are no exception either. And then there's the Wallacean shortfall too. Our distribution knowledge is not well known. So a lot of people will find a species and from a single location, describe it from there. And sometimes oftentimes that's a limit, an artifact of time and money that are available for sampling. But sometimes the due diligence isn't followed, and it's very common to get gaps like these. So here are two of my caddisflies. And you can see they're found in all of these southeastern coastal plain states, except there's conspicuous gaps in Georgia, for example. And I'm now I live in Tennessee, but I'm working in North Georgia. And these are two that I'll be looking for, because clearly, I mean, I would be willing to bet good money that these are going to be found in the state of Georgia. And I work on caddisflies. So these are the most, they're not wasps or beetles, so they're not that level of diversity, but they're, I mean, you can appreciate that there's more than 17,000 species that have been named, and these are holometabolous freshwater insects, so they're the most diverse primary aquatic insects. They're globally distributed, they're pretty cosmopolitan, and because of their diversity, both ecologically and species-wise, and the fact that they're found everywhere except Antarctica, they're used in freshwater biomonitoring a lot. So you can understand that having these insufficient taxonomic descriptions that only address males, for example, let alone not even being able to name so many species, kind of limits what we can do in terms of biomonitoring resolution of fresh waters. They're often referred to as the underwater architects because of all of their beautiful cases that you can see vary across the phylogeny. And of course, some people also make jewelry with these, or in the case of a French artist, they will make cases with whatever you give them. They have preferences, obviously, but if they're stuck with gold and pearls, they will make their cases out of gold and pearls. And so this was part of a much longer talk that I've scaled way back. And so I'm talking just today about two shortfalls. And these are recognizing the unknown diversity. So we already know that there are a lot of unnamed species out there. And so being able to give us a benchmark on or kind of a goal of what we can expect diversity-wise, and also tackling taxonomic black holes that are just non-existent, that basically nothing is known of a certain area, which are challenging. And so again, we know for a fact that there are, known unknowns where we can expect a lot more diversity, but it's good to try to put good statistical estimates on that unknown diversity so we have a goal. So we have a target of how many species we can expect to find. So A, we're not wasting our time if we've met that mark more or less, and B, so we don't give up too early. And so here is, albeit an older map from 2008, but the trends hold up pretty well. The bigger numbers are the number of genera per, the number of species per biogeographic realm, and the smaller number, the number of genera. And you can see Africa, significantly lower than we would expect. The Oriental region's been very well studied, but there are areas that are there that are still needing to be explored significantly, and I'll mention that in a second. And this is the part of the talk where I ask you questions. So you thought you were going to ask me, and you can at the end, but I've got a quiz for you so far. So these are probably, two of the three of these are probably pretty easy. So who can tell me what the one on the left is, the silhouette of that, what country that is? Or I guess the one on your right is what it, I guess it'd be on the right.
India.
Yeah, that's India. What about the one in the middle?
Brazil.
Yeah, that's Brazil. And now the hard one. What's the other one?
Ecuador.
Okay, wow. I can't see whoever said Ecuador, but well done. I didn't know who would get that. I thought, maybe Will's Flowers would get that or something, but that's impressive. I've been to Ecuador 3 times, and I have this in my talk, and I'm still not sure if you gave me a big lineup, I'd be able to pick that out. So that's very impressive. But I would show these countries because these are the 3 countries that trichopterists have tried to give estimates on the total diversity of the caddisfly fauna of the country. So recently, my Indian colleagues and I worked on this for India, but we followed the methods of those folks that have done this in Ecuador and in Brazil. And this was using the Chow-2 species instance estimators. So in Ecuador, they have 310 recorded species, and they have an estimated 578 species. In Brazil, they have an estimated 796 species. They have a reported 796 and an estimated almost 1,700. And just recently, it was just published like a month or two, like 2 months ago, we found in India that we have 1,435 recorded species and an estimate of 3,400 species, which a previous well-known trichopterist, Herbert Ross, estimated higher than that, but not too far away. So we're within the ballpark of previous estimates, but this is the first time people have used stats rather than kind of just ballparking it. And so this is the percent of the fauna that has been described based on those estimates. So Ecuador, about 54%, Brazil and India, less than half, but all of them have significant shortfalls here. I didn't want to be unfair to Ecuador, because if you're comparing India and Brazil, which are just monstrous countries, to Ecuador, that felt kind of unfair. And so comparing this from a unit area measure, Ecuador actually has the most species per unit area. So they've got the most diversity if you take size out of the equation. So kudos to Ecuador on that front. Brazil, you know, sorry to bring in size into the equation there. Our second, the second case here is just total taxonomic black holes. So those are in India and Ecuador and Brazil. These are countries that have really good Trichoptera research history, and clearly a lot of work left to do, but really good Trichopters that are working hard and making a lot of progress there. But there are areas that are just total black holes. So currently, for example, I'm doing a Caribbean project and looking especially at Honduras. And Honduras, there's almost nothing to work on. So it's very difficult because there's no framework. Common features of these taxonomic black holes, they might be small areas, not a lot of scientific infrastructure, very little freshwater habitat, as you can imagine, and a lot of habitat conversion. So, for example, Eswatini in Southern Africa and any number of small tropical islands, it would be unsurprising to learn that there are basically no caddisflies here, or if there are, like you would expect in Eswatini, there are just no records of caddisflies. But there are other places that would surprise you to know that there are no caddisfly records. So, for example, Singapore, until this last year, had 0 country records. And granted, Singapore is a very small country, and it is hyper-developed, but they still have decent natural areas. They have preserves in the country, and they have a very good scientific infrastructure. And so the fact that there are, yeah, I also doubt there are many in the Maldives. I'll just have to see if I can get a grant to go to the Maldives to check just in case. But in Singapore, you know, you would expect some, given that they do have some fresh water. And so just this past year, Singapore colleagues and John Morse at Clemson University went and looked, and they caught the first 12 species records, 12 or so species records of Singapore caddisflies, a handful of which were undescribed. And then, well, okay. And then another example that I've honed in on is San Marino. So San Marino is an enclave of Italy. It is an incredibly small country. It's about the size of Gainesville. But if I'd mentioned to you that if I told you that there are no caddisflies in Gainesville, you'd probably look at me dubiously, and that would be fair. Like, you would expect some caddisflies. Now, San Marino is very developed, a lot of habitat conversion, but again, good scientific infrastructure, certainly in Italy, right around San Marino. And they do, as you can see, have waterways like here, the Ossa River. So you would expect some caddisflies. So let's zoom in on that Ossa River. Look at that beautiful river. Who would not want to take a swim in that beautiful river? And, you know, so maybe it's not so surprising that there are no caddisflies in this particular area. But if you look at some other places, there's still some decent habitat in San Marino, some decent freshwater habitat. So this area near Aquaviva, for example, that's solid habitat, and eyeballing that picture, I would expect to find some caddisflies. So the next caddisfly symposium, which is every 3 years, the previous one was in Ecuador, this next one is in Croatia. So after that meeting, which is in the summer of 2027, I am hoping to hop over to Italy, and while there, stop in San Marino and do some sampling to see if I can come up with some caddisfly records for the country of San Marino for the first time. And if I visit areas like this, I expect that I can. So any Italian colleagues that want to touch base, feel free, and I'd be happy to talk about that more. And so clearly there's a lot of work to do. I love this quote by the senior Oliver Wendell Holmes saying, "I suppose you're an entomologist, but not quite so ambitious as that, sir. I'd like to put my eyes on the individual entitled to that name because no man can truly be called an entomologist. The subject is far too vast for any single human intelligence to grasp." I could work on just the caddisflies of Georgia for the rest of my life and not finish anytime before I die. So clearly a lot of work to be done. Caddisflies are a taxon ripe for plenty of taxonomy. And so hopefully if any of you all get bored with your beetles and wasps, we'd be happy to have you on. So with that, I'd like to thank the Department of Education Title III grant for funding a lot of my recent research, the Center for Systematic Entomology, Dalton State Department of Life Sciences, who've been very, very helpful, and many, many more folks. And with that, I will take any questions. Thank you so much.
Thank you, Alex. We have time for one quick question. Yes, Davide.
Hi, Alex, it's Davide. I wanted to ask you about San Marino. Let's talk about the San Marino in Italy. Do you think it's a matter of lacking infrastructure in San Marino, permits that you know of, or something different? What do you think about that?
Wouldn't be surprised if it's, I'm surprised no Italian researchers have looked there, but and I've been looking into permits, but I think it's probably lack of I don't know about infrastructure, I guess. I cannot find anybody working on insect systematics in San Marino. They've got like one university there that has a science department, but I can't find any folks to contact in San Marino, which is why I'm looking for nearby Italian colleagues that might be interested in joining me. Now we've got plenty of time to plan it because I don't plan to be there for 2 more summers, but yeah, I think it's just nobody in San Marino probably is qualified to look at caddisflies. So, but I know there are folks around in Italy that are.
Yeah, I think it's a matter of Italians are not really interested in San Marino. There are much more better places around it. So that's
Sure.
what I think is happening over there.
Yeah, I wouldn't be surprised.
Okay, thank you, Alex.
All right, thank you.
Now we have our break, and I'll see you in 10 minutes.
Great talk, Alex.
Question. To behavior, stories of tiny specialist beetles. Stage is yours.
Okay, so my name is Ethan Wright, and I'm a first-year PhD student at UCF, and this presentation is going to be mostly about the work that I'm wrapping up coming into UCF, and then my work going forward at UCF. So to start, we know that Coleoptera is known for its immense diversity, which is being challenged by a lot of Hymenopterists today. But many of these non-economically important beetles lack a study of their comprehensive life history. Challenges that present themselves when trying to study these things is cryptic lifestyles, niche habitats, and their small size makes them hard to observe in a lab setting, especially in a field setting. But I think that all these things are what kind of endears me to these smaller groups. One of these smaller groups is Fuchsina in the family Lectrodeidae. It is the definition of a minute beetle. It is only around 1 millimeter in length, and the genus is mostly eyeless. They're likely mosquitophagus. This is a guess because of their closely related species in the family, and they can be found sifting various types of leaf litter in the West Coast because that is where the entire range of this genus is restricted. Previously, there were only 2 described species.
The first one had eyes completely absent and only 10 antennomeres. These are SEMs from Andrew 1976, where he described a second species, Fuchsina arida, based off of the eyes reduced to a single small facet and 11 antennomeres. So knowing these two species, I was identifying a Fuchsina from a project in my undergrad, and I found that there was a specimen that did not quite fit the bill. It was a mixture of both. It had 10 antennomeres, but an eye facet, so it was the intermediate of these two. So we thought, slam dunk, quick one species description. Let's get some material. So my co-authors helped me get some material from the CDFA, the Santa Barbara Museum of Natural History, and then our own personal collections for a total of 528 specimens. And I found that there were actually 5 new species of Fuchsina, so a little bigger than one. I based these new species off of solely morphology. Here's the character matrix, and we'll go into each of these individually. First, these are all SEMs that we took and images that we took for our publication. The antennomeres are only 11 in Arida. The rest of them have 10 antennomeres on the right. Next, we have the Ommatidium being reduced to a small eye facet, or it can be completely absent. Next, the male has these bicarinate ridges on the final ventri of the abdomen, and they can either be present, they can be absent, like completely flat, and you can see the adiagus extended in both of these pictures, so you know it's a male, or it can be extended to the internal ventri as well, in a special case for one species. Next, the entire genus has these fovi on their prosternum, right in front of their prococci. These are basically a little divot with a hole that goes into their pronotum, and this can be distinct, which is characterized by being filled with CD, as you can see here, or it can be indistinct, which one species has it indistinct, where the CD are pretty much absent, and you can see straight into the hole that goes directly into the pronotum there. So here is that without an SEM, you can clearly see all these CD in those holes, while the species that does not have the CD in the holes, it is much harder to see that fovi without SEMs. Next, the adiagus could be broad at its apex, or many of the species have a narrowed adiagus when extended. I also did a lot of dissections for these and slide mounted their genitalia as well. Finally, the overall body shape of these can be elongate, they can be normal, or they could be stout in one species. So there's still so much to know about this. We don't actually know the diet of them. They've never been successfully reared in a lab, and we don't know any of the purposes of all these different and weird morphological features, as well as there is a potential Mexico species, but there was not enough specimens or time to identify that one. So we've named them, but taxonomy is just the base foundation of these species-specific life histories that can be described. So life histories and life history traits, like being macetophagous, like Fuchsina, could be herbivorous, like Chrysomelids, could be predaceous, like Carabids, or it could be Myrmecophilous. Myrmecophily is literally translated to ant-loving. Happy Valentine's Day. So a myrmecophile is any organism that lives in association with ants. These can be mutualistic, commensal, or parasitic. And within each of these, it can be a sliding scale of facultative, so it can live without its ant host, or it can be obligate and it requires its ant host to complete its life cycle. One of these obligate myrmecophiles is the genus Nostis in the family Cnidae. There are 3 currently described species, and they are all fully integrated myrmecophiles with the ant in the genus Chromatogaster. They also have a neotropic and subtropic distribution, as seen by this map I made, and they have a long history of taxonomic misplacement. They even had their own family erected for them at one point. And we have one local species. It's Nostis floridanus. It has 3 currently described Chromatogaster host species. There's been one publication on its behavior so far that kind of laid the groundwork that it was in fact a myrmecophile and observed a couple behaviors.
So the two main hosts that I've found it with and the people I found it with that I've talked to are Chromatogaster panicula and Chromatogaster ashmedii. These two species were actually recently split. They were synonymized and then split. They're sister species, so they look pretty much the same besides their bicoloredness and their habitat preference. But both of them can be found in slash and longleaf pine. The left one, panicula, only being found in pine trees. And then finally, we have a third potential species host, Leucayana, which is in the Bahamas, which is found in the culms of grasses. So collection methods to get these nostis. I remove dead branches and bark from these slash pine and longleaf pine, and I shuck through them, and I remove all these ants from these little crevices of these pine trees. I've also checked ashmedii colonies in vertical oaks, some beautyberry stems, and other places, and have found no nostis in anything other than pine. And then once I remove them from this pine, I put them in an observation nest with glass on top of some wood nests that I make, and then some red acetate on top to make them think it's dark in there. And I record some videos with this camera microscope in our lab. So there was one behavior mentioned in the original publication about their Myrmecphil's behavior, calling quaking. Here you can see the beetle, you can see the beetle shake a little bit. It's more like a shiver. It's very subtle. I found that it does this when it's like looking or searching for the nest or outside of the nest. Here's it slowed down to attempt to be able to see it, kind of. Next, some behavioral observations that I've made. There is active solicitation of trophallaxis from this beetle to its hosts. So you see Gnosis walking up to its host ant and initiating trophallaxis by touching its antennae to stimulate the host to give it food, pretty much. And you can see the mouth parts of the ant connecting with the mouth parts of the beetle. And you may have noticed at the end of this video, it does this lovely little side-to-side shake that I'm calling a listing behavior. I do not yet know the purpose of this. It could be to move food through the body of Gnosis, or it could be a tactile mimic, because if you'll notice that during trophallaxis, these ants wave their abdomen up and down while they're giving food. So maybe it's just like imitating that. And then I've even found that it solicits trophallaxis from a female alate, which is kind of a big deal. If you know about ants, the female alates are basically the reproductive success of the colony. This is like a direct fitness impact if it were to take enough resources from these alates. And it's especially important for these Chromatogaster because when these female alates copulate, they pretty much like box themselves in a branch and do not exit the branch at all until she has some offspring, and she feeds her offspring solely off her fat stores. So if this Nostis is taking food from a female alate before she goes and flies and reducing her fat stores, it could be impacting its fitness going forward. So I wanted to quantify the costs of Nostis in these colonies. I did this by recording footage of Chromatogaster engaging in trophallaxis with nestmates versus Gnosis floridanus, and I timed these interactions. Then on the right here, I have a mean trophallaxis time with standard error bars with the host to host and host to beetle signified by the little pictures. And as you can see, there was not much difference. They all seem to be about the same. I also ran some models with ANOVAs and found that there was no statistical difference between one Nostis and one worker within a colony. This is kind of interesting because so far the most amount of Gnosis I found in a colony is about 8, and these colonies can get up to about 10,000. Wow. So parasite taking 10,000s of your resources does not seem like it would have much of an impact on your colony's success. So it's probably a parasite, but is its impact negligible in these nests, or does it actually have a direct host fitness impact when it takes food from the alates or workers? Another interesting thing that I found is that I can take a Nostis from its original colony that I find it in the field in
in a Chromatogastr panicula colony, and I can take it from that and directly move it into another Chromatogastr panicula colony. As well as I can take it from a Chromatogastr panicula colony and exchange it into a Chromatogastr ashmead colony, all without it ever being attacked or shown any aggression towards it at all. Here are some examples of that. On the right here, we have some ants that were moved into colonies that they are not a part of. It does not go well. And on the left here, we have Nostis getting a spade. The ants are actually grooming it. They are grooming each other. They're showing no aggression behavior. On the bottom right, you can see gastriflagging as a aggression behavior. They're like dispersing chemicals, presumably. And on the top left, top right, you can see that lunging and attacking behavior. But Nostis does not receive any of this, even after being immediately transferred from deep inside one nest to another nest. So some future directions I'd like to go with this is like standardizing the trophodelaxis test, getting the proportions of workers to brood all standardized in there, and then controlling the amount of food that is put into the system, and then timing that as well as some other variables. And then I would like to potentially test some experiments with founding queens, so I can maybe calculate if there is a direct fitness cost if these female alates are getting too much food taken from them, or if a young colony has these parasites in it, if there is a parasite load or something like that. As well as another interesting thing about this whole genus is that they have these trichomes on the hind angles of the pronotum. These might secrete some appeasement chemicals or something that changes the ant's behavior that could potentially be interesting. So some challenges I'm going to face with this is that collection of a whole colony of these ants in a pine tree likely requires the death of a pine tree because I need to strip all that bark down. So I need to find trees marked for lumber because I don't want to just go around killing pine trees. And then lab rearing of Nostis ordalis has so far been unsuccessful. We don't know what the larval stages or immature stages look like at all. And I have gotten them to copulate in the lab. I have video of them copulating in the lab. But I have not gotten them to successfully oviposit. Thank you. Questions?
Great talk. Yes, we have time for questions.
So there is one online. Have you examined Fuchsina gut contents to look for potential food items like spores or other solid matter?
No, but Andrews 1976 did remark how those fovi with the CD in them could be for moving spores around because they were packed with spores. So they could be potentially moving, I don't know what the correct word is, but moving spores around with those.
Right.
We have another one. Yes, no. Oh, yes, yes.
Okay.
Speculate where the larvae.
Well, I mean, What do you know about human larvae? right, it could be a trash eater, it could be, their association with pine trees makes me think that what if it's like in the wood to any degree, but I mean, maybe it's just being like oviposited in these ants like trash piles and the larvae kind of develops on those trash piles or something like that. It could be, but so far I have come, right, but so far I haven't, out of all the brood, I haven't really found, yeah.
Yeah.
No, we didn't.
Yeah. Okay. Okay. Thank you, Ethan. Hey, our next speaker is Andrew Johnston, a taxonomic checklist of Platypodidae.
Full screen? Yeah. So today I'm going to be talking about a short-term method to make a checklist for the Platypodidae species, but also kind of the broader kind of other projects that we're working on Platypodidae. So just quickly about me, I am mostly a research scientist at the University of Florida in the School of Forest, Fisheries and Geomatic Sciences, and the Forest Entomology Lab with Yuri Holzer. And I'm also a biological scientist at DPI, Institute for Agriculture. Yeah, I missed it out. And I'm also the PI for this big NSF grant that's funding this project about platypodines. So first of all, what are platypodines? Of course, they're weevils, and some of you might know that, but really they're like a perfect rodent. They are like this tiny little cylinder, and they have really hard mandibles, and they probably have some sort of metal embedded in it, and they kind of spin around as they bore into wood really quickly and really efficiently. They're also fungus farmers. They are ambrosia beetles. They are possibly even the oldest of the ambrosia beetles. They are older than all the scolytines, and they're older than fungus farming ants. So they are kind of really interesting in that sense. If anyone has done light trapping, especially in Central South America, South Florida as well, and increasingly in other parts of the tropical world, you've probably seen this species. This species is incredibly common in light traps. This is Euplatypus parallelus. We have a similar species in Gainesville. That is a couple more. Yeah, this is the one which most people see. We've got thousands of them in FSCA. But they are, so the platypodines, they're the ambrosia beetles that are not scolytines, which is kind of different to how scolytines are. Scolytines are bug beetles, and some of those are ambrosia beetles. It's like a subset of them. And there's many different types of bug beetles. Bug beetles are ambrosia beetles, and they're separate evolutionary origins. These three here is just three of like 10, 13 different origins of fungus farming. They also do lots of other stuff. They also do lots of different mating systems and everything. Whereas platypodines, they all do about the same thing. They're all monogamous. They all are male-initiated galleries. They all bore into wood. They all grow fungus. So in that sense, they're kind of a lot more stable. But really, we don't know much about them. They're also weird. This is an African species, and I mean, what is even going on there? There's some weird cavity here, and this one also has little pores on the top, which are typically where the mycangia are. It's just bizarre. So actually, I'm going to go back, and so we're going to be starting lots of different projects on platypodines. So I'm going to go through just to introduce all these projects. So in our lab, we've got Laura Richardson, who's going to be looking at these in these fungus pouches, like you mentioned, possibly with this other species. All the platypodines are fungus farming beetles. They go into wood and farm some kind of fungus. And we just don't know much about it. There's a few examples, mostly of species living on oaks, and kind of know a few fungi, but it's really, that's just a handful of more than 1,000 species. So this project will be looking at the actual communities and also looking across diversity, because we're only really looked at a few species in the crown of diversity. We also want to look into a bit more into the mechanics of it. So Miranda Barnes, who's currently finishing a master's, working on the genomics and transcriptomics of scolytines, like seeing the mechanics of the symbioses. She'll be applying that to platypodines, which will be really interesting. Hopefully start that next year. We also have, I mentioned there's lots of fungi to describe, and Katie Dietz in plant pathology with Matt Smith will be actually describing. Here's an example of something that's described a while ago from Prostotarsus from one, I think. But there's so much diversity, and it's actually not being described as a real impediment for groups beyond beetles too. And then we have lots of projects of digitizing the diversity. So we have Avery Kupeda and Nathaniel, who just started, and they are taking lots of photos. And we've actually got it, we've only been doing this for like about a month so far, and we've got it kind of pretty efficient. I mean, this is just some of the specimens we've got. And this is kind of, many projects need photos of specimens. This is trying to get as much diversity as possible. And in just a month, I think we've done 300 specimens or more. So I think it's really attainable that we can get comprehensive coverage of all the species, if only we can get them. Certainly in terms of taking photos, we're really trying to get like a lot and really scale it up so we get multiple examples of different species. But Daniele Sommaggio is also working to kind of modernize the keys just to get it started. There's some really, really weird kind of primitive platypodines. This one's native platypus, which is known to be a basal lineage in Australia, and that's a Platytarsulus, which is another great, probably. And then also, while taking all these photos, a lot of the sample is actually from a big altitudinal gradient set from New Guinea, and they're all unidentified. And so I'd also be working on getting that. There's probably tons of new species there, so it's just like some of the ones. It's really cool to set thousands of meters from New Guinea across altitudes there. But then also using all of these images, Christopher Moraz and Nathaniel would also be working towards using all these pictures to help see if various AI techniques can help to limit species, classify species, identify beetles, actually be useful as tools to identify unknown beetles. There's also tons and tons of other projects. Lots of collaborators are helping from all over the world, which is really important because the United States only has 7 species. Florida only has 4 species of platypodines, whereas there's over 1,000 across the world. Most of them are in the tropics. So we really need collaborators for this to work. And there is stuff happening. They just need to get it connected. So I came here to talk about a checklist of names, and all of these projects, I mean, one thing that unites them all is just a need for names, and it just is so much easier when you have a solid, just something you can refer to and everyone's referring to the same thing, and they don't have to, the baggage of like figuring out if this name's valid or its history or anything like that. And there's just no resources like that. There are probably the best resource is if you remember, there's a catalog by Wood and Bright from 1992, and that one was just a year before a massive revision by Steve Wood, who basically like renamed about 500 species. So that's not very useful as a resource either. There are some resources since, and they're not truly digitized. They're not in a way that you can actually search for the names. They don't come up in any of the other databases. You can't really extract them. And they also contain all the errors for Wood and Bright that haven't been corrected yet. So that is something that's really like important for anything to happen. So ultimately, what I want from this project is to make a digital monograph. I mean, everyone wants this, just everything there. Like every citation, every use of a name, everything about every species. But really, like that's not going to happen. That's a really long task that will take many, many years. I mean, it is feasible. It's not impossible. It just take many years. It would probably take like 5 years of work. And really, like we need these names now. And I could keep them and keep them, share them with my collaborators or anything, but that's not really how it should work. It should be make something now and then share that. Additionally, by doing it sooner, just making a list of names only, kind of compromising on its kind of thoroughness, we can actually use those names to plug into other resources. I don't know if people have used Biodiversity Heritage Library to search for taxon names, but once it knows the name exists, it's really good for searching old literature. It just finds it. So in terms of making this comprehensive catalog, not only is it much easier, but it might not even be needed as much because other things are already doing it. You just need that name, that anchor to kind of link it. And also Plasi, I don't know if anyone's familiar with that, but that's a really interesting project, which is really like digitizing these old taxonomic treatments and actually making it so computers can really understand it. And also make them freely available because that's the biggest challenge, is like getting literature. So my intermediate goal, which is kind of a compromise, is just to get the current valid names and the original description and the description of synonyms. And to me, that's minimal because certainly we're going to be photographing types, and that's you need the synonyms if you're going to be photographing types. So just a workflow of where this comes from. So I mentioned Wood and Bright. That's a huge effort. That's an incredible catalogue. And really that's what kind of fed into Tom Atkinson's database. And that's what a lot of this is based on. You can see on barkbeetles.info or kind of snippets of it, but really that's his like work he's worked on for decades and decades. And he's sharing that with us, and it's really useful. And kind of the process of making the checklist, in some ways I'm really familiar with this because we're also doing the same for all the scolopines, which is 6,500 species. So the process is we're kind of using that. This is a two-way arrow. I mean, part of using it is also giving feedback. And then through a whole network of collaborators, like 10 authors or more, kind of getting that checked, really getting people to check it, because I mean, making it correct is really important. And then from that, generating a checklist, and importantly, generating in a way that's going to be usable for computers too. Because what I really want is I want it to be taken up by GBIF, taken up by all the other resources, which will like use it for other things. So here's a little snippet of the Platypodon checklist. It still needs corrections, still, but it's getting there. We're up to 1,343 species. And this is generally how it's laid out. This is all generated. It comes from multiple tables and databases. So this is just a generated checklist, and I try to make it as kind of pleasant and simple and as much as needed. And because it's generated, I can also do stuff like all this, the letters and everything are generated local for the actual list being present. And just to kind of go through like the minimal stuff that I'm doing, just the current combination, the original combination, the page citation. Doing that is really useful. It makes you check if it's actually a good publication. There's many errors that have been found that way. And then the original descriptions. There's some which I've highlighted, like these ones, the pages don't make sense. They've probably been entered wrong. And I guess one thing which maybe, if anyone's got any opinions on this, I really hate when taxonomic checklists reference a publication, but that publication is actually a section of a publication, and it's really inconsistent because sometimes if it's the same author, sometimes it's not. So I just don't like how that's done. So I've kind of done it more in the actual checklist. And if anyone thinks that's wrong, please tell me, because that's how I'm going to do it. So, I feel like this is a lot more elegant than rather than duplicate entries in a bibliography, because technically it should be that every description you should have a separate entry. I'm not going to do that. So, yeah, that's where I'm. So we're at many and nice summary of how many species there are. And with that, I want to acknowledge lots and lots of people, lots of people funding, all the people that kind of keep departments running and everything, all the administrative people, all the museums who have visited. We're going to visit many more. We're going to try and learn as many specimens from Europe as possible and photograph as many types as possible. And just lots of different collaborators. As I said, that's really essential for it to work. So if there's any questions, I'll wait on this screen.
Yeah, we have a few questions online.
We got one question from Luc Leblanc. Are the New Guinea specimens that you're studying mostly at the Bishop Museums?
Say that again?
Is wondering if the New Guinea specimens are from the Bishop Museum.
I need to check. That's one thing I want to do soon, is compile where they all are. And if they are, it'll be nice to get them digitized. I do know the Bishop Museum had a lot of specimens from, because the Pacific has very low diversity of platypodons in general. But I know that Bishop Museum has stuff that's not in the Pacific too. Right. So if it does, that'd be.
So if you want to see also, there is a comment about Matt about what they're doing with the BHL. So
yeah, so that'll be good. Just want to make sure it gets into the systems and not be like locked in.
Quick comment. The Wood and Bright catalog of 1992, Don Bright and Bob Skidmore did 2 updates at 10-year intervals, updating everything.
Yeah, there's also Bright made another one in 2021, which, I mean, that's really, it's still not fully digitized. It's not in a way that you can actually, although it's available as a PDF, you can't, the names aren't there. They're not integrated with other systems. Which I mean, that's why I think making like either publishing in a journal like Zuki's or something like that, that will actually do the work for you and put it in those places, or including a Darwin Core Archive of the taxonomy or something like that, or using something like TaxaWorks, which will do that for you. That's really important.
Thank you. Okay, so our next speaker is Lily Dieter with the talk, A New Grass-Infesting Felt Scale Insect.
Okay, awesome. Hello, everybody. My name's Lily. I work at FDAX DPI right next door in the FSCA. So I work as a support scientist. I help do the identifications for the scale insects and whiteflies. And today I'm going to be talking to you about this cute little bug here, which I'm currently working on describing. There's a lot of really cool new species of scales that are found on grasses, not necessarily because they're rare, but because they're really, really well hidden right under our noses. So I'm just going to tell you a fun little story about how we found this thing and how we learned more about it. Sounds good? Awesome. So quick background, what even are scales? They make up the infraorder Cocomorpha. You might also hear the word Coxoidea thrown around. And this is a group that is a lot more diverse than I think a lot of us realize. So we have over 30 families and about 8,000 species. I know the Hymenopterists in the room are probably like rookie numbers, but my point is they're more diverse than I think people tend to realize. These also make up, they're all plant feeders. So these are going to make, this group is a lot of important agricultural pests, which is why I am employed. And whenever when we're studying scale insects, we make slides. So we use various chemicals to remove all the fat and wax, and we stain the skin so that we can look at microscopic characters on the derm of the adult females. And another cool thing about scale insects is they have extreme sexual dimorphism. So we look at the adult females for taxonomy, and they basically look like big versions of the nymphs. Some people describe them as blobs with mouths. And whereas the males go through the pseudo-complete metamorphosis, where they emerge looking more like a real insect, where they have visible antennae and legs and wings. So I think it's pretty cool because you can have a male and a female of the exact same species and they don't even look remotely similar. And so we're very interested in collecting scales from rats. So this is a new species that my boss Aaron is actually working on right now. Like I said, there's a lot of really cool new species of scales, but they're hidden because a lot of scales are these fleshy, defenseless little things. So they like to hide in crevices, such as grass sheaths. So one day, Susan, Halbert, and I were outside DPI, this was a few years ago. I don't remember what we were doing, but Susan noticed that one woolly grass plant, Muhlenbergia capillaris, very common landscape plant. She noticed that one plant we had on campus had some sooty mold on it, so she suggested we check it out, and we did. And lo and behold, we found some little white things inside, brought it into the lab, and I made some slides, and Doug Miller is one of our retired research associates. He's a volunteer who identified scales for the USDA for decades. He looked at it and he helped us realize that it was likely a new species of felt scale in the genus Acanthopus. Now, what is a felt scale? I think a lot of us are familiar with armored scales, mealybugs, coccids, and the like. This is one you might not be too familiar with, even though it's the 4th largest family of scale insects. So their name means aereo, comes from wool. Cocos means berry. And so they're named for these very felty, wool-like ovisacs that cover their entire body, kind of like a cocoon when they're ready to lay eggs. Not all members of this group do this. Some only partially enclose themselves. We have some in that upper right-hand corner. Some actually make galls, which is pretty cool. A very fun group, definitely one of my personal favorites. And the genus we're looking at today is Acanthococcus, with acantho meaning thorn, because these are covered in little thorns. My boss Aaron calls these the porcupines of the scale world. And so these are one of the most important diagnostic characters we have for this group because there's a lot of diversity in the shape, size, distribution of these things. I'm not going to bore you with all of the characters we use to identify scales, but I am going to talk about this one because I think they're really fun to look at. They might only have these enlarged CD on their margins. They also might be curved. They also might be dome shaped like this one. This is one of my favorite species that infests our native sundews in Florida. And this is our new species. So you can see it has these long, slender marginal CD only on the edges of the body. And the body itself is very long and slender, which is very common for grass infesting things because they can fit in these long, slender leaves. I actually went outside a few minutes ago and brought in a sample from the moldy grass we're talking about. Very long and skinny. And I mean, look at this thing. It's cute. So I kind of am obsessed with it. So I wanted to collect as many of them as I can. So we found 3 localities in Gainesville. The first one being the one Susan found outside DPI. Julietta actually turned in the next sample we found, which came from Swamphead Brewery, actually. We love Swamphead. And the third one was from a park that's like right by my house. This is very convenient. I mean, this thing was at my job, basically where I live and where I get beer from. So what else am I supposed to do? I have to describe it. I don't really have a choice. Then I did find a 4th location, which was actually at FES last year. I made Felipe pull the car over so I could go check some plants outside the research station there. And this was actually where I found my first male specimens. These are even tinier than the adult females. They're only like a millimeter. So yeah, again, very convenient. I don't think that this bug just likes hanging out where entomologists hang out, even though it seems like that. Odds are this is probably all over the state. This is a very common landscape plant. I call it a parking lot plant. It's all over the place. So why are we just noticing it now? It's very cryptic. I made this cute little figure on the bottom right here, which is only slightly exaggerated. This is what a cross section might look like, but they really they have a very flat dorsum and a very convex ventral surface, so they really just fit so perfectly and snugly inside of those leaves, which I think they're probably host specific. I haven't found it on any other grass that is similar to this. Again, so you can see in that upper right-hand corner, you can see the scale, but a lot of the leaf is completely, it's like a full tube, like completely enclosed in there. So these things are extremely cryptic. And so what I have to do is I have to kind of peel open the leaf. I was using either my nail or some forceps to do that. And I was doing that with a lot of grass and I was getting very bad wrist pain from that. So I had to change my methods. So I started washing them in ethanol. This is a trick I learned from our mite lab at DPI, where I was putting handfuls of the grass into jars with about a quarter or a third full of ethanol, shaking them up, and then pouring them through these sieves. The one on the right in particular is very fine. It'll even catch the first instar crawlers. And it worked really well. I could collect a whole lot at one time and postpone my carpal tunnel syndrome. And as for molecular data, Matt Moore helped us get some CO1 sequences from this and also 18S, which is very helpful. But we also don't really have a lot of data on scales in general. I could get the whole dang genome of this thing, but there's really only 2 other species of scales that actually have their entire genomes, and there's really only a handful of things that have CO1. So we need more data on scales in general. And this is why we rely so much on morphology when we're doing taxonomy. And all this is to say that this isn't a surprise to us. There's so many things that need to be described out there, but there's a lot of really cool things hiding literally in your yard. I mean, you could find new species by going out to the woods and setting traps, all that, but you could also just go into your yard and pull up some grass and find some scales and give them to me. And as for what's next, I thought it'd be fun to show you the illustrations that I've been working on using a camera lucida to literally trace the specimen. I'm gonna ink some finals for those. I also have been finding parasitoids in these. So I think they're wasps. I'm not entirely sure. It's kind of hard to tell. They're very small. Some of the wasp people in the room, maybe you can help me with this. I really want to rear some out, and I've tried to. I haven't done it successfully, so maybe wasp people, maybe you guys can help me. But yeah, that's all I have for you. Any questions?
Yes, we have time.
How are you able to determine that the male was actually associated?
That's a great question. So if they were in the same infestation together, there was really nothing else like around, basically. Ideally, we would be doing molecular to determine like, yes, the co-1 from these two match. But I mean, felt scales are pretty uncommon. And the odds that I found like a Acanthococcus male next to an Acanthococcus female where they're different species just isn't very likely. Yes, in the grass. Yeah, the male. So I found them actually in their pupa. So we call them pupa, even though they're not a real pupa, but they go immotile and they cover themselves in a cocoon. So they look like the adult female opasac, just a little bit smaller. And so they're living together, cohabiting.
How much damage do they do to the grass? And
so I haven't really noticed a lot of damage. I think, so I mentioned the sooty mold on the plant that Susan and I were looking at. I think that was from something else because I found pretty heavy not heavy infestations, but the reliable sources of them, the plants seem to be okay, but I do seem to find them in mixed infestations. So I found them with some other mealybugs infesting that grass, and there's usually armored scales with them as well. So they seem to like plants that are already getting hit a little bit. Susan?
Yeah, does anybody have a molecular data on the great myrtle bark scale, which would be similar?
Yeah, that's one of like 2 species of felt scales that actually has the full genome. Yeah.
Is this grass native or is it introduced?
It's a native grass.
Oh.
Yeah, so I've been curious as to whether is this something native or is it adventive? I mean, it's native, but there's also cultivars that come from outside of the country as well. I have a hunch that it's adventive, but I'm not really sure.
You know that that's fundamental grass or
I'm sorry?
It only attacks ornamental grass, can attack other.
Yeah, good question. So I've only found this on the molly grass, specifically this one. Over here. I have checked a lot of other hosts, like Spartina is very similar. It's got the very skinny revolute leaves. Wiregrass as well. I've looked at a lot of wiregrass samples. Haven't found it. Just because I haven't found it doesn't mean it's not there, but it's looking like it really likes this because its body is just made for it. But also scales have been known to form their body to fit crevices of other things. So they might change a little bit depending on what host they're on. I don't know, still figuring it out. You guys can all go look for grass scales for me and bring them to me.
Thank you.
Thank you.
Yes.
Just a quick announcement before the last talk. For those of you that are not staying for the business meeting and are going to be leaving right when the final talk is done, please be aware that after 5 o'clock, and it will be after 5 o'clock in a few minutes, the front public entrance to the museum will be closed. So you will need to make sure you leave out of one of the side doors in either of the stairwells. Those of you from DPI, you know where you're going. For those of you that may be parked out front in the parking garage, you'll head out that stairwell in that direction, go down one flight of stairs. The door to your left will take you outside, make a right, that'll take you to the main parking lot out front. So just please make sure you don't leave through the public entrance. It will be closed. You might activate an alarm. It would not be recommended.
Thank you. Okay, so we have our last talk, Felipe Soto-Adames, with Adventive trips of Florida detective between 2000 and 2024.
Yes, thank you very much. Thank you very much, everyone, for still staying here and listening to me. I promise this will not take much time. So the Adventive trips of Florida, I am limiting this to the years 2000 to 2000. It's not 4, it's 24. Oh my. So 2000 to 2024. There are more than double the number of species in previous years, but I try to do things that I have identified. Adventive species are a problem. They're all for everyone to see. They're a problem for agriculture. They're a problem for natural ecosystems. For example, these two species, Megalurothrix suscitatus, is an important pest on beans. Thrips parvispinus on ornamentals, peppers, gardenias. So the price of your beans and your ornamentals might have gone up in the last few years because of the additional pesticide that is required to control these things. In terms of Stephanothrips, Japonicus, most of what you can see here is Stephanothrips japonicus, and is in some counties is displacing the native fauna of closely related species. Now, you saw that something similar to this before. Florida is a very welcoming place. It has many diverse habitats, many maritime ports, many airports. We have many tourists that come in every year. And so we've received many things. So for this presentation, I'm just going to concentrate on this, exploring these 3 points. The biogeographical origin of the adventive species, whether they're established or not. And to try to answer that, I'm going to look at the distribution pattern in Florida and the pest status of the species, whether they're pests anywhere else and whether they are pests in Florida. For the biogeographical origin, so the easiest way is to look at that species in the context of congenerics or relatives. If it's a species that is distributed in the Southeast US, it's probably native, even though we have not seen it. If the relatives are everywhere else, anywhere else in the continent or other continent, is most likely adventive. And to establish whether, to decide whether they're established or not, I'm considering only specimens that were collected in field crops, community vegetables garden, like the organic garden UF, weedy fields or roadsides, nature parks, permanent landscape plants at private homes and businesses. I'm not including anything that was reported from potted ornamental plants or retail stores in this, for this presentation. And the pest status of the species, look at the literature to see if they're pests anywhere else. And if they're pests in Florida, it's self-evident most of the time. Yes. So for the results, we have 20 species from 2000 to 2024, and that is in 19 genera. Wow. There is only one genus, Megalurothrix, that has 2 adventives. All the other are in different genera. And they're representing 3 families. Heterothripidae with 1 species, Phliothripidae with 6 species, and Thripidae with 13 species. I'm just going to show you a few of the species. This is Heterothrips limbatus. This species was described from the Northeast, from New York. It showed up in a roadside, weedy roadside in South Florida. These are Phliothripids. I just have 4. Podothrips lucaseni. This is a predator. Adrenothrips rosatus and Stephanothrips are fungus feeder that are only in leaf litter. And Holopothrips tabebuia is a pest of tabebuia trees. It makes galls. These are some Thripidae. They're pretty, some of them. Thrips fibrespinus that I showed you before. It's a pest species. Scolothrips is a predator. Trichomothrips is it's not a pest, it's a herbivorous species. Salpingothrips is associated with kudzu. It's found only in kudzu in Florida. Anaphothrips, Syrphidae, this is some grasses. Syrphidae dorsalis is another very important pest. And all these three, Megalurothripsus suscitatus is a pest. Pulmecula serrata is that is a sugarcane thrips. And Frankiniella salviae is on ornamentals. Now, if we look at the frequency of new adventive species, you see there are big gaps. Most of the years we don't get anything. Then the next most frequent is a single species. We have two peaks in 2017 and 2024. 2017 was when I started working here. So I went out and collected. And 2024, because we hire a very enthusiastic inspector in Broward County, and he's collecting a lot. Yes. When we look at the putative region of origin, the oriental region is the highest by a lot. However, the fact that we can trace the species to that region because it was described originally from that region, that doesn't mean that that's the way we got it. It doesn't mean that we got it directly from Indonesia or China or Japan. For example, Pulmecula serrata is all over the Caribbean on sugarcane. So it might have come from somewhere in the islands. Thrips parvispinus, I personally blame the Netherlands because it comes in ornamental plants. The neotropical, we have a few neotropical species. This is Holopothrips. No one knows where Holopothrips came from, but all the species are from South America, so it must have been from there. And Anaphothrips sudanensis, no one knows where it originated, because when people started to study thrips, it was already all over the tropics around the world. Now, when we look at where the thrips were first detected, Miami wins, as we can expect. But then they're scattered all over the state. Most of the counties have only two. So Miami makes sense. All this area here, this is the main agricultural area. You may think, well, Tampa Bay, but what happened with all these other places? Taxonomies, there was a taxonomist out there in Gadsden, Joe Funderburg. And I'm here in Alachua County. And then these three is most likely, they have been introduced most likely with ornamentals. The nurseries can buy plants directly from the Netherlands and other places. So they don't have to go through Miami. If we look at the distribution of the species by county, so all these species that are present in more than one county, this is the number of counties in which the species is found. That is obvious that they're established. But what happens with this group? There are 8 species that have been There are 8 species that were found only once. Okay, we have to go through them quickly. So the first Adrenothrips and Cerathoscopoides, they have been found in only one county, but in successive years. So you can go to those counties and they're still there. So they're restricted in their distribution, but they are there. Megalurothrips, Distalis and Salpingothrips are associated with Kutsu. And there are 2 counties only, Gadsden and Alachua, where the taxonomy starts. And they have been found in multiple years, so they're really established. But all these other species, we have a single identification. So we don't know whether they're established or not yet. The same thing with Scolothrips azura, Holothrips lucaseni, and Galeothrips falciparis. Now, this species, Ctesisothrips striatus, was collected in 2014. It has not been seen again. And that is a neotropical genus distributed in Northern South America and the Caribbean. We haven't seen another individual, so it's most likely not established yet. The other species, we think will have to go back and do more collecting to try to verify whether they're here or not. So in summary, we have 2 species established, 5 we don't know, and 1 probably not. All the other ones, it's pretty certain that they're established by now. If we look at the pest status of all the species, none of them are pestiferous. They are either predators, fungus feeders, or not pestiferous herbivores. But then the other group is different, the other species, the ones that have larger distributions. One, Stephanothrips, is a fungus feeder, it's not a pest. It's found in 7 counties. I'm sure it's more than that. It's a leaf litter species. We just have to sieve more later. Androthrips ramachandrae is a predator, and it's associated with ficus and tabebuya trees. It feeds on the thrips that make the galls on those trees. These three species, Anaphothrips sudanensis, Franklinella salvii, and Fulmiciola serrata, they are not best in Florida, but particularly Fulmiciola is an important pest of sugarcane everywhere else in South Africa and China. But for whatever reason, in Florida, it's peachy. There's nothing happening with it. It may become a pest at some point. Now, these 4 are all pest species. Megalurothrips, Lucitatus is very important pest on beans, Thrips parvispinus, ornamentals and peppers, Syrphidae dorsalis. Everybody keeps expanding the range of plants that is a pest of. Blueberries is the new victim now. And Holopothrips make these galls on tabebuya trees. It doesn't kill the tree, but it looks ugly. So that's not a good ornament. So the summary, if we have 2 fungus feeders, 3 predators, 7 non-pestiferous herbivores, 3 potential pests, and only 4 major pests. So to conclude, most Adventive species originate that we are receiving now from the Oriental region. It's just a historical coincidence, because that's where our trade is. Everything that was going to come from Europe came already 100 years ago or more. The first detection really depends on local collecting effort, particularly for non-pestiferous species. Most Adventive species are not pests, Only 4 of the 20 or so in the last 24 years are pest. And most species appear to be established already. Some of them, if they're pest species, they get established very fast. And I'm gonna leave it at that. Thank you very much.
Thank you, Felipe. Got a moment for one question, if you want, for Felipe.
Question?
There is a comment, if you want. Oh, sorry. No, please go ahead.
I was curious about that potential pest of sugarcane that's found here in Florida. Now, is it feeding on another host, or is it just in very low levels?
It's only on sugarcane.
Yeah.
Yeah.
But it's just in low levels.
So that we were puzzled because in fact, in China and South Africa, that may cause 30% loss in sugarcane yield. But here, it's not doing anything. Apparently, the environmental conditions and the additions that they make to sugarcane are enough for the plant to overcome whatever the thrips is doing. Yeah.
There is a comment for you, Doris.
Oh, well, yes, it is true that they prefer a drier environment, but we have enough pests, thrips in Florida that cause enough damage. There are a small proportion of all the species that have been established from adventive species, but they are problematic enough. Yes.
Thank you, Felipe.
You're very welcome.
All right, this one brings us to the close of the meeting. We are a little bit over the time, but we got a few minutes. I want to say, first of all, thanks to all of you here present, all of you in Zoom, and all the presenters. Without the presenters, this meeting would have never happened. So thank you all. Please, round of applause for you. There was a request by Luc about how many effectively people we have here and online. So just counted, we have online registered, possibly someone got a little bit more, but 55 online and 51 here. So this is bring us to 106. Very successful. Again, we are opening also the options for next year meetings. So if you got some idea already, start thinking about it, and please submit your presentation whenever we're going to ask you for it. If you need the certificate, you presenters, but also if you need, as you are towards graduation and you need something even for attendee, please send Alessandra an email so we can give you a certificate that you participated. There are going to be my signature, which is fantastic. Don't sell it. And please do that. You can find all the email on the program. If you can find it, send us an email as well. Also, the recording will be available. If you have something against a free recording on YouTube, please let us know. We're going to try to work with you. We're going to have in a few minutes the business meeting as well, so you are free to stay. It's usually fun, I would say. So please stay. Please stay, and if you have some ideas, let us know. Yes.
I took a lot of pictures on purpose, so if you don't want your picture in Facebook, you have to come tell me so I delete it.
I would also personally to thank our chief editor of Insecta Mundi and master Zoom recorder and everything, David Plotnick, for being a liaison for us for the Florida Museum, but also for helping us out with this. So thank you again, David. We're going to meet, for those of you who do not want to stay here for the business meeting, and are coming for the dinner stuff, we're gonna meet you at the Bahama Breeze. If you don't know where Bahama Breeze we are talking, at the end of the program, you can see the address, okay? So you will meet us there around 7 p.m. Also, one big thing that we would like to have from you, Alessandra will send out in a couple of weeks, maybe a week, depending on how the stuff goes, a survey for your feedback. What we did good, what we did wrong, what you would like to see in the next meetings, and possibly what would you like to see improved, okay? Also for you on Zoom. So keep an eye out for this. We're gonna try to do that as well. And that's it. I think, thank you again, all of you. David, do you want to say something?
Oh, yeah. So a little bit of a call to action. So some of you know Esther Serrano is my wife, and I'm sure all of us know someone who have been personally impacted by all the federal firings that have been happening. And so one of the things we could do based on the structure of the government, we're kind of limited to what we can do. But one thing that she just came out of a big union meeting, the one thing you can do is contact right to your congressfolk. At this point, this is the only pushback we have left. And so luckily, I have a list of people on the agricultural committee. One of those people is Kat Kemick, whose office is actually located in Ocala and in Gainesville. And so, but the biggest thing we have to do with some of these political folks is really push the economic benefit of ARS and PPQ. And kind of word it in the way, it's not just science, but it's without our efforts, the cost to agriculture and then the added cost to consumers is going to rise. In addition, I think a lot of them don't realize that PPQ is actually paid by port user fees and not actually tax dollars. So I think there's some of that disconnect. Hopefully our voices could help them realize that. So they're more likely to try to push back against the executive branch and at least slow down and perhaps maybe protect PPQ and our ARS brethren and sisters to hopefully stem the tide. So once again, if you want that, I have that information and yeah.
Thank you, David. Okay, thank you, all of you. And with this one, we are gonna have a small break and we're gonna start our business meeting.
6 p.m.
5:30. 5:30.
No, it was supposed to be 5:15.
Yeah, so let's do a 15 minutes break. 5:30. Thank you all. Alex.